Good morning, all. Good morning, all. Welcome all to the 8th World Conference on Pharmaceutical Science and Drug Manufacturing, organized by Bioleagues Worldwide in association with Association of Pharmaceutical Research, APR. So before we begin, I would like to introduce to you our Association of Pharmaceutical Research, which is one of world's leading professional association for pharmaceutical and life science professionals caring for people. APR is a non-profitable professional association meant for research and development in the field of pharmaceutical science and technology. APR is an international forum for researchers, academicians, doctors, and practitioners for sharing knowledge and innovation in the field of pharmaceutical science and technology.
Stop reading. You logging from another system, another
which also be very good.
Good morning, all. I welcome you all to the 8th World Conference on Pharmaceutical Science and Drug Manufacturing, organized by Bioleagues Worldwide in association with Association of Pharmaceutical Research, APR. Association of Pharmaceutical Research is one of the world's leading professional association for pharmaceutical and life science professionals caring for people. APR is a non-profitable professional association meant for research and development in the field of pharmaceutical science and technology. APR is an international forum for researchers, academicians, doctors, and practitioners for sharing knowledge and innovation in the field of pharmaceutical science and technology. APR aims to bring together worldwide researchers and professionals, encourage intellectual development, and providing opportunities for networking and collaboration. APR meets with its objectives through academic networking, meetings, conferences, projects, research, publications, academic awards, and scholarships. APR strives to enrich from its diverse group of advisory members. APR connect pharmaceutical professionals, researchers, exchange global innovation, and act as a bridge between researchers and academicians by organizing international conferences, world conferences, faculty development programs, providing memberships, establishing chapters, faculty exchange program, and implant training programs.
Welcome to Bioleagues Worldwide, prominently promotes research and development and have brought a revolution in the field of worldwide conferences. In this video, we want you to dive into our world, giving yourself a clear insight of the Bioleague events. But first, who are we, and what do we do? Bioleagues Worldwide, BLW for short, is one of the world's largest non-profitable professional associations operating under the Techno-Read Research and Development Association, DRADA, meant for research and development in the field of engineering, science and technology. Bioleagues Worldwide conferences bring together the professional wizards and leader bodies for scientific professionals in India and across the globe, established in 2000. We extend to every part of the globe, with more than 28,000 professional members and 34,000 student members. Our frowning membership at Bioleagues led to an increase in members from Europe, Asia, Australia, and Africa, fostering networking opportunities that strengthen ties within and across countries and technical communities. Bioleagues works with the motto of creating a better tomorrow by organizing conferences and creating a network which will help grow a better tomorrow with the help of advanced technology and achieve sustainable development. Mission: to connect professionals at an integrated platform for growth to divert knowledge through our professional activities and scientific conferences. Vision: of a united platform to explore research with opportunity to innovate multidisciplinary scopes and applications of professional studies. Bioleagues widely connects researchers, exchange global innovation, and acts as a bridge between other academicians by organizing international conferences, world conferences, faculty development programs, webinars, seminars, providing membership, and more. Some of our conferences include Breast Cancer Dubai, 7th World Conference on Pharmaceutical Science and Drug Manufacturing, Nanosphere UAE, EDUMIS, Indo-Oncology Summit, International Conference on Dermatology and Cosmetology, and many more. Every organized scientific events in association with renowned universities done in the past, with over 28,593 journal publication services, creating opportunities to get your research paper published in high-impact factor journals such as Scopus Indexed, Web of Science, Google Scholar Indexed Journals, SCI, and many more. To make our operations smoother and more perfect, we have been supported by our associated organization, International Association of Oncology, IAO, Universal Society of Food and Nutrition, USFN, International Association of Neuroscience, IAN, International Society of Environmental Relationship and Sustainability, ISERS, Association of Pharmaceutical Research, APR, Society for the Research and Studies of Nanotechnology, SRSN, and many other research associations. Actively upcoming, Association of International Society of Environmental Relationship and Sustainability, ISERS, release upcoming conference Climate Change Malaysia. We at Bioleagues believe that there's always a better way to treat professionals, and that is what we do. If you would like to explore with Bioleagues, visit our homepage www.bioleagues.com. Have Bioleagues your way. Welcome to Bioleagues.
Association of Pharmaceutical Research and Bioleagues Worldwide is organizing today the 8th World Conference on Pharmaceutical Science and Drug Manufacturing. today and tomorrow, the 18th of April 2021. This conference is a multidisciplinary program with broad participation with members from around the globe, focused on learning about pharmaceutical science, research methodologies, formulations, manufacturing, and its advances. We have invited speakers and delegates from all the world. This creates a perfect podium for networking as it brings together renowned speakers and scientists across the globe to the most exciting and memorable scientific event filled with informative and interactive sessions, international workshops, and world-class international exhibitions and poster presentations. Experts and scholars from drug manufacturing and pharmaceutics background will be showcasing their scientific work, research, and emerging technologies on the following topics, such as pharmacology, pharmaceutical biotechnology, pharmacogenomics, drug delivery systems, biodrugs, pharmacovigilance and drug safety, pharmaceutical microbiology, pharmaceutical research and development, pharmaceutical analysis and quality assurance, biotherapeutics, radiopharmaceuticals, vaccine design, formulation technologies, clinical pharmacy, industrial pharmacy, pharmaceutical chemistry, and pharmaceutical science and technology.
Guru, then after the year. What happened? ओ भाई उमा का चल गुरु काम धंधो करो छे नहीं
Next, I would like to thank and welcome to you our supporting partners, Supreme Pharma Tech. Supreme Pharma Tech is a modern nutraceutical factory specialized in development and manufacturing of a wide range of products from ingredients of natural origin. The company is operating 10,000 square meters facilities which are partially powered by solar energy. The production process is automated at maximum level and complies with GMP regulations. All company products are certified by the Thai FDA. The company also passed US FDA registration beginning of 2021. Supreme Pharma Tech is one of the first companies in the world to develop and implement liposomal nanotechnology on production scale. Liposomes can manipulate the solubility of encapsulated active ingredients, as well as protect from degradation and even achieve delivery to specific sites. Oral liposome delivery system is capable of bypassing the gastric system and delivering nutrients to the target cells and tissues in almost full digested amounts. Supreme Pharma Tech is one of few certified factories in Thailand for processing hemp. The company had developed its own of hemp oil with increased bioavailability. Next, I would like to introduce to you Complete Pharma, which is a Finnish Thai factory of food supplements with modern facilities equipped with automatic pharmaceutical machinery. Production process is strictly followed GMP standards, raw materials conforms to international standards. Complete Pharma is involved in research, development, and production of modern nutraceuticals which provide health and medical benefits. We carefully study the latest achievements in related fields and apply up-to-date knowledge to all our formulations. Each and every ingredient Complete Pharma uses is clinically approved for its efficacy and safety. Receiving high-quality standardized ingredients from reliable suppliers in USA, Germany, India, Malaysia, Thailand, China, and other parts of the world enables Complete Pharma to produce high-potency products with maximum health effect. By combining together knowledge of ancient European, Thai, Chinese, and Indian medicines with modern knowledge of nutrients, Complete Pharma is among the most unique producers in the world. In Complete Pharma, we believe that the world health is part of our life, not a word of self-promotion. Complete Pharma does business with honesty and believe in responsibility towards customers who need assistance and help. Next, I would like to welcome all our honorable dignitaries who have joined us here. Firstly, I would like to welcome our chief guest, Giovanni M. Polotti, the Associate Dean and Henry Pfizer Chair from Department of Pharmaceutical and Administrative Sciences, University of Health and Science and Pharmacy in St. Louis, Missouri, USA. Next, I would like to welcome and thank our conference chair who has joined us, Dr. Anantan Naik Nagapaka, the Director and Principal from Amity Institute of Pharmacy, India. Next, I would like to welcome Mr. Rudra Satpathy, the founder and head of Technorate Groups. Next, I would like to welcome our eminent keynote speakers who have joined us for the day. Firstly, I would like to welcome Dr. Monica Gulati, Senior Dean from Lovely Professional University from India. Next, I would like to welcome Professor Marcel Muzaffari, the President and Founder from Australian Nanoscience and Nanotechnology Initiative from Australia. Dr. Sergey Shoko, the Director and Professor from Shenoy University from Russia. Dr. Ranendra N. Saha, the Director, and Shri B.K. Birla and Srimati Sarla Birla, Chair and Professor from BITS Pilani, Dubai from United Arab Emirates. Dr. Animesh Acharji, Senior Scientist from University of Brigham and NHSA from United Kingdom. Next, I would like to welcome our session chairs of the day. Firstly, I would like to welcome Professor Dr. Subhash Chandra Dinda, the Professor and Head of Pharmaceutics from College of Pharmacy, Mahavir University, India. Dr. S. Duraivel, the Dean from Sri Venkateshwara College of Pharmacy from Pondicherry, India. I would like to thank all our dignitaries as well as the participants who have joined us even during this pandemic situation and for imparting their expertise with us. I would also take this opportunity to welcome all our presenters who have joined us from across the globe, who have spared their valuable time in enlightening with us their latest research studies. So firstly, I would like to welcome our chief guests who have joined us here. Giovanni M. Polotti, who is Associate Dean and Henry Pfizer Chair, Department of Pharmaceutical and Administrative Sciences from University of Health Sciences and Pharmacy from St. Louis, Missouri, USA. I would like to speak a few words about our chief guest. He has been serving as the Scientific Secretary of the International Pharmaceutical Federation, providing leadership and strategic direction for this global federation on scientific aspects relevant to pharmaceutical science, practice, and education. Dr. Polotti has received his MS in Pharmacy and PhD in Biopharmaceutics and Pharmacokinetics from the School of Pharmacy at the Swiss Federal Institute of Technology in Zurich, Switzerland. He directs an interdisciplinary research program focusing on novel drug delivery system. Dr. Polotti has published more than 160 scientific papers and is an inventor on 9 US patents. Thank you so much for joining us today. It's an absolute honor to have you here.
Thanks so much for the kind introduction. It's certainly a privilege for me to participate in this esteemed event, and I greatly appreciate the time for me to share a few comments with the audience here. As you mentioned, my role here is Scientific Secretary of the International Pharmaceutical Federation, or FIP for short. And in that role, I'm responsible for facilitating the scientific exchange between our 146 national member organizations, which include professional pharmacy as well as pharmaceutical sciences organizations, and more than 180 academic institutions that are dedicated to the training of future pharmaceutical scientists. Overall, FIP represents more than 4 million pharmacists, pharmaceutical scientists, and pharmaceutical educators worldwide. In addition to this role, I also serve as the scientific liaison between our federation and the World Health Organization, as well as the United Nations Educational, Scientific, and Cultural Organization, UNESCO. And so you can see that our organizations share quite a few common objectives, and I hope we can collaborate on specific projects in the near future. The theme of the 8th World Conference on Pharmaceutical Sciences and Drug Manufacturing is recent technologies and challenges in the pharmaceutical industry due to the outbreak of COVID-19. And we have to acknowledge that all our lives got touched by this global pandemic. What's unique to our profession, however, is the fact that COVID-19 brought pharmaceutical sciences to the mainstream media and consequently to the general public. Suddenly, topics such as repurposing of drugs, vaccine development, clinical trial design, and regulatory review were discussed during primetime evening news. This is great visibility for pharmaceutical sciences and an opportunity to highlight the relevance of our research activities. Now, we also learned that this opportunity comes with challenges. How do we explain to the public what an acceptable risk of adverse event is without instilling fear? And how do we project confidence into an innovative technology such as mRNA vaccines without amplifying concerns that lead to greater vaccine hesitancy? COVID-19 also shed light on some of the hidden risks in pharmaceutical development that can quickly lead to national and regional crisis. And this includes like the impact of a pandemic on academic and corporate research due to lockdown of laboratories or selection of essential and non-essential research activities. The impact of COVID-19 on our supply chain of medical product, which can lead to drug shortages due to limited manufacturing or distribution capacity, or national prioritization of medical products that lead to altered import and export regulations. So over the next 2 days, you will be able to learn about new and exciting developments in pharmaceutical sciences. From the experience with COVID-19, I hope that we can all consider questions relevant to access and equity when we advance those innovative technologies towards market approval. With that, I wish you a successful conference and thank you for your attention.
Thank you so much for joining us and for your kind words. Irrespective of your busy schedule, you have come and spared your valuable time and sharing time with us. Thank you so much. So with this, I would like to welcome next our conference chair, Dr. Anantanayak Nagapaka, who have joined us here. Before that, I would like to inform that with our chief guest, Dr. Giovanni, will be releasing our conference proceeding today. Next, I would like to welcome Dr. Anantanayak Nagapaka, the Director and Principal, from Amity Institute of Pharmacy, India, to speak a few words on this occasion. So are you able to hear us? Next, I would like to welcome founder and head of Technorate Group, Mr. Rudra Satpathy. Next, I would like to welcome our eminent keynote speaker, Dr. Monica Gulati, Senior Dean from Lovely Professional University, India. Dr. Monica, can you hear us?
Thank you. Good morning, and thank you for inviting me here.
Yes.
Happy to join.
Thank you so much. Eagerly waiting for your expertise.
Me too.
Next, I would like to welcome our eminent keynote speaker, Professor Marcel Muzaffri, President and founder from Australian Nanoscience and Nanotechnology Initiative from Australia. I would like to give a short description about our keynote speaker, Professor Muzaffri. He has graduated from the School of Pharmacy and Chemistry from Liverpool John Morris University in 2015. He has authored more than 100 publications, including 3 patents and 3 books, which are among the first books in the field of pharmaceutical nanotechnology and nanobiotechnology. His book, Nanoliposomes and Fundamentals to Recent Developments, is the very first book on liposomes, nanoliposomes ever written. Professor Muzaffri possesses 20 years research and teaching experience both in the academic as well as the industry in different countries, including England, Turkey, New Zealand, and Australia. Next, I would like to introduce to you our keynote speaker, Dr. Sergey Shukov, the director and professor from Shinoy University, Russia. He has attained his postdoctoral degree in at National Institute of Immunology from Russia. And from 1989 through 1995, he was the head of the lab of clin immunology at Moscow. From 1995 through 2004, he was the chair of the Department of Clin Immunology at Moscow Clinical Research Institute. And in 1993 to 1996, he was the secretary-in-chief of the Edit Board, Biomedical Science, an international journal published jointly by the USSR Academy of Sciences and the Royalty Society of Chemistry, UK. At present, Dr. Sergey is the president and CEO of the International Center for Personalized and Precision Medicine in MedStar at Russia. And he's also the professor of Department of Human Pathology, Shinoy University, and Department of Clinical Immunology at Moscow, Russia. He's also the Secretary General, United Cultural Convention, Cambridge, UK. Next, I would like to welcome and introduce to you our keynote speaker, Dr. Ranendra N. Saha, the Director, and Shri B.K. Birla and Srimati Sarla Birla Chair, Professor BITS Pilani, Dubai, from UAE. In 2011, he has been awarded the Shri B.K. Birla and Srimati Sarla Birla Chair Professorship at BITS Pilani for contributing in teaching and research. He has vast experience in the field of pharmacy, especially in pharmaceutics, novel drug delivery systems, and pharmacokinetics. He received Pharmacy Professional of the Year 2013 award given by Indian Association of Pharmaceutical Scientists and Technology. He is also a recipient the Best Pharmacy Teacher Award for the year 2005. He has more than 33 years of teaching, research, and administrative experience. He has published more than 90 papers in international, national journals and presented more than 100 papers in conference, seminar, symposiums. He has also delivered several invited lectures at seminars, conferences, universities in India as well as abroad. Next, I would like to welcome and introduce to you Dr. Animesh Acharji, Senior Scientist from University of Brigham and NHS from United Kingdom. Dr. Animesh has earned his PhD from Wageningen University in Statistical Machine Learning applied on omics analysis. After his PhD, he moved to Lyon for his postdoctoral study with Synergy Lyon Cancer Center, a statistician. After his postdoctoral study, he was offered the position with BASF Plant Science, Gent, Belgium. Before joining the University of Brigham and Queen Elizabeth Hospital, he worked at the University of Cambridge, focusing on metabolic-driven diseases like obesity, type 2 diabetes, and liver cancer using high-throughput transcriptomics, metabolomics, lipidomics technologies. His research interest includes high-dimensional analysis, biomarkers, discovery quantum genetics, and network biology. He has been granted several international fellowships from many organizations such as International Society for Computational Biology, Newton Katib Celebi Fund, Sao Paulo Research Foundation, British Council, and Dutch government. Once again, I would like to welcome our session chair, Professor Dr. Subhash Chandra Din, Professor and Head of Pharmaceutics, College of Pharmacy, Mahavir University, India. Tirthankar Mahavir. Next, I would like to welcome Dr. S. Duraivel, Dean from Sri Venkateshwara College of Pharmacy, Pondicherry, India. Before moving on to the technical session, I would like to inform you all. Today, we'll be discussing about our technical sessions will be handled by 9 tracks. We'll be discussing about COVID-19 drug development, drug delivery technologies, drug targeting and drug formulation, drug design and development, pharmaceutical formulations. We'll be discussing on the following on day 1. So next, I would like to begin with our keynote session. Firstly, I would like to welcome Dr. Monica Gulati, who has joined us here.
Hello.
Thank
Hello.
you.
Good morning, sir.
Good morning.
Yes, our conference chair has joined, Dr.
Hello.
Anit Naik Nagpaka.
Just a
Are you
moment.
able to hear, sir? So we are beginning with the keynote session, and our keynote speaker, Dr. Monica, has joined us here. So before that, would you like to address our keynote speakers and the participants who have joined us here?
Good morning, sir.
So can you please unmute?
Yeah. Yeah. Can you hear me?
Yes, yes.
What is the time limit given to the speaker?
So you may
Thank
just speak
you.
a few words and address our participants and keynote speakers who have joined us here. Dr.
What
Monica
is
will
the
be beginning this keynote session.
what is the title of the what is the title of the session? Sorry, yeah.
We are moving to the technical keynote session, sir.
What is the theme of the session? Just I want to know about it. Please. What is the
The session would be, for my session would be basically biotherapeutics. You know,
biotherapeutics?
the
Yeah, I know.
Yeah.
Yeah. Okay.
Okay, so we'll begin with Dr. Monica, ma'am.
Just a moment. The biotherapeutics, although very old, and it has updated and it came into the very recent therapies. Biotherapeutics involves all gambit of starting from insulin therapy to the today's monoclonal antibodies and all those things. And biotherapeutics, although very expensive right now, it would be a saver for many diseases, and it is going to herald a new era in the therapeutics. Okay, madam, you can start your session now.
Thank you, sir. Thank you. So I hope the audience can hear me.
Yes, ma'am. You may
Good
begin.
morning to all. And, as Dr. Giovanni shared and Dr. Nagappa also shared, we are in the middle of a pandemic, in fact, the second wave of pandemic. And, every day the strain is changing, or probably our perceptions are changing, the scenarios are changing, and so on. And what was considered to be very effective a year back is no longer considered effective. What was considered to be the mode of transmission a year back is no longer considered to be the current mode of transmission of the the current strains which are there. So we are left simply wondering what is right and what is not right. And somewhere, despite all the vaccines and therapies which are available, still we are skeptical, still we are scared, and we are still looking for certain things that will help us in prophylaxis, more of prophylaxis and then of treatment, of course. And what I wanted to share at this platform is that apart from the conventional vaccines and other treatments which are going on, there are a few unexplored kind of things also, which to some extent can be tried to increase, enhance our immunity. So I wanted to share it at this platform so that the younger generation people who are there amongst the audience would take that home. And second thing, I also believe that there are many people who can get the things moved, the movers and shakers of the pharma profession who are there. For these kind of unconventional therapies, though the clinical trials are going on, but the limitations that we are facing as scientists is that our labs most of the times are not equipped enough to handle the COVID virus. You need biosafety facilities of the utmost grade, and most of the labs, most of the facilities available, at least in our country, are not equipped enough to handle that. So what I wanted to convey to the movers and shakers of the profession is that these kind of therapies should also be given some kind of attention, a little more rather, so that we might be able to prevent at least the pandemic by 5%, 10%, or whatever is possible. So I'll start right away with my presentation. I'll share my screen and see if you people can see my presentation. I'll be very quick. So can you see the screen, please?
Yes, yes, it's visible.
Yeah. So I'll be quick with it. I know my time is limited only. So we are going to talk about the gut microbiota, the microbes that live in our gut. And, they create such wonderful substances inside our gut that we can harness them to increase our immunity. We can harness them to, treat many diseases also, which are not only in the originating in the intestine, but extra-intestinal also. Currently, I'm going to talk about this, the gut microbiota, how we can harness the gut microbiota. So we all know the current strategy which are being used are vaccines, then convalescent plasma, which is, I keep on hearing is in shortage at certain cities and in certain parts of the country. Then repurposing of the antivirals. As Dr. Giovanni also said that, we are repurposing the drugs. This is the time to repurpose. Then thromboprophylaxis also, because, it has been found that microclots are being formed by the virus at very strategic parts of the body. And then finally, we are also trying in the severe cases, we are also trying the recombinant monoclonal antibodies. So I am going to focus on the non-pharmacological prophylaxis as well as therapies by harnessing again the gut microbiota. So, ancient times, in India also and in other parts of the country, this is the statement by, the one of the biggest of the scientists or philosophers, that death sits in the bowels, a bad digestion is the root of all evil. So somewhere, it was recognized by the ancient scientists also that our gut has a capacity to, introduce the disease and treat the disease also. So then we talk of dysbiosis. Dysbiosis is basically, the microbes that stay inside the gut. They are in a lot of abundance. They are there in large numbers. They are there in large diversity. So dysbiosis is basically, when this equilibrium gets disturbed, and then the pathogenic bacteria, they increase in the numbers and in ratios. The good bacteria, they do go down in ratio, and then there are certain good bacteria which simply disappear. And of course, the diversity of the bacteria, the diversity of the, not bacteria, I would say, it contains virome, it contains bacterium, it contains fungome, it contains archaeum. So all these inhabitants of the gut, their diversity gets decreased, both alpha and beta diversity. Let us not enter into too many details. And then, of course, there are many metabolic diseases like obesity, like diabetes, which are, and even people say that Alzheimer up to, there is a gut-brain axis also. But let us now focus on the infectious diseases only. So the infectious profile of the dysbiosis, when dysbiosis occurs, when there is a disturbance in the equilibrium in the, microbiome of the gut, it can lead to these kind of infections. I quoted all of them. It can lead to sepsis. It can lead to hepatoinfections. HIV can be there. Skin and skin structure infections can be there. UTI can be there. Then upper respiratory and even lower respiratory tract infections are there. Enteral, of course, is, it is gut which is involved. So since, the equilibrium lost in the gut leads to these kind of infections. So scientists eventually thought that if that equilibrium is restored, probably, it will help in the treatment of these infections also. And HIV infection treatment being done by, restoring the eubiosis from dysbiosis gave a lead that, we can try this in the viral infections also. And then, there was a wonderful thing that came into light, that gut-lung axis exists in the body, in which, we say about 10 years back, even I used to think that lungs are sterile, but it is not. I was also surprised to know. And, there is a crosstalk between the lung and the gut microbiome. Of course, the lung microbiome is much smaller as compared to the gut microbiome. The lung microbiome has very selective kind of inhabitants, while in the gut there are numerous. The ratio would be, I would say, 1:100. And then, the lung gets basically its microbiome either from the air that we inhale, or maybe from the oral tract, or some of it, from the gut also, when microaspiration is there. Whenever you cough, or whenever there is something gets stuck in your throat, so microaspiration keeps on taking place. So microaspiration, air inhalation, and something from the oral microbiome also enters into the lungs. Now, apart from the lung having its own microbiota and its own ecosystem, alterations in the gut microbiome also affects the lung microbiome. So gut microbiota play a protective role against bacteria and viral pulmonary infections by regulating the innate and adaptive. Both the immunities are regulated by the gut microbiota. So if the there is a dysbiosis, that means the equilibrium is disturbed, the immunity goes down. So it is of utmost importance that in these days when we all need, our best kind of immunity, we must take care of our gut microbiota. So innate as well as adaptive immune responses, they go down if the gut microbiota equilibrium gets disturbed. Certain pathobiomes are recognized by the host immune cells. What is happening these days is everybody is scared of the cytokine storm. People say that, we were able to save the patient till a time, but then, there was a cytokine storm and we lost the patient because the body reacts very, very aggressively to it. So certain pathobionts are there. Pathobionts means the microbes which are pathological and that stay in the gut. They are recognized by the host immune cell, and that results in the cytokine release. And then the patients with chronic, this has been found that chronic disorders like asthma, like COPD and cystic fibrosis, they have higher incidence of IBD as well as IBS, and vice versa. The patients of IBD and IBS have higher incidence rates of asthma, COPD and cystic fibrosis. So there, this simply, makes sure that there does exist a crosstalk between the lung microbiota and the gut microbiota. So what kind of dysbiotic changes are occurring in the COVID infection? There it has been found, there have been, I've gone through about 11, 12 papers in very good journals from very good groups, which say that during the COVID infection, there are dysbiotic changes in the gut. So there is a decrease in the number of bacteria as well as fungi, yeast. There is a decrease in the diversity of both bacterium and fungum, and there is a decrease in the number of healthy bacteria and the healthy yeast species, and there is an increase in the number of harmful bacteria. They've given the entire chart, which species increases, which species decreases, but since we didn't have that kind of time, so I just gave the overall kind of picture. So, it is still under recognized, I would say. We are not focusing much on this part of the prophylaxis as well as treatment, though we know that microbiota health status and its alteration play an important role, albeit it is under recognized. And, we keep on wondering, I was also there, person was also there, XYZ were there, but only X caught the infection, Y and Z did not. Right, we were in close vicinity, 5 of us were there, but only 2 caught the infection. So it is somewhere, our innate immunity and our resilience and our susceptibility is somewhere being regulated by the gut microbiome. So then we'll discuss about the non-pharmacological options. We have probiotics. We all of us have heard about them. We've seen. We are taking curd most of the time. So that contains the live microorganisms. So probiotics are the live microorganisms. Then we have prebiotics. Prebiotics are basically the fiber containing food items, which are actually the food for bacteria, food for good bacteria, I would say. But they are not live. Then we have symbiotics, which are a combination of live as well as non-live, because they contain both these. They contain the probiotics and they also contain the food for the probiotics, so that ensuring that they are supported inside once they reach the gut. Then we have metabiotics, which are basically the non-living kind of products which we get from good bacteria, probiotics. They are basically they are metabolites, or the cell wall parts, the fragments of the cell wall. And it is said that they are almost as effective as the probiotics. The only advantage is, since they are non-living, so we do not have any kind of risk. And then finally, we have fecal microbiota transplant, in which, the fecal matter of a donor is transplanted into the gut of a recipient. And it has also been found, in fact, this treatment option has been found to be 90% effective in Clostridium infection, the chronic Clostridium infection. And it has been found to be effective in COVID also. And a few clinical trials are still going on. So let us move to each one of them one by one. So first I would talk about probiotics. So as you can see, even when we are healthy, the probiotics enhance our immunity, right? So they reduce the susceptibility to infection, including the COVID infection. When we are sick, they help us recover. And when we are in the old age, basically our systems have gone down. Even then, the probiotics are quite supportive for the geriatric patients also, geriatric population also, because they, confer that kind of advantage, which only by increasing the immunity, the innate immunity also, adaptive also, and keeping your, since gut-brain axis is there, keeping you very attentive also, so preventing Alzheimer and dementia and everything, right? I'll move on quickly. I know my time is running short. So how do they act? Once we talk about the mechanism of probiotics, it will cover the mechanism of all others, because I'm sorry for the error in spellings. Because everything else is dependent upon the good microbiota only. So when you have good microbes, so the competition of the dietary ingredients is there. So of course it is going to suppress the pathogenic microorganisms. They lead to the bioconversion of sugars into fermentation products, like you have monounsaturated fatty acids, you have short chain fatty acids, which are very, very good for our health. And then there is production of growth substrates. For example, they lead to vitamins, production of vitamins, especially vitamin K and vitamin B. They are produced by the good gut microbiota. Then they antagonize the bacteriocins produced by pathogenic microorganisms. Their own bacteriocins are going to kill the pathogenic microorganisms and vice versa, right? So of course then they will also compete for the binding sites inside the gut, right? So this is the way they are going to act. Then I talk of use of probiotics in COVID. So prophylactic and therapeutic use has been demonstrated already. You can, if you want to see, you can see this reference. Of course, it is from a very recent one only. I think all the papers talking about COVID would be recent ones only. And then, all the probiotics are not equally effective, because there would be certain bacteria which would be effective in COVID infection. There would be certain bacteria which would be effective in Clostridium infection and so on. And, there is a clinical trial going on, which I would like to share with you. There is a combination of Lactobacillus species and Pediococcus species, a combination of these two, which is in clinical trials as an adjuvant therapy for COVID. So if anybody wants to see what is going on, you can look at this reference and find it out. Then we come to prebiotics. As you can see in the picture, these are the probiotics, and this is the food for probiotics. So it is basically, allowing the probiotics or the good bacteria in the gut to thrive. So prebiotics are the non-digestible food ingredients. Our body cannot digest it, so it goes to the colon only, and the bacteria in the gut, the microbes, they are able to digest them. What we are not able to digest them, and they are, if we talk of definition, they have some beneficial effect on our health, and they lead to selective stimulation of the bacteria. The good bacteria only will be stimulated, and they, of course, the ultimate is they improve the health of the host. So both the probiotics and the prebiotics, they act on the gut microbiota. They pass through the stomach, they pass through the intestine, and they reach the colon. And they are non-absorbable oligosaccharides, so they are not absorbed from the gut and reach colon. And live bacteria also reach the colon, and there, together they improve the barrier function of the gut. They suppress the pathogenic bacteria and they enhance our immune system. So I think I've already covered this. Yeah. So how are they effective in COVID? They lead to changes in the intestinal microflora. Ultimate role, the heroes are the microbes only. These are the supportive substances. These are going to allow them to thrive, and they operate through the support of useful bacteria, either native to the gut bacteria which are good, or the gut bacteria which have administered in the form of probiotics. So there is a very beautiful article in the journal Nutrients, which talks about the role of prebiotics. They call it in the role of nutrition in potential mitigation of COVID-19, they talk of the fibrous substances, the polysaccharides which are not absorbed by the human gut. Then we talk of synbiotics. Synbiotics, as the name suggests, it is a combination of probiotics and prebiotics. So a part is live, a part is not live. So these, as I said, are the mixtures of probiotics and prebiotics, and they affect the host beneficially and allow the implantation of the live microbial culture. They are now probiotics, when we have say, if I have two children, one of my child will like probably salty substances, the second one would probably like sweet substances. So that is the case with bacteria also. One good bacteria will like some other kind of prebiotic, and another good bacteria will like some other kind of prebiotic as a food. So the combination has to be selected very, very carefully. The food on which a particular bacteria thrives upon, right, that combination has to be selected and that combination has to be given. For example, I've taken one example also. So, fructo-oligosaccharides are given in conjunction with the bifidobacterial strain, and lactitol is given in conjunction with the lactobacillus strain. So they have, they combine the advantages of probiotics as well as prebiotics, and they are known to reduce the typical symptoms, the GI symptoms of COVID, which include anorexia, nausea, vomiting, diarrhea, and abdominal discomfort. And for synbiotics also, 2 clinical trials are going on, are underway to assess the effectiveness of synbiotics. So if anybody is interested, you can see these 2 references to find out the status of the clinical trials of synbiotics for COVID. Then there is something which is very interesting. It is claimed that, the metabolites of good bacteria, of good microbes, they are equally effective as that of the probiotics. Now, only disadvantage is that they'll not replenish themselves. The bacteria or the fungi, being we have Saccharomyces or you have lactobacilli, they are going to multiply themselves. So you give them once, you can wait for a few days, they'll keep on replenishing themselves. With metabiotics, that advantage is not there. But another very big advantage with metabiotics is that risk of introducing the live microorganisms into your body, that goes. So chances of any kind of contamination or any kind of overgrowth or too much of competition or pathobionts being introduced erroneously, that goes. So metabiotics are basically the cell-free extracts of the probiotics. They are probiotics and prebiotics are mixed, they are allowed to ferment, and the supernatant is taken so that it does not contain any live cell. Advantage is they are measurable, they are analyzable, and they are not live. And generally, what do they contain? They contain short chain fatty acids, which are all know that, which are very, very good for our overall health. Polyunsaturated fatty acids, they contain their own bacteriocins. They contain polysaccharides, amino acids, plasmalogens, vitamins, and aptamers. These are the main constituents of metabiotics. Now, how do they act? They decrease the colonic pH, so they allow the good bacteria to thrive. They inhibit the growth of pathogens. They improve the integrity and function of colonic epithelial cells. This is very, very important because then, toxins are not able to travel into the body. So they enhance the immune function. They have pro-apoptotic properties and they have antioxidant properties, and then they also lead to formation of vitamins. And then their role, you can see that probiotic metabolites. This is one very beautiful article on the role of probiotics. They're also known as postbiotics. So you will have to switch between the terms metabiotics and postbiotics. Sorry, metabiotics and postbiotics. So these are the common conditions in which the postbiotics have been found to be very effective. Just I gave to share, that they have been found to be very, very effective. And then we are going to talk about the fecal microbiota transplant also. As I told you that, the poop from one donor is transferred into the gut of the other. And it has been found to be very effective in certain infectious as well as non-infectious diseases. So you can see all the diseases in which it has been found to be effective. RCDI, the recurrent Clostridium difficile infection. IBD, diarrhea, IBS, constipation, obesity. These are the ones which are associated with the gut, and then non-gut disorders like metabolic, diabetes, Parkinsonism, Alzheimer, depression, gout, multiple sclerosis. Then comes COPD, the one which is related to lungs, and then arthritis. A lot of data has been generated where the effect of FMT has been demonstrated in all these conditions, and they are available in the market also by a company which gives you a free freeze dried composition of FMT. The name of the company is Open Biome, and they call it donation center. They have a donation center wherein the donors come, their tests are done for all kinds of infections. When they are found to be satisfactory, then their fecal matter donation is taken. It is processed and finally it is freeze dried, and they are available for the different kinds of deliveries. You can they are available as capsules also. They are available for administration through the upper respiratory tract, upper GI. They're also available for administration through colon. So this is another option which is available, and it has also been found to be effective in COVID. This is the paper which says that convalescent fecal microbiota transplantation is the possible treatment of COVID-19. It has been found to be effective in a small set of patients. And then, a clinical trial is also going on. You can visit the clinical trial site also to find out more about this. Thank you. That's all from my side. So I'm open to any questions.
Participants, if you have any questions, you may ask now or you may write down in the chat box. Thank you, ma'am, for your presentation.
Thank you.
So as we are getting short of time, we will quickly move on to our next keynote presenter who have joined us here. Next, I would like to welcome Professor Marcel Muzaffari, President and founder from Australian Nanoscience and Nanotechnology Initiative from Australia.
Good morning from me. At present, I am in Thailand. Good morning. I want to thank the organizers for all the hard work they went through to have this scientific meeting.
Thank you so much, sir.
Right. So you let me know when do you want me to start my lecture. I'm waiting to be told when shall I start my lecture.
I think you can begin, sir. Dr. Marcel,
All right.
you can begin.
Okay, good day, good morning, and good afternoon, depending on the time zone you are in. My topic of presentation is innovative theranostic solutions for green tech pharmaceutical manufacturing. And as I mentioned in the abstract of this presentation, in the next few years, biopharmaceutical and nanobiotic industries are expected to be reshaped, revolutionized. It is anticipated that biopharma and nanobiotic companies will continue to develop.
Professor Muzaffari, I think is the audio clean? We cannot hear you.
I don't know what happened to the voice system. May I continue? Right. Okay. Can you hear me?
Yes, I can hear you.
Okay, I continue. There was a delay due to a technical problem with voice system. It is anticipated that biopharma and nanobiotic companies will continue to develop new strategies to diagnose better, treat, and cure a wide range of traditional, already exist, and new pharmaceutical challenges, including COVID-19 and managing, preventing by the means of vaccination, SARS-CoV-2 and associated viral pathogens. Many people talk about viruses, but I want to grab your attention to my transcript going back to 1993. Do I have a cursor to show? Yes. And as you see, I took the course virology 28 years ago. And virology was one of the many courses I lectured to PhD students. So I know very well about the virus, its pathogenesis, its molecular biology, its genetical construct, and the theory, at least, behind how to make vaccines. And in practice, we have also developed oral functional food products approved by Ministry of Health Malaysia to prevent or minimize the susceptibility to viral infections and to boost our immune system. The entire pharmaceutical sector is in an urgent need for both innovative technological solutions and fundamental scientific studies at R&D level, enabling the production of personalized, efficient, and smart, or otherwise known as targetable, drug products, industrial scale manufacturing of complex theranostic drug delivery systems with minimal or no side effects using the existing technologies is challenging. Here we see a list of some of the challenges currently we face in pharmaceutical industry manufacture. Exposure of graduate students to industry, a majority of new graduates or even people with experience of postdoctoral fellowship, they lack sufficient industrial experiences. A consistency between animal studies and the real clinical outcome of the drug that passing through R&D phases, adequate budgeting, managing batch-to-batch consistency of raw material. In the last 2 years, because of the pandemic, many of companies supplying ingredients, they reduce their manufacture. So this is a burden, this is a challenge for pharmaceutical and food supplement manufacturers to receive what they have been working with for a number of years. The next challenge would be byproduct and waste management. Chemical challenges, we will talk about this more in the next slides. Problems with solvent use, mechanical challenges, and health and safety issues. Improving manufacturing sustainability is one of the challenges that we are facing in pharmaceutical manufacture. And as you can see in this slide, pharmaceutical manufacture typically has more than 100 kilograms. This is a typical statistic from GSK pharmaceutical company. Raw material consumption per kilogram of product. Disposal costs, infrastructure costs, and energy bills. We need to find efficient ways to somehow convert the byproducts to cash flow. We need to budget very precisely and accurately, otherwise consumers have to pay a lot from their pocket for prevention, for vaccination, and for treatment. The next slide shows main problem that we are facing in manufacture of biotech products, particularly with respect of green manufacturing. We need to maximize resource efficiently with respect of energy and mass, eliminate and minimize hazards, pollutions, giving wastage to environment should be seriously managed, design systems using life cycle analysis thinking. This is from a publication in Journal of Green Chemistry, which explains more about key green chemistry research areas and perspective from pharmaceutical manufacturers. And you can see that for particular products of the emphasis of the scope of this paper, we see that we need to do a thorough investigation at atomic level to see that how we can improve the efficacy of active pharmaceutical ingredients with no or minimum extra budget burden, particularly for consumers. And the next slide gives us some specific chemical detail of manufacture with respect of biotransformation challenges, ester to amide conversion, nitrile to primary amine, chiral epoxidation of alkenes, nitrogen alkylation via activated alcohol. These are some few examples of what a typical pharmaceutical company is facing currently. After addressing chemical transformations, we have to look into physical transformations. in biotech industries. This graph is from Journal of American Chemical Society, published in 2003. And it is a literature explaining challenges faced in industry. Figure in top left is litraconazole. Bottom line is crystalline active pharmaceutical ingredient, API. Top line is amorphous. Can we have the cursor, please, on the graph? Yes. Top line is amorphous material on a bead. Middle recrystallization of succinic, tartaric, and malide acid. Microscopic picture is a spray-dried API on a polymer taken from Bend Research website. Next, we have to look into problems and challenges we face as facilities and supply chain. Greener facilities are required. At the moment, only 10 to 30% of total energy associated with the facility ends up going into a product. So we see something like at least 70% energy wastage currently is happening in such industries. Function of facility designs, energy use option. And what does facility of the future look like with respect of global supply chain? Okay, now what measures we have to take to address these challenges? Problem one, intelligent solvent selection with sustainability as objective. Commonly used solvents are still mostly petroleum derived. Heavy use of chlorinated solvents are happening even with FDA approved products. The manufacture of many pharmaceutical and biotech products require use of potentially toxic solvents. Solvent selection performed through screening rather than prediction. Little use of alternative solvents, for example, ionic solvents, is happening. Sorry, any question? And need for acceptable halogen replacements, knowledge of which ionic makes sense, intelligent selection of solvents to meet sustainability needs. Problem number two, dipolar aprotic replacements. As you can see in this graph, towards this end, our contribution was in the form of a paper and patent, which I will explain shortly. The paper was published in 2018 in the Elsevier journal Helion, and it emphasizes on the importance and effect of raw material, excipients, solvents, and other parameters with the manufacture of theranostic smart drug delivery systems. And this was followed by a patent, if I remember correctly, I'm the inventor here. It came out in 2011, I suppose, if I remember correctly. And I just enlarged this section. The patent presents, it relates to a carrier compositions. This is drug delivery system, otherwise known as encapsulation system, comprising a phosphate group of an electron transfer agent and a polar aprotic solvent. Instead of using chloroform, for instance, methanol, diethyl ether, or even acetone are used in some occasions in R&D. We used polar aprotic solvents to minimize environmental hazards. biologically active compounds formulated with a carrier composition have been shown to have improved properties, and the data are presented in this patent, which is available online on Google Patents. Unit operation selection and optimization comes next with respect of other challenge in biotech industries. I don't go into detail of this. Generally, we mentioned that we need to design the unit, design the manufacturing of our aimed targeted pipeline products with respect of energy efficacy, environmental issues, and cost effectiveness of the product which is aimed to manufacture. And then stability tests, life cycle analyses, the packaging are other issues which need to be adequately addressed. Equipment and technology, of course, we need to make the manufacture as simple and as straightforward as possible to minimize the wastage and increase the quantity of the pharmaceutical product in need. The best example is the vaccine need of the ever-increasing population of our planet. The population increasing 8 billion, they need enough vaccines to cope with the pandemic we are facing. And it is not only one shot of vaccine which will require. In some occasions, vaccines need to be repeated. So we need to have adequate facilities to meet the market demand and consumer demand of vaccines, for instance. Towards this end, this is a picture from one of our machines to increase the efficacy and reduce wastage and harm to the environment. We need to study the system thoroughly, not only with respect of the chemical and thermodynamic aspects of the manufacture, but also fluid mechanical aspects. This is another patent which you see my name as a sole inventor, and it describes a new and simple method for manufacturing targetable smart theranostic drugs on micron and nano size. And the machine which enable doing this is through several years of study, now a very simple machine whose mechanical details with respect of fluid mechanics and thermodynamic are very well studied. And even the shaft, you can see here, used in the manufacture of ancient products are very well investigated. This is some futuristic scheme showing roughly drug manufacture using 3D printing. And as you can see, new ultra-precise single-cell resolution 3D bioprinting technique is shown in this slide. Although inkjet-based and drop-on-demand approaches have previously demonstrated single-cell precision, they often suffer from both low accuracy and viability. Alternatively, acoustic cell patterning techniques have proved less damaging and achieved high levels of resolution, but these methods don't allow for the manipulation of specific cells. Now, this is just something towards the future. We need to optimize the system with respect of scale up and cost efficacy. With respect of green synthesis and green manufacture, we published a few papers. I just here give you a couple of examples. Let me know if you require the whole article for your private studies. This is another paper published in 2018 in International Journal of Nanomedicine. And here we emphasized on green synthesis of nanoparticles with the aim of finding a solution for certain types of tumors. And in summary, examples of common pharmaceutical problems have been provided. Main required challenges will be for chemical, bio, and physical transformations. Sorry for this location and the sequence. Anyway, it doesn't matter. All of the items are equally important, regardless of the sequence they are shown. There are many more problems to address. The list will change in time. Many solutions will likely benefit from multidisciplinary research. For example, can a continuous reactor affect the way a transformation is discovered? How are solvents selected to meet both an ecological and synthetic objective? How do bio-enhanced formulations perform in animal models? Do we have correct animal model for a certain study of a particular drug? We look forward to contribution in making Thailand, where we are at the moment, a leader in green manufacturing. Thank you for your presence.
Thank you, Professor Muzafri, for your presentation and your time for sharing with us your expertise. Participants, if you have any questions, you may ask or please do write down to us in the chat box. Professor
I have a
Muzafri, would
question.
you like to share?
I
Yes.
have a question.
Yeah, sure.
I have put it in the chat box that snotizes nitric oxide nasal inhaler. It is 99% effective. What the professor will say about this? The chat
Yes,
box, my question. Yeah.
I'm trying to find your question in written in the chat box.
Snotizes.
Okay. Is it, it's a nitric oxide therapy
Yes.
by
Yeah.
nasal spray?
Ah.
And your question is?
Whether it is effective, although there's a clinical trials and all, whether it can control the COVID-19.
Right. I couldn't hear your question, but I get some part of it, and towards that issue, which I was able to hear, I can give you a very good example. The AstraZeneca vaccine passed accelerated regulatory phases. Because of the urgency of finding a cure for the pandemic, accelerated studies are bypassing many steps of a conventional drug development phases. A typical pharmaceutical compound needs to pass on average 20 years studies and investigations and different phases of clinical studies. When certain company in their well-equipped and well-financed R&D perform studies to develop a pharmaceutical company, including nitric oxide therapy by nasal spray that you mentioned, they select the people to participate in their clinical study randomly. But When all the phases pass successfully and the drug is licensed to be launched into the market, the story may be different. Because the population, even if the number is 100,000 people participating in the clinical study, may not necessarily be smokers, may not necessarily be people who use alcohol, people who use certain medicines or contraceptive pills. This is the story that posed some serious problems with the vaccine that I mentioned. And it had to be recalled back from the market. Because the population that AstraZeneca selected for their study, they did not anticipate certain lifestyles. Probably, this is my guess. So now this experience put more aspects to the stringent regulatory aspects of drug development.
Thank you, Professor Mazafri,
Thank
for
you.
answering to the question. I hope he has answered to your question, sir. Can we move on further?
Yes, yes.
Thank you
Yes.
so much. Thank you, both of you, for joining us. With this, we are moving on to our next keynote presenter, Dr. Sergi Shuko. Unfortunately, he was unable to join us live, but he has sent us across the presentation. So we'll be moving on to his presentation next.
బా ఈయన చచ్చిపోయాడంట బా ఇది కరోనా అంటనే హార్ట్ అటాక్ వచ్చింది కరోనా వ్యాక్సినేషన్ వేసుకున్నాడంట వ్యాక్సినేషన్ వేసుకుంటే హార్ట్ అటాక్ వచ్చిందంట
ఇవాళ 17.
Professor Sergey?
Yes, it's okay. The Wi-Fi is good.
Okay, is it possible for you to present?
Sorry, would you say that again, please?
Is it possible for you to present?
Yes, I am absolutely open.
Okay, so we'll play the presentation.
Yes, visible and very good quality.
Okay, playing it.
If you would not mind, may I start up?
Yes, sure, you can.
Thank you. Good morning, the chair, colleagues, partners, and possibly friends in the future to come. Thank you so much for this invitation to share with you the issues of might be possible mutual interest, unveiling the secrecy of the upgrading model of healthcare services and the biodesign translational applications to get it rearmed. Thank you so much. Doesn't move. Okay, thank you. Thank you. One moment. The name of the link that might exert reliable control over morbidity, mortality, and disabling rates, as well as significantly optimize the cost and efficacy of treatment for those who have fallen ill and for persons at risk is, please, next one. Okay, personalized and precision medicine. Next one, please. EPM as a model of healthcare services of the next step generation is just the science and art, illustrating application of sets of the different tools generated by biodesign, bioengineering, and translational applications. Next one, please. significantly optimizing the cost and efficacy of treatment for those who have fallen ill. Two categories, patients and persons at risk now. Next one, please. PPM as being the grand challenge to forecast, to predict, and to prevent is rooted in a big and a new science generated by systems biology. Next one, please. This is just the illustration in general of the letter. Next one, please. It's the leader and founder of systems biology at the Institute of Systems Biology in Seattle, Washington. Next one, please. And translational medicine or translational application. The next one, please. To connect basic research and daily clinical praxis through a special bridge to get it done. Thank you. The next one. Whilst integrating and consolidating platforms and fundamental sciences, newer ideas, next one, please, and omics technologies. Next one, please. It's just illustrating the letter. Next one, please. Whose data is analyzed, integrated, mined, and clinically interpreted by a set of algorithms and software of bioinformatics, blockchain, and AI. Next one, please. And are implemented into the daily medical practice to secure clinical, subclinical, prognostic, predictive manipulations being individualized or personalized. Next one, please. PPM as a new model of healthcare services is the fundamental clinical and translational science and bioengineering art, illustrating application of the different tools at the population, community, and individuals. Please, next one, please. To implement PPM fantastic resources into clinical practice, there is a strong need to develop a principally new strategy based on biomarkers. Next one, please. Just look around. This is a disease pathways which need to be monitored through different biomarkers. Next one, please. NIH, whilst being a strategic center for American and international medical research and practice, next one, please. This is NIH Bethesda, Maryland. Next one, please. Has included PPM associated with translational resources into a list of the 5 greatest priorities of development of biomedicine and healthcare services in the century. Next one, please. And have the unique decision approved on setting up in the US 2 academical clinical research networks, including, first, Centers for Personalized and Precision Medicine. Next one, please. It's just the illustration of some of the biggest. Next one, please. Next one, please. Next one, please. And second, Centers for Translational Medicine to secure healthcare services rearmed with armamentarium of the next step generation. Next one, please. Next one. Next one. To really understand PPM, we would have to exploit and practice PPM and genomics tools in particular. Next one, please. Just look around. It's a scope and portfolio of genomics related technologies. Next one, please. Each disorder or disease is featured by its own signature. Next one, please. It's some examples of the signature: genome-related, transcriptome-related, proteome-related, and others. Next one, please. Illustrating a set of shifts in gene transcription and expression to be specific for the given disorder or disease, genomic or transcriptional pattern. Next one, please. It's just a specific illustration of a case, I mean clinical case of a person, which is a person at risk. And mama print, technologies, pattern signature. It's a prognostic gene signature and an example showing how prognostic signature can be used to identify different outcomes in cancer patients. In a couple of years, these healthy persons would become cancer persons. Next one, please. So, genomics is considered to be a set of the unique biomarkers and thus the molecular tools to probe genome for its quality and now even be tested for predictive and prognostic biomarkers. Next one. Next one. It's just the illustration of 3 types of genomic biomarkers. Next one, please. The landscape of genetic testing has changed considerably with the emergence of DTC genetic testing. Next one, please. Like type 1D patient, but the patient with genomic disorder. Next one, please. It's one of the first DTC package sets. Next one, please. To secure genetic risk prediction tools for a wider array of common diseases and thus the national health stability. Next one, please. A scope of aims to the above mentioned testing is rather broad, from WGS of individual pre-selected genes to identify a specific mutation and thus the diagnostic, predictive, and prognosticable risks. Next one, please. The illustration of WGS and, more important, look at the dynamics of the WGS market. It's a fantastic explosion. Next one, please. Targeted exome sequencing to identify mutations for some disorders, illustrating here. The next one, please. It's just the illustration of a whole exome sequencing and targeted. Next one, please. And this is the market of the latter. Fantastic. Look. Next one. And transcriptome sequencing. Next one. Look, it's just the illustration diagrammic transcriptome sequencing. The next one, please. Next one. It's the market, dynamics of market of transcriptome sequencing. The next one, please. Ideally, identification of a new specific genomic biomarker would be matched with a specific predictive diagnostic or risk associated program or clinical protocol and therapeutic intervention that target that vulnerability. The next one, please. And genomic biomarkers are thus becoming of great significance to be translated into the daily practice to predict risks of the chronification and thus of disabling. The next one, please. As a tool, we can identify genes that increase disease risk. For example, every person is at risk for cancer, but each person's risk is absolutely individualized. Next one, please. Look at some examples with BRCA1 and BRCA2 biomarkers related dynamics to monitor persons at risk. The next one. And each risk could be assessed and managed. Next one. Look at the monitoring and management of disease-associated risks, personal lifestyle plan, risk modification, and disease management, and now more, risk management. And this is decision support tool. Next one. Improved patient outcomes with the use of the biomarker test must consider not only increased survival or quality of life, but also improved CDS and making. Next one, please. It's just the illustration of CDS making and algorithms management. The next one. Having access to genomic information will become increasingly important as as physicians are progressively receptive to incorporating genomics into clinical practice. The next one. The next one. No. Fourth. A unique example of translational applications to illustrate subclinical and predictive risks as applicable to tumor progression to be screened, monitored, and analyzed. The next one. The next one. Genome being neighbored with phenome. The next one. Phenome is a bridge from genome, proteome, metabolome, which are two important elements of phenome. The next one, please. Look, this is a way to move ahead to connect genome and phenome, illustrating high throughput measurements with the phenome phenotype from science to medicine. Next one. Look how cytomics, proteomics, metabolomics are being withdrawn from genomics. The next one. Next one. Next one. The next one. So, for instance, a combination of genomic and proteomic biomarkers are becoming as a tandem of great significance to be applied into PPM. The next one. Next one. And for the prediction of chronification of disease is just the applications of proteomics. The next one. The other, but also unique example of cytomics-related translational applications to illustrate subclinical and predictive risks as applicable to tumor progression is CTC, circulating tumor cells. Next one. Look at that. Now, cell search method is the only FDA-approved system based on the surface expression of epithelial cell adhesion molecule APCAM to control and to monitor CTS in blood. The next one. It's just the examples. CTS in colon, breast, and other cancers. Next one. Exosomes, one more unique example of combinatorial translational applications to get cancer management significantly improved. Next one. It's just example of the application of exosomes in daily clinical cancer practice. Next one. The latter would open a way for analyzing the intratumor, not intratumor molecular heterogeneity, and to predict the risk, to prognosticate the evolution of the tumor, and to tailor the personalized targeted treatment. The next one. Next one. And finally, omics-based. Next one. Look, metabolomics illustrates the functional state in reality of the cell at the level of metabolism and could be used as a special set of translational tools for practical medical applications. Next one. Of special interest, really special the latest days, is interactomics and interactomics related approaches. The next one. It's just the illustrating of two types of bridges: genome, proteome as a part of phenome, and interactome. The next one. And interactomics based networks, I mean biological networks. Next one, please. For normal physiological state, please, next one. It's the normal physiological state illustrated. Next one. And pathobiological states. Next one, please. Look, leukemia-related networks. The latter may both serve with network-based biomarkers, NBB, to secure diagnostic, predictive, prognostic, and monitoring the multifactorial disorders. Next one. Who will patients or persons at risk and their physicians trust to store and interpret the clinical data collected, harvested, and mined? Next one. Next one. Healthcare IT does offer a potential solution to those barriers. Next one, please. Look around. It's a scope of devices to secure the outcome of IT monitoring. Next one. Cloudy technologies. It's a fantastic applications to secure the physician to place and to use networks among the globe. Next one. As a result, a patient or a person at risk, next one, becomes a unique data carrier. Next one, please. Look. 665 terabytes is a medium average source of the human body. Next one. Whilst learning about possible risks of a disease, and the physician can reasonably select a kind of preventive and personalized protocol routing for the predictive assays made. The next one. An example, you have an illness and the standard treatments, I mean canonical therapy, are not working for you. What now? Next one. Modern therapeutic modes and applications. Next one. Well-known cellular therapy, including stem cell-based therapy. Next one. Gene therapy. Next one. Using genome editing technology alone, precise gene surgery in any human cell and organism. Next one. It's just the illustration of mechanisms of editorial reading. The next one. It's a fantastic outcome, the only in the world. Incurable leukemia. It's a fantastic newborn, where genome editing has been used at Johns Hopkins, Harvard, and Cambridge, UK. Next one. Next one. Next one. Next one. Targeted therapy is a way to recommend a therapeutic protocol being tailored individually and to secure the highest clinical efficacy and minimize the adverse effects and reactions. The next one. Look, for example, the most popular targeted therapy is applicable to cancer receptors and types of targeted therapy. So ligand receptor interrelationships are the base of the targeted therapy of the future to come and now. Next one. Next one. Personalized nutrition. With the progress of technology in the field of genomic and molecular medicine, this is becoming a fantastic, promising, and attractive field. So, using a nutrigenomic test, you may manage any kinds of preventive manipulations using the personalized diets. Opportunities exist at every stage of disease initiation and progression to develop a personalized health plan, addressing lifestyle, risk modification, and disease management, and later, personalized health management and wellness program. Okay, yeah, I'm finalizing, sorry. Sorry, it's next one. Next one, next one. 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 As an illustration of drastic changes in big pharma and translational market-related areas, see below. Next. Look. It's a proportion of drugs in the area of personalized precision medicine. Next one. Next one. We are finalizing. Next one. This approach should be based on postulates which, next one, will change the incarnate culture and social mentality, and the next one, and will create the future generation of creative thinkers, entrepreneurs, and clinical leaders. Next one, next one, next one, next one. We would need not primitive physician, we would need the clinical specialist of the next step future to secure preventive precision personalized medicine and its outcome be predictive and guaranteed. Thank you so much for your patience.
Thank you so much, Dr. Sergey, for joining us and for your amazing presentation. Attendees, if you have any questions, you may write to us and we will make sure we answer to your queries. Due to short of time, we'll be moving on to our next keynote presenter who has joined us here. Next, I would like to welcome Dr. Ranendra N. Saha, the Director, and Shri B.K. Birla and Srimati Sarla Birla Chair, the Professor from BITS Pilani, Dubai, from United Arab Emirates.
Good morning. Can you hear me?
Yes, sir. Good morning. We can hear you.
Okay. So should we start?
Yes, sir. You may begin.
Okay, just a minute. I have to share my screen.
Sure.
My goodness, just a minute. Why it is not moving? Okay, can you see my screen?
Yes, sir. It's visible.
Okay. Good morning to all the participants, plenary lecturers, speakers, and organizers. It's a pleasure to be participating in this program. Thanks to the organizers for inviting me to deliver the lecture. As the time is limited, I'll go and straight to the content. What I'm going to present the work we have done in our group on design and evaluation of novel nanoparticulate drug delivery system for the brain delivery. You know that delivery to the brain is always a challenging. So novel drug delivery system is a very, very important for the existing drugs, which is having some drawbacks. And by designing novel systems, we can overcome many of the problems or deficiency to achieve better therapeutic efficacy, better patient compliance, and avoiding many toxic or side effects. So if you look to this, why this important from this diagram, I'll try to explain that when you deliver a formulation, conventional formulation, it is getting absorbed through the routes, whatever you have given, oral, parenteral, or any other routes. Drug is absorbed and coming to the systemic circulation, where out of ADME character absorption is over and then the distribution. So when it is coming to the systemic circulation, it goes to the site of action. For this case, I specifically mentioned brain because I am going to discuss about selective distribution to the brain. So if I assume the site of action is the brain, some drug goes to the brain, but other drug goes to the other sites where there is a possibility of side effects or toxic effects. And it is a very, very important for anti-cancer drugs, where it is a serious problem for the patients that due to non-selective distribution of the drug, the patient suffers from severe side effects or toxic effects. So this is always a challenge. And how I can explain you a very simple example. If you assume 20% of a drug goes to the brain in normal or conventional dosage form, and you need 20 milligram to the brain for a 24-hour action, then you have to administer 100 milligram. 20 milligram goes to the brain. Then remaining 80 goes to the other part of the body, which is a big challenging and problematic. But if you can modify this selective distribution by a novel delivery system, even to 50%, from 20% to 50%, you can selectively distribute to the brain. You understand that for getting same 20 milligram to the brain, you now need to deliver only 40 milligram. So your dose is suddenly decreasing from 100 to 40, cost will reduce, but obviously there will be cost for designing novel delivery system. But how much going to the other part of the body? Only 20 milligram. So this is the benefit we see, that from earlier 80 milligram, it is only 20 milligram going to the other side of the body, which obviously will avoid or lower the side effects or toxic effects. So with this background, you try to understand that people are working on novel delivery system for selective distribution or targeted delivery system. Now, what can be the nanoparticle or the nanoparticle benefit of drug delivery? The drug stabilization in vivo can be possible because many drugs have a stability problem which can be changed, enhanced in the system. Facilitation of the absorption, better availability, which can be possible, or modified PK, elimination, etc. can be changed. Controlled drug release is possible, for which you can have the therapeutic efficacy for a longer time, so that interval between the dose can be increased. Drug targeting or organ targeting is possible, whether it is organ, tissue, or brain. Modulation of the spatial drug distribution or selective distribution, more efficient of utilization of the drug, and thus cost reduction. And obviously, the most important part is the patient compliance, which is the most important for any drug delivery, is the patient compliance and better quality of the drug, better quality of the life, sorry. So what I'm going to present is some of our work which we have done in nanoparticulate system for effective delivery to brain, and their pharmacokinetic and biodistribution studies for evaluation. Because when you design a novel delivery system, it's very, very important to see the pharmacokinetic and biodistribution profile, whether it has been changed and as expected. Then only you can think of better efficacy. If we achieve the selective distribution, we achieve less side effects. That means we can expect better therapeutic efficacy and patient compliance. We have worked many drugs, but today I'm presenting here the 3 drugs which we have done and which are normally essentially to be delivered to brain. Dopamine, tapentadol, temozolomide, and keep in mind that some of these drugs we have done on the request of the pharmaceutical industries for development of the product. You know that brain is a very, very important target organ for many therapeutic efficacies. Parkinson's, Alzheimer, Huntington's, schizophrenia, migraine, brain tumor, meningitis, many other purposes we require to deliver the drug to brain. But due to blood-brain barrier, which makes a challenge to deliver drug to the brain. And it have happened that many drugs are not reaching enough in quantity. Either they are not directly given or they are given large dose so that required amount can go to the brain. So the challenge is the blood-brain barrier, and the challenge is the drug's character, which doesn't make it selective distribution, rather make it non-selective distribution, where it can go to the other parts of the body, as I mentioned earlier, which can cause cardiotoxic or other toxic effects, side effects. So that is the reason it is a big challenge to make delivery system to deliver to the brain. So basically what we can do is a non-invasive approach to deliver the drug to the brain. Now if you see, nasal can be a very good route to deliver the drug to the brain, and part of our work on industry-sponsored project, we worked on nasal delivery to the brain. We initially worked with dopamine, and you know it is a big challenge candidate drug for brain delivery because it has a very poor permeability or entry to the brain. And that is the reason that methyl dopa and other derivatives are given, which has a better permeability and then gets converted to the brain. So to develop a platform technology, we worked on the dopamine first. So what happens, or the hypothesis, that if we deliver to the nose, to the nasal route, we can expect more direct delivery to the brain, avoiding the systemic circulation. If you see that the location where you deliver is an important part. If you look here, the therapeutic administration nasal cavity, if you give respiratory route, but it is going to the system. Olfactory is also there, and trigeminal is there. But you can see that olfactory and trigeminal, it can be possible to take directly the drug to the brain. But there is a possibility of little bit to go to the CSF drainage also. So we try to approach this and see how the delivery can be made to the brain. We took dopamine, we took tapentadol, and I'll see what is the advancement has been made on this. And really, a product has been brought to the market also, very recently. What we tried for the dopamine and tapentadol is a liposomal drug delivery system, where audience, I am sure, have some idea of the liposome, which is made by using the phospholipids, and the drug can be entrapped in these phospholipids and then can be delivered. A lot of work has been done. So that is the way liposome is not new. What we tried to using the liposomal delivery for the dopamine first. Sorry. So what is important that we worked on liposomal preparation and entrap the drug there, dopamine and tapentadol, and try to see what are the results obtained. I'm not going to discuss the liposomes as I expect all of you are having, but it has the advantage, its bioacceptability, biodegradability, good therapeutic efficacy can be achieved. By this process, you can have a controlled release and thus biological half-life can be increased and less toxicity compared to polymeric nanoparticles. Dopamine is well known. I just kept a slide, but what I try to tell is a small molecule, highly hydrophilic, and thus it doesn't permeate through the blood-brain barrier because log P is very low. But it is a useful substance, neurotransmitter, you know, and if it is not available, it requires to be given externally, but dopamine is not given, it is given methyl dopa or other prodrugs to reach or get the dopamine in the brain. What we worked, we prepared the dopamine-loaded liposomes, characterized by using different instruments and techniques, optimize the processes to make the best formulation with high entrapment efficiency in drug content, and also we did the stability study. If you see, I'm just only minimum slides I'm taking for the characterization of the nanoparticles or liposomes. This is the TM pictures where it shows that nanoparticles have been prepared, a liposome of nano size have been prepared. This is the TM picture, and this is the AFM picture, which indicates that the liposomes are really of nanoparticle size. When you studied the release, you can see that using different like phospholipids, we could achieve the release extension from 8 hours to 50 hours, more than 50 hours. You can see the picture on the right bottom where the release extended more than 50 hours. Some of the cases we got different. That means what is that we found it, that using different phospholipids in different proportions, you can achieve different extension of the release of the drug from the liposomes, which give you the flexibility of designing according to your need. So what we did ultimately is the in vivo pharmacokinetic study on animal model. We used rats, and first we treated the rats for 5 consecutive days for DA depletion or the dopamine depletion by treating with reserpine to ensure that the animals are not having any natural dopamine present in the brain. Then we administered the dopamine solution, dopamine loaded 3 types of liposomes by nasal route as well as IV route also, and then collected the samples from the blood and some tissues and the brain to determine the drug transfer to the brain and other places. At different time interval, animals were sacrificed and the tissues and organs and some blood samples have been analyzed to determine the drug concentration after administration by nasal route and by IV route. I'm going directly to the result. You can see here, the bottom one is the drug solution given by nasal route, which indicates a very little amount of drug has gone to the brain when delivered drug solution, pure drug solution. What happened? Someone has come. Can you see the screen? I don't know. Someone has come to the picture.
Yes, sir. Mr. Wasim Akram, can you please stop sharing your screen?
Oh my goodness. Again, I have to go. No, what happened? It is. Okay. I don't know. Suddenly, why someone entered.
Yes, sir.
Okay, now it is working. Can you see?
Yes, sir, we can see, we can see.
Okay, fantastic. Okay, so you can see here that pure drug when given nasal route is a very little reach to the brain, whereas 3 phospholipids, which is different type of phospholipids and mixture of the phospholipids, 2 are individual phospholipids and 1 mixture of the 2. And you can find it that the amount of drug reach to the brain is a much higher than the pure drug solution when given nasal route, which indicates that using the liposome, the dopamine delivery to the brain has increased many-fold. And further, you can see that they are remaining in the brain for a longer period of time. Even after 50 hours, the concentration in the brain remains very high, which indicates that duration of action can be extended by a single delivery. When the same dopamine concentration in the brain is checked by delivering the liposome through IV route, here you can see that the amount of drug gone to the brain even by IV route of liposome is found to be much higher for liposome, which also indicates that liposomal delivery through IV route can enhance the dopamine reaching to the brain much higher with comparison to pure drug solution when it is given IV route. So liposome is basically providing 2 things. One is even it's given nasal route, it can reach much higher amount, but even IV route, the dopamine delivery can be increased because if it is delivered in the liposomal formulation. If you see quickly the pharmacokinetic data, you can see here AUC for the liposomal preparation increased millifold, where the pure drug AUC is 0.416 by nasal route, whereas the 3 phospholipids, it is about 50, 156, and 77. Similarly, Cmax also you can see increased manifold from 0.331 for the pure drug to 2.37, 2.53, and 2.74 for the liposomal preparation. For the intravenous administration also you can see pure drug AUC is only 0.034, where the liposomal preparation 10.62, not as high as was the nasal route, but still by IV route also this formulation can be delivered. We studied also in the drug reaching to the plasma from the nasal route to see whether the drug is coming to the system from the nasal route or is it via system through the blood-brain barrier. We found in case of pure drug, it is a much higher is reaching to the system, but for the liposomal preparation, nothing is going to the brain to the plasma from the nasal delivery. So which indicates, this result indicates that maximum amount of drug from the nasal route of the liposomal formulation reach to the brain, nothing is going to the plasma. So that's why side effects or any other things may not happen when we use some other drugs also. Based on these, we did tapentadol also, which was the need, and I share with you, the company has brought a nasal preparation recently for tapentadol in the market. What we find also similarly, tapentadol, which solid lipid nanoparticle and the gel and gum preparations, you can see the AUC that brain oral drug when it is given tapentadol, the area under is 3.326, but the gel and gum, it has increased to 923. And SLN also, it has been benefited by that process. So which indicates that tapentadol also can be taken to the brain by nasal route to great extent, and selective delivery is possible that way. The last drug which I'll present it, time is less, 30 minutes, only 10 minutes left. I'll finish another 5-6 minutes. We also worked on temozolomide, which you know, it is very much used for the brain tumor or brain cancer glioblastoma situation, temozolomide is a well-used drug. We worked on delivery of temozolomide using nanoparticle system based on polymer and lipid. We have formulated, characterized, and did the pharmacokinetic study in the animal model and found enhanced delivery of temozolomide to the brain when administered to the animal model. I'm directly going to the pharmacokinetic study of temozolomide, but we prepared PCL, polycaprolactol based polymer system nanoparticle and lipid based nanoparticle. Brain tumors, you are aware, not much to discuss, but yes, it is a very challenge to deliver the drug to the brain selectively, because as I explained at the beginning, that even certain percent of the drug going to the other places, which can cause side effects or toxic effects, which is a very, very serious problem for cancer treatment. So this is the profile of the temozolomide, and it is a suitable drug for delivery to the nanoparticle, and we try to find out that how this selective delivery is possible. We characterize the nanoparticles, which is prepared using the polycaprolactol polymer and different lipid-based nanoparticulate system, characterized by SEM, AFM, etc., and also studied the release profile. And we found by using those polymers or lipid, the extension release is possible to many-fold, 3-4 days is possibility. What we did, the pharmacokinetic and biodistribution studies, that we used male Wistar rats and delivered the drug, pure drug, by intravenous intraperitoneal route. We administered pure drug as well as the polymer-based nanoparticle and lipid-based nanoparticles. Plasma and tissue samples were collected to determine that the staying of the drug in the plasma, selective distribution to the brain and to other organs like heart, kidney, etc. And all these samples were analyzed by the biological analytical method or bioanalytical method developed in-house by my group of students and colleagues. I'm going to the pharmacokinetic result. If you see, here you can see that the temozolomide intraperitoneal pure drug, IV, the pink color one is the IV and the blackish one is the IP. You can see that the drug profile in the blood, a plasma, is much higher when it is given as nanoparticulate system, whether it is polymer based or lipid based. The profile indicates that the drug remaining in the blood in higher concentration for much longer period of time. You can see that the pure drug IV route, it is coming after 8 hours close to zero, whereas the nanoparticulate system is showing higher concentration throughout and remaining high concentration for longer period of time, even beyond 16-18 hours. If you see the AUC for the temozolomide IV is 17.58, whereas for the polymer-based it is 27.68, and for lipid-based nanoparticle, you can see it is higher, about 19 and 25 plus. Similarly, the biological half-life also you can see has been enhanced from 1.15 in IV and 1.35 for IP route, whereas the nanoparticular case, it has been 2.43 hours or more, which indicates the drug remains in the blood for longer period of time and high concentration. Here you have to understand an important point that if the drug remains in the blood, it indicates it did not go to the other organs probably, and it has a greater ability or prospect to go to the brain, because as long as the drug is in the blood, the permeability to the brain also is going to be higher. So that indicated by the result that more drug has gone to the brain as found it from the AUC and T-HAP. Now it is fine. It is a blood sample, plasma samples I'm telling you. What happened to the actual brain? If you see we studied also the brain, the drug delivery into the brain, and you can find out the pure drug, the profile is the lower layer. You can see this is the color, the orange color, which is showing much lower profile in the brain, whereas the nanoparticulate system, irrespective of whether it's a polymer based or lipid based, they are showing much higher concentration, and even the duration point, if you see, it is a much higher with respect to the pure drug administration by IV route. It is having higher concentration even after 8 hours. So it indicates that more drug is definitely going to the brain when it is given as a polymeric system, whether it is a nanoparticulate system, whether it is a polymeric or it is a lipid based. So this indicates that selective distribution really achieved by delivering as a nanoparticle use polymer or phospholipids. If I see the spleen, where it is the opposite, though not big difference, but yes, in case of pure drug, it is higher, slightly higher. But most important part, you see the heart. In the heart, you can see the high concentration reached for the pure drug, comparatively much lower concentration has been achieved in case of nanoparticle, which indicates that definitely that the nanoparticulate system is providing more drug to the brain, selective distribution is achieved, and lesser drug is going to the heart or other places. So two aspects is required. One is the more drug going to the brain and less drug going to the other places where there can be problem, which has been achieved, found it both in case of animal study, but yes, obviously it is also required to be volunteer study or the patient cases. But this indicate that this novel delivery system can be useful for the selective delivery of the drug to the brain, whether it is anti-cancer drug, whether it is other CNS active drug, but nanoparticulate system can be a suitable approach or a suitable delivery system which can provide better selective delivery, less or no side effects or toxic effects with better patient compliance, which are expected to be from a novel delivery system. I cannot finish without presenting about BETS Pilani. I am at Dubai, though I spent 28 years in Pilani, and last 7 years I am in Dubai, so 35 years I am with Pilani. My research lab is still continuing in Pilani, and my projects, sponsor projects and PhD students are working there. But BETS Pilani has 4 campuses. Main campus is in Pilani, which right side top corner. Then we have established Dubai in 2000, but I am here from 2014. Then we have a campus in Goa and Hyderabad. And very recently this year, we have created a School of Management campus in Bombay. So obviously I must confess my contribution of my PhD students who are co-authors, a co-presenter in this one, is Archana and Vivu. They are now working for industries in abroad or in India, but without the contribution of my students and research scholars, this could have been possible. And also support from the industries who sponsored these projects, provided all the support for work. I must say that this is the Saraswati temple present in Pilani campus, which blessings all of us at Pilani. Thank you very much. I think I have finished 2 minutes. Before any question, I'll be very happy to answer that.
Thank you so much, sir. Thank you for your amazing presentation. Participants, kindly let us know if you have any questions.
I find my ex-colleague, Dr. Nayak, is present there.
Oh, that's so nice.
Dr. Nayak, good morning and greetings from Dubai.
Thank you.
And I also see Subhash Mandal. Oh, he's my junior at Jadavpur University. Nice to see you, Subhash. Professor Subhash. Yes, please.
Sir,
I can receive any question to my mail also, which you can share.
Nice to interact with you, sir.
You can share your email address in the chat box. So if any questions, participants can directly ask the questions.
Sure. I'll type that one.
So as there are no further questions, we will move on to our technical sessions. So before that, I would like to welcome our session chair, Dr. Subhash Chandra Dinda, sir.
Yeah. Good
Dr.
morning,
Suresh.
everyone.
Hello, sir.
Yeah, good morning, everyone.
Good
First
morning.
of all, I'd like to thank the organizer, the Association of Pharmaceutical Research and the Bioleagues International for inviting me for chairing a scientific session. And I'd like to say, as you know, world is fighting against an unknown enemy called COVID-19 pandemic, and all are fearful about it, and all are searching either alternate medicine or maybe the vaccines. So what happens is everybody eagerly waiting whether we are getting in therapeutic alternate. And in meantime, to know what is happening throughout the world, what is happening, what the scientists are working throughout the world, different corner. This association is need, and we have to gather in a form of seminar, symposia, or say maybe conferences. But you know, physical mode controlling all this activity is now restricted. So it is very essential to invite all the professionals, researchers, scientists from throughout the world, and gathering them a platform that can be shared by the scientific community to share the knowledge. So updating knowledge is very important, and I think this platform will help and create a, because the deliberation from the stalwarts of the pharmacy, the scientists from the pharma field, from industry, from academia, what they are sharing their knowledge and what they are sharing their research contribution, and in the form of deliberation, as well as this the people, the participants, the delegates who are delivering their research output in the form of oral or poster presentation, they will open up a new horizon in the field of pharmaceutical science for the betterment and upliftment of the pharmacy profession throughout the world, because it is now globalization. Not we cannot say from India or from Dubai, or so we are together in this platform. Maybe from Russia I have seen, maybe from USA, from India, every person connected through your organization. So I am very much thankful to your organization for arranging such a nice conference, and coordinating everybody to have a sharing their views and exchange the research views, outputs. As you know, Professor R.N. Shah, great person from Pilani, and his deliberation include brain delivery and using nanoliposomal technology. So it's a very upcoming field, and now our new generation, the budding scientists, they can adopt this technology and can they target the new the translational health, the previous speaker told. So this modern upcoming field we can get into, or into the insight of these areas, and we can utilize for the research purpose, and can develop certain alternate to combat the diseases like pandemics like COVID-19 and so on. So maybe still there is a threat of AIDS, cancer and all. There is no solution still. So we are struggling, all world is struggling for this type of disease. And from these deliberations or the conference, I think it will give you better yield for the betterment of the therapeutic outcome. Thank you. And I would like to thanks all the organizers for giving me this opportunity once again. And now we should not spare the time, valuable time. We should utilize our speakers, or say participants, to deliberate their research outcome. Thereby we can get into it and learn so many things to be utilized for the betterment. Thank you.
Thank you, sir. So we'll be moving on to our technical session 1. And our first track for the day is COVID-19 drug development. And I would like to welcome our first presenter of the day, Dr. M. Senthil Raja from J.K. Nataraja College of Pharmacy, India, who will be discussing on the prediction of potential inhibitors for SARS-CoV-2 using comprehensive drug repurposing and molecular docking approach. Dr. Senthil Raja. Before we begin, I would also like to welcome our session chair who has joined us here, Dr. S. Duraivel, who would like, Dr. Duraivel.
Hello.
Yes, Dr. Senthil Raja.
Yeah, yes, madam. Yes, madam.
Yeah, can you
Share your screen and start because time is short.
Yes, time is short. Kindly begin.
Yeah, we'll start now.
Everybody should stick to your time. So 10 minutes given. So by 7 minutes, you try to wind up and 3 minutes for question answer.
Dr. Senthil Raja, let us know what kind of difficulty are you finding in presenting?
Yeah. I could not able to see my presentation slide. That is what I am searching.
You have option of share screen at the bottom. Can you please click on it? Then you can share your screen. We are not able to see your screen.
Share screen where?
So it's in green color right at the bottom.
Yeah.
Yeah.
Okay.
Then you open your file.
Yes, now you may open your file and begin. Before we begin, I would like to inform all the participants you will be given 10 minutes for your presentation. You will have to conclude your presentation within the 10 minutes allotted. So thank you, Dr. Senthil Raja. You may begin, please.
Is it what?
Start now.
So can you please begin, sir?
Hello?
Yeah, start now.
Oh, okay. Yeah, very good morning to all. First of all, I would like to thank the organizer. Since I am the organizing member of, I am also myself, and also I would like to express my sincere thanks for the organizing members, including me and others, for providing the opportunity as a first person to give the presentation in the oral as a oral. So, okay, let me start with my presentation. So my topic is prediction of potential inhibitors of SARS-CoV-2 using comprehensive drug repurposing and molecular docking. So this is what the title. I am Mr. M. Senthil Raja, Professor and Head Department of Pharmaceutical Chemistry, JK Narada College of Pharmacy, Tamil Nadu, India. So here the abstract. First is I would like to show abstract. Actually, my idea is prediction of potential inhibitors by using comprehensive drug repurposing and molecular docking approach. So this is what my title. The abstract belongs to this title is pandemic prevalence of COVID-19 has become very serious global health issues. Scientists all over the world have been heavily invested in the discovery of drug to combat SARS-CoV-2 infections. So it has been found that RNA-dependent RNA polymerase, that is what RNA-dependent RNA polymerase, that is called RDRP, short form. It has given RNA-dependent RNA polymerase. It plays a crucial role in the SARS-CoV-2 replications. This could be a potential drug target. This will be a potential drug target. So comprehensive computational approach means including what and all means repurposing, drug repurposing, and as well as molecular docking. So these two approach has been used already in this work, in this study, to produce effective drug candidate targeting RDRP means RNA-dependent RNA polymerase enzymes of SARS-CoV-2 virus. That is what this. Coming to the next slide of the same abstract, here we have used 5 drugs. Actually, I have used 5 drugs. That is, one is omifenavir, another one is raltegravir, third one is delaveridine, fourth one is fidaxomicin, fifth one is gemcitabine hydrochloride. So these are the 5 drugs that have been selected for my study. These 5 drugs, it revealed that may have the potential inhibitory effects or interaction with that RDRP of COVID-2. So which has a potential inhibitory interaction with RDRP and could be effective drugs for the COVID-19s. So next slide is, in addition to that, visual screening of some zinc database also have been developed, that is called zinc 57 and 58, also included for the prediction with pharmacophore features to produce effective RDRP effectively with RDRP proteins. So this indicates that selected drugs will act as a potentially effective drug or inhibitors for this particular enzymes. So the main aim and objective of the study is computational biology and molecular docking approach have a wide variety of applications in drug discovery. Thus, which gives novel insights to the screening of potential drugs for the treatment of COVID-19. So the main objective of the present study is, the main objective of the present study is drugs with proven activity were selected and analyzed using comprehensive computational approaches such as drug repurposing, molecular docking, as well as pharmacophore modeling techniques. For what? To target RNA-dependent RNA polymerase enzymes of coronavirus, of SARS-CoV-2, and that virus multiplications in order to stop the virus multiplication. Main objective of the study is to select already proven activity drugs to be analyzed by using various computational approaches such as drug repurposing, molecular docking, and as well as pharmacophore modeling techniques against RNA-dependent RNA polymerase enzymes of coronavirus. As a result of these findings, this approach can open a new insight for a fight against COVID-19. So there are some methods have been optimized or used or employed. What is the methods are retrieval of structure of SARS-CoV-2 RNA polymerase drug candidate. That means first we have to select the structures of drugs. Already exist drugs, then the protein structures. That means RDRP structures. So the RDRP structures of SARS-CoV-2 have been retrieved from protein data bank. And further, so in this case, there are 35 number of drug candidates have been optimized. That means selected. But among the 5 drugs, 35 drugs, 5 drugs only could given proper potential or effective against RDRP of SARS-CoV-2. That is what you may think that people may think that why is 35 drug candidates were selected, but finally you said that only 5 drugs. Selection of drugs may be quite vast, but only 5 drugs could have produced beneficial effects or potential effects against RDRP proteins, that particular enzymes. So here, once that selected drugs, this is one method. Another the method continuation is screening. First, we have to find out the structure of both RDRP and as well as drug candidates. Then screening of that particular inhibitors, polymerase inhibitor. That means the drugs. First, we have to select drug structure. Then we have to screen on RDRP of SARS-CoV-2. So for that, AutoDock Vina software of molecular docking approach have been selected or used. Autodoc, we know software. For what purpose means for screening of the drugs against RD-RP of SARS-CoV-2. So first in the Autodoc, first crystal structure of RD-RP was retrieved from Proton Data Bank, and it was processed by removing water and complex molecules using 5-mol software, 5-mol. So after preparing this structure, it produces some 2D or 3D dimensional ligand protein interaction diagrams that can be generated by plot plus to find out what is the interactive position between inhibitor drugs and as well as with inhibitor drugs and PDP, RD-RP. That this is what this methods. So another second method is structural insights of drug surface hotspot. So in that what we have means LigPod plus Discovery Studio, 5-mol were used to figure out drug surface hotspot. For what purpose means to get polymerase inhibitors. Okay. Hello. I am audible?
Yes, sir, you are audible.
Okay. So in between some noise is coming. That is what. Okay. So remdesivir, favipiravir were used as a positive control here. So far we have seen methods. Another one is because I just within 10 minutes giving only small part. Another one is the result. The result is pharmacophore modeling and visual grading. What pharmacophore modeling and visual grading? For in order to perform pharmacophore modeling, pharmacogist was used for modeling of pharmacophore features. That is essential for interaction of that particular RD-RP inhibitors. Sorry, that particular selected drug inhibitors with the RD-RP of SARS-CoV-2. In this study, the topmost inhibitors were used for the pharmacophore modeling. Topmost inhibitor means that means Umifenavir, Reltigravir, Delviridine, Fidaxomycin, and Gemcitabine hydrochloride. These are the 5 drugs mainly has been selected as I said in the previous slide. So those drugs have been given. These novel compounds were further used for developing new inhibition studies with the RD-RP.
3 minutes left.
Okay. So another one is results. These are all the results of that particular top. I have found that. So you can see in this slide. Omiphenavir binding energy. So there is a binding energy because docking studies have been Fidaxomycin, Delaviridine. These are all things. These are the docking figures. So we can able to see the structures now. Docking figures. So the first structure is we can see that Umifenavir. Second is Raltegravir. Third one is here Fidaxomycin. So you can see that there are some background of RD-RP structure. In between some pink color, it shows that is the drug inhibiting or inhibitors. Another one Delaviridine. Third, fifth one drug is Gemcitabine. So these are the docking structures. Another one we can see the interaction of RD-RP with the interaction of this RD-RP. RP with that enzyme, that particular RD-RP polymerase enzyme with these drugs. So in different diagrams. The third structure is structural analysis of drug hotspot in the RD-RP. So this is what drug hotspot. The fourth structure, it shows that it will be a ligand-based pharmacophore model. By using ligand-based pharmacophore model, it shows violet color, some aromatic features, yellow color represent hydrogen features. So this also by using zinc compounds. So results, the whole number, you can see that these are all the results which has been produced by using various molecular docking and molecular modeling structure process. So you can see that these are all drug likeness properties, all those things. So based on this results, the result discussed at present, COVID-19 is a very global challenge for the communities. But today, no satisfactory breakthrough has been developed for the treatment of COVID-19. Several attempts has been made to treat this disease that drug can remain questionable. So this is based on the discussions. So the computational approach is one of the approaches with the drug proposing methods could be effective approach to this COVID-19 challenge. In this study, several polymerase inhibitors targeting SARS-CoV-2 have been studied. So already those drugs, in this case, the selected drugs have already having antiviral effects such as hepatitis C effect against hepatitis C virus, HIV, Zika virus. So it has been selected as antiviral. But we could assume, I could assume that this antiviral drug definitely have the effect on RD-RP in enzymes. So hence, drug proposing along with molecular docking methods have been used for screening that particular effect. So these are all the discussions. Moreover, the vast number of things have been produced. Drug surface hotspot also indicates very good effect. So if you come to the conclusion, COVID-19 has been created disaster effects. The present study aimed to compare this global effect, suggesting potential drug candidate to treat COVID-19. So in this study, the selected drugs like imipenem, fidaxomicin, aldehyde are it shows screened for novel compounds to common surface hotspot. It acts as a potential inhibitor in the RDRP, RNA-dependent RNA polymerase of SARS-CoV-2, which highlights this could be a effective in the treatment of SARS-CoV-2 infections. That's what my presentation. Thank you. Because my presentation, it was very vast study, but only 10 minutes allotted, so I just restricted only in this part. Thank you. Thank you for the organizer to provide me opportunity to present in front of all. Thank you.
For nice presentation.
Thank you, sir.
It is for question session.
Thank you for the
audience.
Thank you, madam. Thank you, madam. Thank you.
Please, if you have any questions, you may write at the chat box and you may discuss with the presenter. And next, I would like to
out of slide. You can remove your slide.
From Chulalongkorn University, Thailand. She'll be discussing on the adverse drug reaction reports of coronavirus diseases 2019, a retrospective study at a university hospital in Thailand. Ms. Chotirat, are you online?
Let Senthil Kumar should remove his slide.
So can you please stop sharing your screen?
Is he online?
Yes.
Okay, can you see my slide?
Yeah.
Okay, let's begin. Okay. The topic that I will talk for about 10 minutes from now is the adverse drug reaction reports of COVID-19 drugs, a retrospective study from University Hospital, Thailand. Okay. First of all, I would like to thank all of my team for this successful study. The first group is the infectious disease patients from my hospital from Thai Red Cross Emergent Infectious Disease. And the second group is my pharmacy residency in infectious diseases. Thanks to them for helping me to data collection and also take care of patients in this specific group of patients. Okay, I will talk a little bit about the coronavirus disease to 2019. I think that everybody here knows a lot about it right now. COVID-19 is a newly discovered coronavirus and it spreads primarily through droplets of saliva or discharge from nose. The symptoms vary from mild, moderate to severe, and patients in the older people and those with underlying disease like cardiovascular disease, diabetes, chronic respiratory disease, cancer, and obesity are risk for both infection and risk for severe illness. Protection is important. As you know, we don't forget to do the social distancing, to wash your hand, and now we already have the COVID-19 vaccine. How about the situation in Thailand right now? As you see, the first one is on the April 3rd. The new case looks good, right? But now on yesterday, the new case looks very bad because since last week, we have a new phase of new infection, the phase 3 infection, and the new case from now range from 1,000 to 1,500 patients right now. This is a nice slide to talk about the clinical symptoms, clinical signs, and potential therapy. I would like to focus a little bit about time to start antiviral. If we start antiviral agent as soon as possible, especially on the stage 1, for the early infection, the benefit of start this medication as soon as possible is achieve viral clearance, reduce duration of symptoms, minimize contagiousness, and prevent progression to severity from stage 1 to stage 3 and 2, from stage 1, stage 2, and to stage 3. This is a nice slide explaining about the mechanism of actions of potential therapeutic targets. As you know, a lot of publication for the clinical study makes that some of these medication are not recommended for treatment anymore. For example, the hydroxychloroquine or chloroquine, because of its low efficacy and very high adverse drug reaction. Okay, how about the treatment in Thailand? First, we have to categorize patients into 4 categories. For adults, we have 4 categories: the confirmed case, asymptomatic COVID-19, symptomatic COVID-19 without pneumonia and no risk factor for severe disease, symptomatic COVID-19 with risk factor for severe disease or having comorbid or mild pneumonia. And the last one is a pneumonia with hypoxia or progression of disease. The children are categorized as the same as the adults. How about the treatment? The main treatment antiviral agents for us are favipiravir and remdesivir. And if patient have a severe disease, we will add the corticosteroid. The duration range depends on the severity. If it's a mild severity, we normally use favipiravir for 5 days. But if the moderate to severe, we can extend the duration of favipiravir to 10 days. And if the patients have problem about oral intake, remdesivir will be used. Or if patients doesn't have the effect from the favipiravir, we will for favipiravir we will use the remdesivir. Okay, so what is the gap of knowledge right now before I do this study? Actually, it's the new drugs for COVID-19 and they have a limited data for adverse drug reaction and efficacy. Okay, back to my study. The methods. This is a retrospective study and we include hospitalized COVID-19 patients who receive favipiravir or remdesivir. That's that is a mainstay of treatment in our country, and receive inpatient care at King Chulalongkorn Memorial Hospital from January to February 2021. Why we use this period? Because only this period we use only the favipiravir and remdesivir. For the first phase of COVID-19, that doesn't have a lot of publication, so we use a lot of drugs. So it's quite difficult to tell the adverse drug from which drug at that time. Okay, how about the results? The baseline demographics, as you see, 60 patients were enrolled, but only half patients received antiviral treatment based on the guideline, and about 12 patients received remdesivir, 20 patients received favipiravir. The mean age is 48 years, and 20 patients had obesity. The mean BMI was 28, and 50% of patients had comorbid, and hypertension is the main comorbidities, and no report of in-hospital mortality at my study. In the favipiravir group, the median loading dose is 3,200 milligrams, and the mean maintenance dose is 1,200 milligrams. Duration of treatment is 5 days. We found only 5 patients had adverse drug reactions. 2 patients had elevated of alanine aminotransferase more than 40 units per liter. 2 patients with white blood cell count less than 3,000, and 1 patient with hemoglobin reduction less than 11. No reports of the vomiting, diarrhea, arthralgia, or QT prolongation. We compare with the previous adverse drug reaction. We see the same, which is the hepatic and hematologic, but we didn't see about the gastrointestinal adverse reaction. For remdesivir group, the loading dose is 200 milligrams per day, and the maintenance dose is 100 milligrams per day. 5 to 10 days of treatment.
Sorry to interrupt.
Okay.
You have another 2 minutes to wind up.
Okay, and on 2 patients on renal replacement therapy, the adverse drug reaction we found only in 10 patients, and no report of infusion-related and QT prolongation. The main adverse drug is the same, is the alanine aminotransferase rising, and the second one is elevated of serum creatinine and the reduction of hemoglobin reduction. Remdesivir, as we compared to the previous ADR, we didn't see any of the anaphylaxis reaction, but we see some of the anemia and AKI elevated of ALT. So we recommended to monitor liver function test and renal function test before and after start of the treatment with the remdesivir. And we have a problem, remdesivir has problem with the renal impairment because of the SPECD for the dilution. But we used it in 2 patients. We didn't see any difference in the adverse drug reaction in this specific group of patients. And we do the pharmacist intervention, 6 cases for intervention, 5 cases for dose adjustment, mostly meropenem and voriconazole, 1 case for that interaction. The acceptance rate is 100%. And before we stop, we would like to share our pharmacist role. We do provide the pharmaceutical care by case tracking. If we have problem, we will talk to the physicians by the line application or via the phone. We do the drug information service and consultant. We do the dose and administration drug guideline, and also we do the adverse drug reaction data collection. What did we learn from our study? No serious ADR was found for both favipiravir and remdesivir, and we need more study. As you see, it was a very small number of patients, but this is a pilot, and now we are going to do a lot more about the ADR about this. It's hard to tell that the adverse event comes from the disease or the the antiviral agents. And pharmacist is an important healthcare provider for COVID-19 team. Thank you.
Thank you, Ms. Chutrat, for your presentation.
Thank you.
May stop sharing your screen, and we'll be quickly moving on to our next presenter. Participants who are interested to discuss about your presentation, you may share your email address at the chat box and interact with other researchers who have joined us here. So next, I would like to welcome Dr. Firoz A. Thamboli from Bharatiya Vidyapeeth College of Pharmacy, Kolhapur, Maharashtra from India, who will be discussing on the COVID-19 pandemic, the role of medicinal plants as an immunity booster. Welcome, Dr. Firoz. You may begin.
Okay, thank you very much, ma'am. Is it visible, my slide?
Yeah,
Yes.
yeah. You can put to the full mode. Full screen.
Okay.
Presentation mode. You make it to presentation mode. Full screen.
Okay, so just a minute. Okay.
Yeah.
Now it is okay.
Start.
Okay. Thank you very much, organizing committee, to give me a chance to present a paper on particularly COVID-19 pandemic and role of medicinal plants as an immunity booster. Whole world, this is fact. Whole world is facing COVID-19 pandemic, so there has been a lot of interest to boosting our immune system and thus build a first line of defense against the deadly virus. Since ancient times, the medicinal plants, herbs, spices were well known for their medicinal properties. Therefore, the medicinal plants and herbs playing a critical role to boosting our immunity during the COVID-19 pandemic. It is also very important to consume supplements in the form of immunonutrients such as vitamin A, C, E, D, B complex, zinc, and copper that will support our body to fight against the pathogens. COVID-19 attacks people with low immunity system and people especially people of under and overage. The immune system is built on beneficial live bacteria that lives in the gut, which protect the human body from various diseases. When the immune system response is low, weak, or damaged, it becomes an open invitation for infections such as number of diseases that coronavirus or other diseases like diabetes, heart disease, or even the cancer. So these are some drugs that is used to treat as a immune booster. In that, someone is here mentioned, that is elderberry, that is garlic, ginger, glioili, then vitellius, margosa, clove, ginseng, licorice, ashwagandha, cinchona. These plants are known to be rich in antioxidants, vitamins, proteins, carbohydrates, dietary fibers, amino acids, minerals, steroids, alkaloids, antiviral, antibacterial phytochemicals, which will help in regenerating the immune system and also helps in killing the invited viruses. Immunity, as we know, that is a very important role to protect ourselves. The immune system includes various parts of the body, including the thymus, spleen, lymph nodes, special deposit of lymphoid tissues, particular B cells, T cells, and antibodies. Immunity, the state of protection from infection disease, has both less specific or innate and a more specific or adaptive components. This is the types of immunity: adaptive immunity, innate immunity, natural, artificial, in that passive, active, and passive active. These are medicinal plants, as we know that according to the WHO, Around 80% of the world population uses herbal medicines for primary healthcare, particularly across Europe and South Asia. Research indicates that many of these herbs not only have anti properties but also build up the body's natural immunity. And unlike allopathic medicines like antibiotic, which can have serious side effects, most of these herbs spices are very relatively safe. Here, first drug, that is Sanversa. Here, elderberry, scientific name is Sanversus nigra, is an intense purple berry and Europe, and it is mainly found in Europe and Western Asia. It has been in use for many years due to its enormous health benefits. Here, particularly, some literature indicates that there is a preclinical event that elderberry, here, a replication and viral attachment of human coronavirus NL6, which diverse from that is COVID-19, but also belong to the coronavirus. Elderberry is the most effective means of preventing or combating coronavirus infection at an early stage. It mainly contains the nutritional information of elderberry. Next one is Allium sativum. That is, it is more, as we know that very well, it is antioxidant properties and also helps in the reducing stress and high blood pressure. The antiviral properties of this drug, that is reducing the severity of the cold, flu, or COVID-19 infection. Hence, garlic boosts the immune system and helps in fighting against viruses and other diseases. It has allicin, which is broad spectrum antibiotics. Next one is Tinosphoria cardifolia, that is called as a giloi, a plant being from a centuries for the medicinal values. Many studies have reported the hydrophilic, lipolytic, hypoglycemic, hepatoprotic, anti-cancer, antibacterial, anti-inflammatory, anti-obesity, anti-mutagenic property of giloi. Gudici or giloi is suggested to contain various diterpene compounds. These polysaccharides and terpenoids are immunomodulating and adaptogenic in nature. Next one is gingiber. Gingiber officinalis, that is gingiberacea. Ginger is well known for its anti-inflammatory, along with other very important, that is extensively used for curing colds and cough, nausea, asthma, travel sickness, morning sickness, etc. Immunomodulatory action of this boost of the immune system of the patients, which further prevent the load of infections. Ginger is beneficial to fight respiratory problems. Here, a relieve congestion association with the common cold, strengthened immunity due to the high level of antioxidants, reduce stress due to combination of the strong aroma and healing property, improve blood circulation due to presence of vitamins, minerals, and amino acids in ginger. It can help restore, improve blood circulation and prevent fat from deposition in arteries, helping to prevent heart from, that is cardiac vascular disorder. Next, that is ostium. As we know that it is called as tulsi or holy basil. Many in vitro and in vivo reports of animals in human proven its therapeutic potential as antioxidant, anti-aging, anti-cancer, antiviral, antimicrobial properties, immunomodulatory activity, also antipyretic activity, anti-arthritic activity, treatment of gum ulcer, kidney problems, RSM, menstrual irregulation, arthritis, anorexia, malaria are also used as a hemostatic in childbirth, etc. Interestingly, the role of tulsi for scientific evidence against COVID-19 has also been elucidated. This is curcumin longa. Again, it is called as curcumin domestica. That is called as haldi. Haldi milk. We know that very popular during the pandemic.
Excuse me, sir. Sorry to interrupt. You have another 2 minutes, sir.
Okay, ma'am. Commonly used in India for curries and other dishes. Also, it is one of the very important drugs to boost immunity of our. Then next one is the sesanem, that is we called as clove. It is also having a property. Clove may be considered as the most candidate to combat the coronavirus and protecting against the sudden death seen in some patients infected by coronavirus COVID-19, resulting from the embolism associated with hypercoagulated formation. And this particularly clove having this particularly prevent this coagulation properties. That is reported, literature reported. This Vidania somerifera, it is a very powerful aphrodisiac drug that is in, and right from the ancient, it is mentioned in our that is literature, acts as a very good, that is even a modulator. Some plants here, ginseng is very known for the immunomodulator. Fenugreek also, that is methi, we know that it is mentioned in Ayurvedic system of medicine, which acts as a natural antioxidant and a standard immune system. It is not only used as an herb, that is dried or fresh leaves, spices, vegetables, but also as a condiment in artificial flavoring of maple syrup or in the production of steroids. These are some nutraceuticals in that dietary minerals, selenium, zinc, iron, and acetylcysteine. These are mainly used as a dietary minerals and vitamins. These are probiotics, omega-3 fatty acids, and beta-glucan. So these are particularly mainly used. Okay, so conclusion is what that is medicinal plants and herbs are better alternative to treat several diseases during this COVID-19 pandemic in December 2019, where no proper allopathic medicine was available to treat COVID-19. Various traditional medicinal plants and herbs have been used as a medicine and resulting in positive health effects among the COVID-19 patients. In the present paper, that is, we have discussed the possible potential uses of medicinal plants and herbs to boosting immunity against these viruses and also prevent or even treat COVID-19 infection. These are some references. Thank you. Thank you very much.
Thank you, Dr. Firoz, for your presentation.
Thank you very much for giving opportunity.
You're most welcome.
Can be
So
used
next,
as a
you please
booster, not
start
as
sharing your screen.
measures.
We will
Maybe
quickly move to
because
our next
edit
speaker.
their
I
data
would
with
like to
you
welcome
to
Dr. B. Senthil
have
Kumar
to be
from Vinayaka Mission Medical College and Hospital from India. He'll be discussing about the phytochemical screening of polyherbal formulations used in Indian system of medicine for the treatment of COVID-19.
Sorry, you can remove your presentation.
Oh, wait.
New one can join. Yeah. Thank you.
Dr. Senthil Kumar.
Yes, ma'am. Yes, ma'am. I'm
Yes.
sharing my screen.
You may begin.
Is my screen visible, ma'am?
Yes, it's visible.
Okay. A very good afternoon to the organizing person and the chairman. And today I'm going to present the phytochemical screening of polyherbal formulation used in Indian system of medicine for the treatment of COVID-19. Ayurveda and the Siddha are the two traditional Indian system of medicine that has been practiced widely many centuries. Both system of medicine has various polyherbal formulations which were used to treat various diseases. And one of those two are, one is Kabasura Churana, is a polyherbal formulation used in Siddha medicine to treat various types of fevers, flu, along with respiratory complications. The other one is Mahasudarshana Churana, is one of the Ayurvedic polyherbal formulation traditionally used as antipyretic, anti-malarial, as well antiviral. The composition of both Kabasura Churna and the Mahasudarshana Churna, they have the common ingredients of around 20 common ingredients, which has been listed out here, starting from Picora, curcumin, curcuma longa, leucus, saccharum officinarum, andrographis paniculata, which is one of the most important ingredient, having febrifuge antipyretic activity, Glyceria glabra, Piper longum, solanum, justicia, Gingiber officinale, Piper nigrum, Hedioides, Convolvulus alpinia, Clerodendrum, and Tachispermum ami. So these are the ingredients which is commonly present in both Kabasura Churana as well as in the Mahasudarshana Churana. Apart from this, the Mahasudarshana Churana has two more extra ingredients. They are the Coriandrum sativum and the Ocimum sanctum, whereas the Kabasura Churana has one extra ingredients, that is Syzygium aromaticum. Coming on to the phytochemical screening, the preliminary qualitative phytochemical screening was done as per the standard protocol, where when compared between both Kabasura Churana and the Mahasudarshana Churana, alkaloids, carbohydrates, glycosides, cardiac glycos are present in both Churanas, whereas flavonoids were present only in Kabasura Churana. Amino acid, protein, phenols, saponins, tannins are present in both. Terpenoids, volatile oils, and vitamin C, a trace was found in Kabasura Churana, whereas it is not present in Mahasudarshana Churana. Phytosterol was present in Kabasura Churana, and it is absent in Kabasura Churana, and it is present in Mahasudarshana Churana. So this is a very basic preliminary phytochemical screening, and further the drugs has been analyzed for GCMS and HPLC analysis also, which will be discussed in later, and it will be published as a paper. At present, I am projecting only the preliminary phytochemical screening over here in this presentation. Coming on to the pharmacological activity of the ingredients that is present in both. So the gingiber officinal, which has antipyretic immunomodulator activity as well as antiviral. Piper longum, expectorant and improves the immunoglobulin A. Syzygium aromaticum is an expectorant as well as it will be resolving the laryngitis and the pharyngitis. Trachea treats high fever. Anaclus pyrethrum, which is released from the nasal congestion and problems of upper respiratory tract. Hygrophia, mainly it is used for treatment of asthma, where it acts as a bronchodilator. Termina chebulia, it is an antiviral as well as anti-inflammatory. Justitia, it is an antihistamine, antispasmodic and expectorant as well, along with antimicrobial action. Coleus aromicus helps in cold, cough and concentrated decoction, which is usually used for all the respiratory diseases and the infections in the traditional system of medicine. Costus specius, it is mainly used for anti-inflammatory, analgesic and antipyretic activity. Tinospora cordifolia, which is an immunostimulant and anti-inflammatory as well as antipyretic. Coming on to the other ingredients, Clerodendrum, traditionally used for all the respiratory diseases, asthma, fever, inflammation. Andrographis paniclata, which is the main ingredient of both the churnas, which reduces the severe symptoms associated with respiratory infections in humans. Sida acuta, treating infections, fever, asthma. Cyperus rotundus mainly act as a diaphoretic and as well as diuretic. Piper nigrum helped in treatment of bronchitis and sinus congestion. Santalum album, traditionally used for ophthalmic condition as well as virus infections. Xenos, which is used for decoction, which is used in high fever and inflammation. Moluga, anti-inflammatory as well as antiviral and antimicrobial action it has. Plicanthrus has a diaphoretic action. Trichontheus curcumin has a use for curing headache and the fever. So coming on to the conclusion, both Kabasura Churna and the Mahasura Churna both are polyherbal formulations, which is comprising of bewildering range of phytochemicals whose synergetic action is expected to balance the three doshas in our body, and that brings back the body condition to normalcy. In the current scenario of COVID, the use of Kabasura Churna and the Mahasudarshan Churna for respiratory viral infection, as well as an add-on formulation for alleviation of symptoms of COVID-19 viral infection is found to be rational. These are my references. Thank you. Thank you, ma'am.
Thank you, Santil Kumar, for nice presentation.
Thank you, sir.
Thank you for your presentation.
Thank you, ma'am. Thank you, ma'am.
You can invite next presenter.
Yes. So shall we move on to the next presenter, sir? Dr. Subhash, sir?
Yeah. We can go.
Okay, sir.
We are within time, no? So no problem.
Yes, we are on time. So next I would like to welcome Ms. Laiba Salim, who will be discussing on the Remdesivir and antiviral drug against SARS-CoV. Laiba Salim, she's from Aligarh Muslim University from India.
Hello everyone, I am Laiba Salim, a sophomore undergraduate from Bachelor's of Science, focus in Industrial Chemistry from Aligarh Muslim University, Aligarh, India. So the presentation is Remdesivir, an antiviral drug against SARS-CoV-2. We all are familiar with the fear in our life due to the global pandemic. SARS-CoV-2 appears to be a spectrum of clinical presentation ranging from asymptomatic to severe respiratory failure. Topic of pandemic, scientists are working on war footing to develop new therapeutics, vaccines to combat the COVID-19. In this life-threatening scenario, the drug repurposing of already used medicine has become an effective strategy to short run. And the various drugs are in the various phases of clinical trial, such as hydroxychloroquine, azithromycin, chloroquine, remdesivir, etc. Among them, remdesivir has emerged a promising candidate, which was originally evaluated in the clinical trials to combat the Ebola outbreak in 2014. Remdesivir, a broad-spectrum antiviral, is an intravenous product of adenosine analog. Talking about the history and development of remdesivir, remdesivir was originally developed by Gilead Sciences and emerged from a collaboration with US CDC and US Army. A hit-and-trial method of 1CA and adenosine Nucleoside, along with a product form of monophosphate, was found to be highly potent against a panel of RNA viruses such as yellow fever virus, dengue virus, influenza, SARS, etc., and had gone through various primary assays such as cytoprotection effect, etc., and was found to be highly potent. Moving to the synthesis of remdesivir, as you can see on the screen, remdesivir can be synthesized in multiple steps from the ribose derivatives. Various intermediates are formed. Intermediate A can be prepared by L-alanine and phenyl phosphorodichloride in the presence of triethylamine and dichloromethane. Similarly, intermediate B and C can be formed with various reagents and respective reactant. And then the isomers were separated via reverse phase HPLC. Remdesivir can be obtained through chiral-resolution methods. Talking about the action against SARS-CoV-2, I would like to give an example. If a motorbike wants gasoline and you put more and more petrol in it, it would be of no use, and the machinery of the motorbike would be jammed. Same is the case with remdesivir. Remdesivir is a nucleoside prodrug which perturbs viral replication by binding to viral RNA-dependent RNA polymerase, inhibiting viral replication through premature termination of RNA transcription. Nucleosides analog are synthetic acyclic nucleoside phosphonates that are intentionally designed to circumvent the phosphorylation process. These compounds mimic their physiological counterparts, and the integration of these lead to the hurdle in molecular processes. Let's see how. So, when the coronavirus enters in a cell, in a ribosome, positive RNA sense is created and translation occurs through RDRP, and the RNA replication takes place, which in turn forms assembly of mature viron and packaging by endoplasmic reticulum, which in turn lead to transmission of diseases from one person to another. But what happens when a remdesivir is administered in a body? This remdesivir is a product component, and the nucleoside core, when it is administered in the cell, it undergoes successive phosphorylation, and which results in active metabolite, which perturbs viral replication and delays the chain propagation. And in turn, it leads to the hurdle in assembly of viron making, and thus prevent the disease from transmission.
Excuse me, Ms. Naima, you have another 3 minutes left.
Yes, ma'am. Okay, so the bioactivation of remdesivir. So it was found out that in the bioactivation of remdesivir in vitro, so there is a ideal bioactivation. In comparison to in vivo, there is the presence of enzyme, enzyme like kinase, which delays the activation. So coming to dosage and efficacy of remdesivir, so remdesivir is the first drug to receive USA FDA approval for the treatment of COVID-19. Talking about the efficacy of the other drugs by the, I'm really sorry. In the survey by Glade Sciences, it was found out that the hospitalized patient with severe COVID-19 who received remdesivir instead of placebo recovered 31% more quickly, allowing them to leave the hospital after about 10 to 11 days instead of 15 days. So coming to conclusion, there's an urgent need of the global efforts currently underway towards the discovery and development of vaccines and therapeutic agents for the prevention and treatment of COVID-19. And yet, drug repurposing is another important strategy for the development of effective therapeutic against COVID-19 in limited time frame. So we anticipate that in upcoming months, biologists will identify new targets and medicinal chemists will screen more drugs and molecule libraries against novel and existing targets in their efforts to find a cure for COVID-19. Thank you for a valuable time.
Thank you, Laiva, for a nice presentation. I have a question. Do you have an idea about the mutants, the effectiveness of the remdesivir against the mutants? You know, the virus go for mutation rapidly. So is there any idea about it? Do you have gone through the articles or the research?
No, sir, I really don't have any idea about the mutation.
Okay, thank you. Nice presentation. Thank you.
Thank you, Laiba, for your presentation.
Thank you so much, ma'am.
Participants, if you have any questions or interactions, can be done in the chat box. So next, moving on to our next presenter. Next, I would like to welcome Bhagyashree Agarwal from LCIT School of Pharmacy from India. She'll be discussing on the role of recent technologies and its importance in pharmaceutical education due to outbreak of COVID-19.
Am I audible?
Yes, you're audible. Kindly share your screen. You may begin the presentation.
Okay, ma'am. I have already shared the screen. Is it visible?
Yes, you may start your presentation.
Okay. Good afternoon, everyone. I am Bhagishri Agrawal, Assistant Professor
Excuse me,
from LC.
Ms. Bhagishri,
Yes.
you haven't started playing the PowerPoint.
Oh, just a second. Is it playing?
No, it's not playing now. You may start playing it.
Is it visible now?
No, it's not visible. It's your screen. Share screen.
It is not visible.
Now, is it visible?
No. Yeah.
Yes, yes. You may put it on full screen.
Okay, fine.
It's a poster presentation, right?
No, I opted for oral presentation. I didn't know why it is poster written in the manual.
Because poster takes only 5 minutes, no?
No problem. I'll go for that only.
Okay,
I would like to introduce something which was my real experience as assistant professor in a mediocre college in a mediocre city in Chhattisgarh, Bilaspur, which is in a small state in India. So at the time when education ministry started that no education institute will have the physical classroom, the online mode of teaching will be conducted. It was not easy for a student, for a teacher, both to start the teaching in a both the education in a online mode. That time we opted two apps, Zoom and Google Meet, from which we were ready to use the syllabus, subject module, assessment methods, everything by that only. But we developed that Google Classroom is more convenient as we have the four quadrant method, which is more useful for the student and the teacher. In the four quadrant method, we were using first quadrant as teaching content, second as e-lecture, third as assessment, and fourth as discussion. In the teaching content, we share the notes, PDFs of the books, any other presentation prepared by the educator. Then the e-lectures, it was the recording of the classroom. After that, we provide the assessment, which is MCQ question, marking scheme, grade sheets, and afterwards, the last one is discussion, so that the doubt clearing session, educational chat, and QA session can be performed with the student and the teacher. Their fellow mates can also answer in the discussion forum. My real experience as educator, it was like virtual classroom was not easy to accept, but as the time flows and the situation become more challenging, everyone has to accept it, and now the time is we are enjoying it. Now we are very much interested in doing education from this. The pros and cons of this system. First pros is health is wealth. If we are safe, we can study more. Second, it is an interactive learning. The attentive students, the technology savvy students, they get to learn more from this. The student become more responsible when they have to study by home. And the cons included some technical and network issues, which is very common in mediocre cities. Some students are not that up to the mark. They are ignorance in their attitude for the study. So they are in the lack of availability of gadgets are the problem, and the most important con was the practical exposure to experimentation was missing. So finally, I would like to conclude that the technology helped the educators to serve better during even at the outbreak of COVID-19. Also, it establishes the proper contact between the student and the academic staff for the better learning. So it is a type of icing on the cake, so that the maximum learner can learn it from their home, as well as they get their education on time. So that's it. That is from my end. Thank you.
Thank you, Ms. Bhagishri, for your presentation. You may stop sharing your screen.
Oh,
Moving on to our
thank
next
you.
presenter. Next, I would like to welcome Pragati Singh from Dehradun Institute of Technology, India. She'll be discussing about the status of COVID vaccination in pregnant and nursing women. Ms. Pragati?
Ma'am, is my screen is visible?
Yes, yes.
Good afternoon, everyone.
You are not audible. Unmute your mic and talk.
Am I audible?
Yes, yes.
Pregnant women who get coronavirus vaccine may pass their antibodies to the newborns. And getting vaccinated is totally a personal choice. The COVID vaccine
Voice is breaking.
Pragati, you're not audible. I request all the other participants to kindly mute from your side so that there is no interruption. Only the presenter can share your audio. Pragati, can you please enable your audio?
Ma'am, now is it visible? Now is it audible?
Yes, yes, please continue.
Okay. Getting
Pragati, you're not audible.
She is not audible, I think.
How is it audible?
Yes, please continue.
COVID-19 mRNA vaccine generates robust protection in pregnant and lactating women with the immunity level and side effects similar to that observed in non-pregnant women. And in addition, they add immunity and transfer it via placenta and breast milk to the newborns. There's a transfer of antibodies in the cord and breast milk, suggesting that vaccine also deliver antibodies to babies. Vaccine-induced antibodies were detected in all umbilical cord blood and breast milk sample. Gestational age of maternal antibody production includes the level of SARS-CoV-2 specific antibody that is detected in cord blood specimen, implying that there may be ideal time for maternal vaccination prior to delivery of optimized protection to the fetus. IgG detected in newborns with negative IgM and negative result on polymerase chain reactions. The SARS-CoV-2 specific antibody appear inefficiently transferred across the placenta following third trimester. Vaccine dose increases specific IgG immunoglobulin detected in maternal blood and breast milk, but not IgA. Pregnant people are at increased risk for severe illness from COVID-19. Although overall risk for severe illness is low, pregnant people are at increased risk for severe illness from COVID-19 where compared to non-pregnant people. Severe illness includes illness that result in intensive care admission, mechanical ventilation, or death. Additionally, pregnant people with COVID-19 might be at increased risk for adverse pregnancy outcomes such as preterm birth compared with the pregnant woman without COVID-19. V-Safe Pregnancy Registry. In terms of safety data, more than 60,000 pregnant women have signed up for government's V-Safe program, which monitors people who have received a COVID-19 vaccine. VSafe is a CDC smartphone-based tool that uses text messaging and web service to provide personalized health check-in after vaccination. A VSafe pregnancy registry has been established to gather information on health of pregnant people who have received a COVID-19 vaccine. If people enrolled in VSafe report that they were pregnant at the time of vaccination or after vaccination, participation is voluntary and participant may opt out of any time. People who are breastfeeding, clinical trial for COVID-19 vaccines currently authorized for use under emergency use authorization in United States and did not include people who are breastfeeding because vaccines have not been studied on lactating people. There are no data available on safety of COVID-19 vaccines in lactating people, the effect of vaccination on breastfed infants, and the effects of milk production or excretion. COVID-19 vaccines authorized now are non-replicating vaccines, meaning they are able to create an immune response but do not reproduce inside host cells. Because non-replicating vaccines pose no risk for lactating people or their infants, COVID-19 vaccines are also thought not to be a risk to breastfed infants. Therefore, lactating people may choose to be vaccinated. And conclusion, as per the various studies, pregnant and nursing women receive as much
Time is up.
from vaccine. Yes, ma'am.
Your time is up. Can you please conclude?
Yes, ma'am. Conclusion of protection from vaccine and non-pregnant women. Therefore, there is no ethical reason to not include pregnant women in phase 3 trial of COVID-19 vaccine. And there are clear evidence that COVID-19 vaccine can induce immunity and will protect infants and does not elucidate any potential risk to people. And the future directions, there have been recent calls for the pregnant women to be included in early stage of vaccine trials in order to protecting them and their newborns. It also highlight important next steps. Larger studies include to investigate when the best time in pregnancy is to vaccine. There should be detailed analysis of how mothers respond to vaccine at different stages of pregnancy, whether the vaccine prevents placental inflammation and preterm birth, what effect the timing might have passive immunity in newborns, and how effective is the immunity transfer to infant, and how long the vaccine-induced viral immunity. Ma'am, now it's complete. Thank you.
Thank you, Ms. Pragati, for your presentation. So next we are moving on to our track 2 on drug delivery technologies. So first I would like to invite Dr. Sunita Reddy from JNTUH, India. She'll be discussing about the lipid-based drug delivery system of ibrutinib, enhancement of bioavailability in vitro, ex vivo, and in vivo studies.
Thank you, Ms. Mustafa. Thank you. And can I request the before presenter to stop sharing?
Pragati, remove your poster.
Pragati, you may stop sharing your screen.
Pragati, remove your poster.
Or you guys can stop the sharing, whoever the host.
Yes, yes.
Yeah.
Yes, ma'am, you may begin.
Yeah, I can share this.
You can start sharing.
Yes, yeah. Okay, good afternoon. Good afternoon, everyone. I can hear some noise, please. Okay, good afternoon, everyone. I am Dr. Sunita Reddy from Jawaharlal Nehru Technological University. I'm going to present on lipid-based drug delivery systems of ibrutinib and in vitro, ex vivo, and in vivo characterization. I'll be discussing very brief on each and every aspect of this, that from introduction to the summary and conclusion. And most I'm going to concentrate on in vitro and ex vivo and in vivo evaluations of liquid self nano emulsifying drug delivery systems or solid self nano emulsifying drug delivery systems. Okay. And you know that the world is making a lot of new drugs, but 40% of new drugs are new moieties are they're poorly soluble. They are very poorly soluble, and they will be belong to class 2 or class 4 drugs. And at JNTU, we have a facility that will take that class 2 drugs and class 4 drugs, and we'll develop into self-emulsifying drug delivery system. So far, we have developed about 14 to 15 drugs of different categories, from starting from the antiviral drugs and beta blockers and calcium channel blockers. Okay, and these as a sets have a specific nature in such a way. Practically insoluble drug we are going to bring into soluble liquid form, and that we are going to keep the drug into oils and surfactant and co-surfactant, and this can be delivered as a solutions as an oil phase, or we can convert into the solids, and we can compress into tablets or capsules. Okay, so this is a literature review, and we have to thank the Fountain because he has given all lipid-based excipient study and how to do it and how to select it. And so far we have done several drugs, like for example, I have given here ephedrins and azilsartan, one of the drugs, and which was published. We have published about 15 to 20 papers on it. So my objective of today's presentation is this one, and I have selected the ibrutinib, the anti-cancer drug. We have done the validation by HPLC method, and we have done the we have checked the solubility studies and compatibility studies. Then we have chosen the oil, surfactant, co-surfactant. Then we have formulated, and we have evaluated by different ways of evaluation, and we did not leave any of that. Okay, and ibrutinib is one of the anti-cancer drug, and we have a log P of 3.97. When you do the self-emulsifying drug delivery systems, we have to select the log P value close to 4. Then that is a better way that we can do. Okay, and we have done the method development for this by HPLC method, and we have done like 8 different trials. And the last one, the orthophosphoric acid and acetonitrile is the best one, and we proceeded because it is RT value is 2.5. And this is our calibration curve, and is a perfect. We got 0.9996, and this is the chromatograms. You can check it. This is the blank, and this is the drug. And after that, we have done the linear regression, system suitability, sensitivity, precision, and accuracy. Because this has to be within the limits, then only we can proceed the drug evaluation. And this is already published data, and we are within the limits. Okay, and then we proceeded with the solubility studies of ibrutinib. You can see here we have selected several components, and these are all from the Getafase products who has given by the France company, and some of them are BASF drugs, like we have a Capamil, Labrosol, Capriol, Coliferase BASF, Labrosol, and all these, the oil, surfactant, co-surfactants, these are supplied by the Getafase products, and we have checked, and we end up with a Capriol as an oil which has a maximum solubility, and Colifor EL is the another surfactant and transcutol. And we made a formulation. Before we went to formulation, we have checked with the FTIR studies to know the any incompatibility with this excipients with our drug. This is our drug FTIR, and you can see in the bottom that we have checked with the Caprior, we have checked with the Colifor, we have checked with the transcutal HP, and we have done the combination of them to check whether these groups, whatever the groups which are present in the drug, whether they are retained or not. And we came to know they are well and good, and they have retained, so we proceeded with the formulation. Before formulation, a pseudo ternary phase diagram is important to know where we have having the self nano emulsifying region, because we have done the micro emulsions, but the stability was the problem. So we want to know whether they are within the nano emulsions or not. And this is called water titration method. We have to select the different concentrations of oils and S mix, the surfactant and co-surfactant mix. Here we have taken 3.1 as a surfactant mix, and we have taken 1:9 to 9:1 from the oil to surfactant, and we have done the titration study, and we and 2:8 and 1:9 The best ones, which is bluish white emulsions or bluish transparent emulsion. Then we went through the plotted the triplot 3.4 version, and this is the bluish portion is the self nano emulsifying portion, and the greenish one is the green emulsion, the orange one is the coarse emulsion. And we have done the same thing with the 4.1 S mix and 3.1 S mix to know which concentrations we are getting the nano emulsions. By doing this nano emulsions, not only that, we went to again composite design to select which is the best droplet size that we can get it. And we have done the 15 runs by selecting these concentrations 3:7, 2:8, and 1:9. And out of this composite design, it has given 2 and 11 and 14 are the best one with a self-emulsification time is like within a few minutes, and the droplet size is below 50 nanometers. And to know that whether they are with fit in or very any significant difference is there, then we have done the ANOVA table, and they are very significantly different, and we selected 2, 11, and 14. So then we have done the preparation of the snets by keeping the drug as a 40 milligrams, and we have changed the concentration of oil and surfactant and co-surfactant, and we have prepared the self-emulsifying drug delivery systems, and we evaluated them by self-emulsification time. And you know that below 30 seconds of self-emulsification time is the best time, because when we give the oral preparation, they will be entered as a solution with a mild agitation in the stomach. They will become an oil in water. From there, the drug will be released. Okay, so here you can check that 2, 11, 14 are within below the 30 seconds. And we have done the dispersibility test. And if they are in the grade 1 means the particle size is very, very nano size, and they are transparent. Okay, and then we have done the phase separation studies. There is no phase separation, and we have diluted the samples with water and HCl and phosphate buffer, the simulated gastric fluids and intestinal fluids to see whether any separation is there. Then there is no separation. And also we have checked the drug loading efficiency, because ibrutinib available like a 70 milligrams, 170 milligrams and 250 milligrams in the market, and person has to take about 4 grams per day, the people who have a lymphatic leukemia. So this is the study we have done. We have taken only 40 milligrams, but the drug efficiency was 98% to 99%. So you can reduce the dose and give a small dose to give a good efficiency, and the loading of the drug to the any human can be reduced. Okay, and droplet size, we have done the MAL1 zeta sizer. They are below 50 nanometers, and zeta potential is very good, that indicating they have a repulsive forces. And here is the study that I can show. These are all, this is 2, and this is the 11 and 14, the zeta sizing.
I'm sorry to interrupt.
Yeah.
Ma'am, you have another 2 minutes to wind
yes,
up.
I'm going to finish in it. Okay, and this is the zeta potential. Okay, and here we have done the study for the in vitro drug release for the liquid snets, and it say that 99% is regret. And once the liquid snets are better, then we have converted into solid, and we compare the liquid and solid with the pure drug, that indicating about 98%. After that, we have done the ex vivo study in the stomach mucosa we have taken from the sheep, and we have studied here, it indicating the liquid snets has released very fast, but the solid snets have released at a given time within a 90 minutes, about 99%. Then we went to the animal study, and in a Winster rats, with the, here you can see the graph. This is solid snets has releasing about 20 to 30%, and liquid snets has released a very highest amount, and solid snets are released about 98% to 99%. So, and when we study the relative bioavailability, it is 140 to 130% that we can.
To conclude.
Yeah, this is the conclusion. Here it is the based on the solubility studies we have done selected here, and we have done the pseudo-ternary diagram to know the concentrations. Then we have run the composite design to select the formulation, and we have done the study in vitro, ex vivo, in vivo, and within the 95% of drug release within the 30 minutes. Okay, that is an excellent study. And self-emulsifying drug delivery system, especially self-nano-emulsifying drug delivery system, the best choice for the alternative to oral therapy. Okay, thank you very much. Any questions on it?
Thank you for your presentation. Remove your poster.
So next I would like to welcome Dr. V. Kamala Kannan. From JKK Nataraja College of Pharmacy from India. He'll be discussing about the development of time-delayed capsule device for chronopharmaceutical drug delivery system.
I don't know, heavy sound is coming from which place?
Dr. Kamalakanan, can you remove one device? You have logged in from two devices. You may start your presentations. We are running short of time.
You are not audible.
So please unmute and please begin your presentation. So can you please unmute and begin your presentation? So you can move on. We can move on to our next presenter. Maybe you can meanwhile check your audio and come back to us. So we will move on to our next presenter, Mrs. Matthew.
Difficult to listen to you.
So there's an audio problem, sir, from your end. It's very disturbing. We'll be moving on to our next presenter. Meanwhile, you can look into the problem and resolve it. Kindly stop sharing your screen. We'll be moving on to our next presenter, Mrs. Roma Matthew. Ms. Roma, can you please begin your presentation?
சரி இது ஆஃப் பண்ணுவோம், ஏதோ ஒன்று ஆஃப் பண்ணுவோம். வால்யூம் ஃபுல்லாக வெயிட் பண்ணுவோம். இல்லை வால்யூம் ஃபுல்லாக ஜீரோ பண்ணுவோம். இதை வைக்கவே கூடாது சார், ஜீரோ. மொபைல் யூஸ் பண்ணுவோம்.
Yes, Roma, your screen is visible. Please begin.
Good afternoon. I welcome you all to the oral presentation.
Madam, my voice is clear.
Solid self-emulsion.
So we will come back to you, sir. We have begun the presentation with Ms. Roma.
Okay, next I will give, madam.
Yes. Yes, Roma, please continue.
I welcome you all to the presentation of my topic, solid self-emulsifying drug delivery system for enhancement of oral bioavailability and overview. Oral route has been a preferred route of drug administration due to several advantages like safety, efficacy, ease of administration, patient compliance, high degree of flexibility, etc. But the wide acceptability of oral route is limited due to its performance based on bioavailability of drug, which resulted in variable absorption rates and variable serum concentration. This problem of poor bioavailability led to the development of new dosage forms, one of which is self-emulsifying drug delivery system, which is a very promising approach, which consists of isotropic mixture of oil and surfactant, which forms emulsion within the gastrointestinal fluid when it comes in contact with the gastrointestinal fluid components, that is the aqueous component, under the mild agitation provided in vivo, thereby enhancing the oral absorption of the drug. Now, the method of preparation for self-emulsifying drug delivery system is quite simple, which involves droplet size homogenization as well as reduction using equipments like homogenizers and microfluidizers. The experimental part of evaluation also is quite simple in the sense dispersibility tests, rheological properties, stability assessments, dilutions, durability evaluation, zeta potential measurements, emulsification potential, zeta potential, in vitro and in vivo bioavailability studies. So this is a very promising approach for enhancement of oral bioavailability. Now coming to our topic, that is solid self-emulsifying drug delivery system. Several studies have been done in self-emulsifying drug delivery system, and many drugs are in the market. An example of which is Neorol, which is cyclosporin self-emulsifying drug delivery system in the form of soft gelatin capsules. But there is a disadvantage that is associated with liquid sets, that is the presence of hygroscopic material that may lead to dehydration of the capsule cell, or polar molecules that may migrate and penetrate into the capsule shell. Therefore, the need for solid self-emulsifying drug delivery system is very important, which provides advantages like lack of contact of liquid material with capsule cell, low production cost, convenience of process control, high stability and reproducibility, and better patient compliance. So these are some of the methods that can be used for converting liquid sets to solid sets, important ones which are hot melt extrusion, adsorption onto solid carriers, pelletization, spray drying, and freeze drying. Now a short discussion on those topics. First one is encapsulation of liquid and semi-solid sets, which is a simple technique where liquid sets are simply filled into gelatin capsules and sealed using microspray or bandit process. In the case of semi-solid sets, it is first melted and then incorporated into capsules and then cooled, and the product is used. Another technique is the conventional spray drying technique, which include conversion of liquid sets mixed with the solid carriers and a suitable solvent, and the solvent is atomized into a spray of fine droplets, which is introduced into the drying chamber and then connected, converted into solid particles, and the dry particles can then be filled into capsules or made into tablets. Another method is adsorption to solid carriers, which includes liquid sets that is adsorbed onto free-flowing powders that possess very large surface area and are capable of absorbing high quantity of oil material. Mixing liquid sets and the adsorbent in a blender or by physical mixing can be done. This is a very simple technique and very widely used. The solid carriers used for this purpose can be microporous inorganic substances, high surface area colloidal inorganic substances, or cross-linked polymers. Another technique is spray congealing or spray cooling, where the molten formulation is sprayed into a cooling chamber and the mixture comes in contact with cooling air and the molten droplets congeal and recrystallize into spherical solid particles which will be collected and formulated into solid dosage forms. Another one is melt granulation technique. Here it's a one-step process where the powder agglomerates are obtained, adding binder that melts or softens at low temperature. Now, if it is a thermoplastic property material, thermoplastic volatilization can be used. This is used for the formulation of solid dispersion, sustained release formulation, and immediate release formulation. Now, coming to another technique, that is supercritical fluid technology. Here, supercritical fluid technology uses supercritical carbon dioxide. The lipid materials may be used in supercritical fluid technology either for preparing solid dispersion or for coating drug particles. The drug and the lipid excipients are dissolved in an organic solvent, and the coating process involves dispersing the drug particles in supercritical fluid containing coating material. The solubility of the coating material is sustained by elevated temperature and pressure. The coating is facilitated by gradual decrease in pressure and temperature, which increases the solubility of the coating material, leading to granule deposition onto drug particles, forming solid particles. Then you have exclusion spheronization technique, where the solvent-free technique that converts pellets using exclusion and spheronization process. The liquid set is first mixed in a blender, then water is added until the mass is become extrudable. Then the extruded mass is then spheronized to form uniform size pellets. Pellets are then dried and stay separate. Now, this is a comparison of the different methods that are used for quantification of sets. Now, all these methods have their own advantages and disadvantages. For example, spray drying and hot melt extrusion provides solid powders that is having excellent formulation characteristics but cannot be used for formulation of thermolabile materials. In that case, you can go for adsorption to solid carrier. But this is a comparison of liquid and solid cells. The third and the fourth bottle was consisted of microemulsion that was formulated using. The second one is formulated from liquid sets, and the fourth one is formulated from solid sets. Now, studies have shown that the reconstituted products from both the liquid and the solids have shown similar characteristics. Now, finally, the solid dosage form, solid drug particles can be encapsulated or converted into different dosage, high emulsion, capsules, tablets, pellets, solid dispersions, microspheres in plants and suppositories. To conclude my overview by saying that oral route has been the major route of drug delivery for the treatment of many diseases. Major limitation of oral route of drug administration is that drugs taken orally for systemic effects have variable bioavailability. Sets is a promising technology. Now, solid sets is formulated by incorporating liquid sets in powder form by different solidification techniques, which are very simple. Advantage of solid sets include low production cost, convenience of process control, high stability, and better patient.
Thank you, Ms. Roma Matthew, for your presentation.
Can go for next.
Yes, so we will just check with Dr. V. Kamala Kannan whether he's ready with his presentation.
Yes, madam, I am ready, madam.
Yes, yes. Kindly share your screen. I'm requesting all the other participants kindly mute your audio so that the presenters are not interrupted. Yes, sir. Kindly share your screen and you may begin. So can you please begin?
Yes, madam.
Sir, share the message. Share பண்ணுங்க. You just share it.
So we are running short of time. Shall we move on to our next presenter if you're finding difficulty sharing the screens?
We are shortage of time, running shortage of time.
Yes, so your screen is visible. Please begin.
Yes, madam. Today I am going to discuss about for development of time-delayed capsule device for chronopharmaceutical drug delivery system of dilithium hydrochloride. So, chronopharmaceutic is mainly for time-delayed capsule device formulation. Chronopharmaceutics is a branch of pharmaceutics mainly for designing for evaluation of drug delivery system of bioactive agents, mainly based on the rhythmic circulation. So I am going to designing about for pulsing cap design formulations. That pulsing caps we have for designing using for hydrogen plug, mainly using for the polymers. So what is the aim of the work? Aim of the work mainly for us going to designing of site-specific drug delivery system. And the drug delivery system is going to delivering on the specific site of action, mainly under a specific time of period. That using for the polymers and copolymers, that is specific for the pH sensitive polymers we are going to using. The designing of formulations mainly for the oral formulation using for intended and they are giving for angina pectoris, that is for chronobiology of angina pectoris, especially in the site specific for pollen or targeting. That's especially for chronopharmaceutic approaches given for treating in the angina pectoris. That is the main aim for formulation. And we are going to, what is the main objective? That is the objective for that is going to synchronize of drug delivery in the circadian rhythm and provide the maximum drug delivery and minimize the frequency of drug administration and improve the patient's compliance. That is the main objective for the formulation, our formulations. And we are going to designing for the formulation mainly for first we have to designing for preparation and evaluation of microsphere of dilithium hydrochloride. Then after we have to designing of the formulation for pulsing cap formulation. That is the main planning of work of then we have for the literature surveys did that, and we have for drug profile for dilithium hydrochloride, that is for calcium channel blockers, and we have to go for the experimental works. Mainly we have did for the pre-formulations, then after for drug experience comparative studies and hello.
Yeah, continue.
Yes, sir, continue.
Yes, sir. So experimental work I did for the pre-formulation studies, and then after for drug experience comparative studies for FDAR, then finally for drug experience comparative DS studies, that is for pre-formulation studies. Then after, first steps is going to involving for the preparation of dilithium hydrochloride for the microsphere formulation using the method for solvent evaporation technique method. For that we are going to using for getting more yield value. For that purpose they are going to using for solvent evaporation technique. And we have to preparing for the formulation for 24 formulation. Mainly for that is going to optimize the formulation based on the dissolution studies. So we have to optimize, we are using the polymers mainly for utrogen and utrogen RLPO, RSPO. Totally 4 polymers they are going to use. For that is going to optimize for one formulation, that is for FM13. And this FM13 is going to optimize based on the dissolution studies. So this is the formulations we are going to using prepare for that. The first steps is going to involving the preparation of microsphere. Next we have to go for that. This is the evaluation steps is involving for the microsphere. That is getting the better formulations based on the evaluations. Mainly they're using the procedure for percentage yield value, entropy efficiency, particle size determinations, and go to particle size reduced on the same photography, and in vitro studies. Based on these evaluations, we are going to optimize one formulation, that is for FM13. And the next steps is going to involving the preparation for hydrogel plug. This is going to filling in the, after filling the capsule, they are going to plug
time
it.
is up. Kindly conclude.
Yes, madam. Yes, madam. So hydrogel plug, as we prepared for that, using for the polymers for hydroxy HPMC, that is where maximum giving for the more capacity for in the hydrogel plug. That using the different formulation for FM, FP, 12 formulations are prepared. From that is going to optimize it for that one formulation. So based on the capacity, this is the evaluation involving for the preparation of hydrogel plug. And finally, we have to go for the preparation of the capsule formulation. This is for the filling of formulation, especially for this is a formulation. These formulations. So this is the formulation. So this is a formulation. For this is going to using filling of the capsule along with the hydrogel plug. Then is going to be coating with the cellulose coating. So that going to maintain for the long duration of action, especially for the lactam, they have to maintain it. And we have to go for the dissolution studies. Mainly before that, we have to for the pre-formulation studies. That is for we have to go for standard flow for different pH. That is for 1.2 and 7.4 and 6.8. That different pH we have to using, and we have to go for the FDA studies. That is mainly for the compatibilities, and we have to did for the DSC studies. That is also for that compatibility for the drug and excipients compatibility, and we have to determine the particle size discriminations. For that is going to optimize the formulation for one formulation based on the particle size, and we have to determine for especially for dissolution studies. So dissolution studies also we have to do for that, and same photographies. This is same photography, and we have to go for the dissolution studies. So this is going to optimize the formulation for FM13 is the best formulation, and especially for we have to go for FC4, this is optimized formulations based on the lag time. So lag time especially for 7 hours. So what is the concept of the formulation? We have to giving the treatment, especially for angina pectoris, that angina pectoris in the early morning time. Our formulations we are taking in the night time. That right time they're going to releasing of the formulations. So this is the different evaluation steps is going to involving for is going to optimize the formulation mainly for dissolution studies. Then after for stability studies and same photography studies, different steps is going to involving for this going to optimize the formulation. For that is going to optimize that our formulations from that is optimized for one formulation, especially for FC4. That is the formulations is going to optimize that formulations giving for better treatment, especially for angina pectoris. Means that we have to take from the night time, that going to release in the early morning time. That 7 hours we have to stay in our body, especially for that based on the
so we have
biology.
To stop the presentation. Time is up. So thank you, Dr. Kamalakanan, for your presentation.
Next.
Participants, if you have any questions or interactions, you may chat on the chat box provided. With this, now we are moving on to our next presenter. I welcome Mr. Nitin Singh, who will be discussing on the designing and experimental application of emulsion-based hydrogel for topical delivery. Mr. Nitin Singh is from Jivaji University, India. So can you please stop sharing your screen?
Hello, ma'am.
Yes, Nitin, you may begin.
Stop sharing your screen. New one can join.
Yes, we have.
You can remove him.
Ma'am, you can't start the screen.
You can remove him. Yeah.
Nitin can start your presentation. You can start sharing.
Ma'am, while the other participant is sharing, I can't start the screen.
1 minute. 1 minute. 1 minute. Yes, now you can. Yes, Nitin, please begin.
Hello, ma'am. Good afternoon, ma'am. My topic: designing and experimental evaluation of
Please start playing your slide.
micro-based hydrogen for administration drug through undergo first pass metabolism. Recently, drug delivery system via skin.
Excuse me,
Ointment.
Nitin, your presentation is not visible. Can you please display your poster? Please share again. Display your poster again. Yes, yes. Now it's visible.
Yes, ma'am.
Okay, please continue.
Topical drug delivery is the one for the suitable route of administration for drug that undergo first pass metabolism. Recently, delivery of drug via skin using ointment, cream, and gels have been critically investigated. Bifonazole
You are not audible, Dr. Nitin. You are not audible.
Yes, sir.
Yeah.
Bifonazole and imidazole antifungal drug that has been used many skin and nails infection caused by fungi, highly lipophilic drug with a very short half-life, 1 to 2 hours, and minimally absorbed following dermal application, 0.6% of the applied dose.
You did something else. Green, I think.
Objective: the microemulsion bifonazole assumed to increase the cuteness absorption. Thus, it assumed the topical microemulsion of bifonazole with increased bioavailability by controlled release of the drug with longer relief with time with favorable treatment, fungal infection, and symptoms relief. After formulation, pre-formulation STD FTI spectra of bifonazole, the sample IR was interpreted and matched with reference IR spectrum is shown in figure of functional groups present in sample drug which authenticated of the purity of the drug, the study of the proof that the gift sample of drug thermogram of the degree centigrade and corresponded to the narrow melting range and crystalline nature. The normal melting range of the drug is 149 degrees centigrade to 154 degrees centigrade, and this graph depicts to exact melting point from 20 degrees optimized and specific optimization pituitary phase diagram. From the pituitary phase diagram, micro ratio 1, 2 ratio 1, 3 ratio 1, and 4 ratio 1. So in figure, the phase diagram at 3 ratio 1 being ratio obtained highest area of emulsification, and so it was selected for further formulation. And different formulation, ma'am.
Kindly conclude. Your time is up.
जी मैम, okay मैम.
Shall we move on to our next presenter?
Yeah.
The optimization calculate the optimum to 6.59 drugs contain 1993.1. Disputability the range 4.6 to 7 gram in the viscosity. Study indicates the pastry plastic barrier or microemulsion based gel formulation. The study microemulsion gel tamarind gum containing gel exhibited desired drug release 94.3% at the end of 24 hours.
Mr. Nathan, thank you for your presentations. We'll be moving on to our next presenter.
Welcome.
So next I welcome Mr. Ramkant Joshi.
Am I audible, ma'am?
From Shivaji University, India, who will be discussing about the transdermal delivery of fluorbuprofen using natural penetration enhancer, effective management of rheumatoid arthritis.
Am I audible, ma'am?
Yes, you're audible. Kindly share your screen.
Yeah, ma'am.
Yes.
My screen is shared. It is visible or not?
Yes, it's visible.
Yeah. Very good afternoon to all dignitaries. Today here I am going to present my PhD research work on the topic of transdermal delivery of fluorbuprofen using natural penetration enhancer, effective management of rheumatoid arthritis under the guidance of Dr. Navneet Garud sir. So the present investigation for the research was to develop matrix type of the transdermal drug delivery system of fluorbuprofen with various ratios of the matrix polymer hydrophilic and hydrophobic concentration of plasticizer and natural penetration answer by box-Benton statistical design to investigate the combined outcome of selected independent variables for effective management of rheumatoid arthritis. So the influence of binary mixture of polymer plasticizer and penetration answer on physicochemical characterization like tensile strength, percent elongation, etc. in vitro drug dissolution study, in vitro drug permeation study, ex vivo drug permeation study. So here the ex vivo drug permeation study accompanied via the porcine skin revealed through the friend diffusion cell revealed that HPMC-K15 and ERL100 at the ratio of 3:1 natural penetration answer D-limonene at 7.5% give the desired flux and improve the penetration of the drug. The results of the skin irritation test was revealed that drug formulation is free from any type of the skin irritation study like erythema and edema. The objectives of the research was to that the low viability issue due to low aqueous solubility will be ruled out through the transdermal drug delivery system. The most common side effects of NSAIDs is the GI bleeding and GI irritation will be ruled out through the transdermal drug delivery. Being a novel drug delivery system, it is capable of the avoidance of the first pass metabolism. So provide the drug at the steady state or zero order release. The most common drawback of the conventional dosage form in the system can be minimized by using the transdermal drug delivery system approach, and hence patient compliance may be increased by less frequent dosing. Materials and method. Here we have prepared the transdermal film by the solvent evaporation method by pouring the polymeric solution in mercury containing petri plate. The different composition of matrix for forming polymer ratio of HPMC-K15 and ERL100 was dissolved in dichloromethane, methanol 1:1 solvent and allowed to swell for the 2 hours. The separated formed drug solution 15.8% based on the total polymer weight in ethylene was mixed homogeneously to matrix polymeric solution with constant stirring. Non-quantity of the dibutyl phthalate as plasticizer and prefix quantity of the D-limonene penetration answer was added drop-wise to the polymeric solution. The resultant drug polymer matrix solution was discharged to a petri plate using fabricated glass ring of 10 centimeter square, and inverted funnel was partitioned for covering the petri plate for controlling the rate of the solvent evaporation maintained for the 48 hours at room temperature to get that desired dried film, and then cut into the suitable size at 3.14 centimeter square. Here table shows the first table is showing the independent variables and their labels. Table second is showing the independent variable and dependent variables results. The table third is showing the physicochemical characterization of the drug. Here, these are the contour plots regarding the tensile strength for our three dependent variables: tensile strength, steady state flux, and the cumulated amount of drug permeated in 24 hours. Here is the graph showing the ex vivo drug permeation study. Now go to the conclusion. The current research determines that the fluorbuprofen transdermal film can be produced and optimized using the different concentration of polymeric excipient HPMC-K15 and ERL100 ratio, a plasticizer dibutyl phthalate, and penetration answer D-limonene by box-Benton statistical design. Based on the outcomes of this research, it can be concluded that the FBPT7 film out of the 17 runs was able to accomplish a well-controlled release and ineffective skin permeation of the drug in the incidence of permeation enhancer for long periods of the time. The preparation constituent have a direct effect on the produced performance. Polymer matrix enhances the elasticity and sustained drug release. Plasticizer enhances the tensile strength, and permeation enhancer increases the drug flux. However, more in vivo pharmacokinetic experiment may be carried out in order to demonstrate the relevance of the effect of in vivo pharmacodynamics study of the in vitro penetration and to access the therapeutic level of the drug. So here are the references for my research work related to my research work. Thank you. Thank you very much.
Thank you, Mr. Ram, for your presentation. So next we'll be moving on to our next presenter. So meanwhile, I would like to inform that the participants can answer to the questions that has been written in the chat box, and you may exchange your email addresses for further research related projects in the future. So
okay, madam. Thank you.
Once again.
Thank you.
So,
thank you very much.
Next, we'll be moving on to our next presenter. Next, I would like to welcome Mr. Rajendra Chauhan from Jivaji University, India, who will be discussing on the formulation and evaluation of ripaglinide transdermal patch using natural polymers. Share your screen and begin your presentation. Mr. Rajendra, am I audible to you? Yeah, please begin your presentation.
Hello. Hello.
Yes, you're audible. Kindly begin your presentation.
Hello.
You are audible. You try to present. Is there any problem from your side?
Mr. Rajendra, we will be moving on to our next presenter. Meanwhile, you can resolve your issue, technical errors. So next, I would like to welcome Mr. Wasim Akram from Jivaji University, who will be discussing about the development and characterization of biopolymers based multi-particulate system potential for targeted drug delivery.
Am I audible, ma'am?
Yes, you're audible. Please begin.
A very good afternoon. I am Wasim Akram, here to present my research work, designing and optimization of biopolymer-based nanosystem, basically potential carrier for control and site-specific delivery. Our current approach is to target the colon with minimum loss of active therapeutic and less toxic effect in our GI tract. So here we developed a nanoparticle decorated with a mesalamine. Basically, we go for a disease that is target a disease that is inflammatory bowel disease. For this, we are using two natural or a biopolymer, that is inulin or a chitosan. Inulin is mainly used also used as a prebiotics, and prebiotics have a great role in the inflammatory bowel disease, because these may enhance the growth of the probiotics that are very good beneficial bacteria for our gastrointestinal disorder. So in our research work, we are using chitosan also, and chitosan we are using, the purpose of using a chitosan is that we used chitosan for to increase the transit time, because the chitosan may possess a very good property, that is a mucoadhesion. That's why we are using chitosan in our formulation, and after that we are going for after developing literature method and literature study and methodology in we are using inotropic gelation method for the development of the nanoparticles, in which a strong polylactide complex will be formed between the carboxylic group of the carboxymethylenolin and the amino group of the chitosan. These form a strong polylactide complex and form a matrix system for the nanoparticulate system. And after that, we are using design expert box Penkan factorial design for the optimization of our formulation. After that, we are going for various characterization parameters like particle size, zeta potential, polydispersible index, FPIS studies for the compatibility studies, and also mucoadhesion studies. These are our three independent variables. That is concentration of drug and concentration of chitosan and concentration of CMI, that is carboxymethylenolin, in different level, that is mid, low, and high. After with using box Penkan design, we formulate 15 formulations. This table shows that the 15 formulation was prepared using box Penkan design. After that, the response variable, that is particle size and interpret efficiency of the formulation F2 was found around 160 nanometer diameter, and the interpret efficiency was found maximum up to 88%. And we are using this graph particle size. The optimized particle diameter was found in the range of was found around 170 nanometer, and the 10 micrograph shows uniform diameter size, and the zeta potential and the polydispersibility index was also found in the range that shows this that our system is very stable and having a uniform particle distribution. And the counter plots, these may also shows that the particle size may increases, as well as the increases the polymer concentration. And the next counter plot shows that the entrapment efficiency was also increased with increasing concentration of chitosan. And the mucoadhesion studies was indicated by the green graph, which shows the very good mucoadhesion with the colonic mucosa by using ex vivo mucoadhesion studies. And now we are moving for our conclusion. We developed a nanoparticulate system for inotropic gelation method. That's biocompatible, biodegradable, non-toxic polymer chitosan and has been synthesized and characterized or present. No stabilizing or cross-linking agent were required for the method of synthesis. Reliable adaptation and easy, precise verification of final product make this nanocarrier very attractive for colonic disorder. In the range of 180 to 120 to 180 nanometer, the obtained nanoparticles show a very uniform diameter and were resistant to hydrolytic degradation, because the inulin may only degraded with the inulase enzyme that was found in the colonic mucosa and protect our formulation from the upper gastrointestinal tract. That was the most important criteria for any targeted or for any colonic drug delivery system. So in our system, we found that the drug release in pH 1.2 or gastric mucosa is only less than 5%, and the pH 7.4, that is similar to colonic mucosa, the release was found more than 80%. And so we can conclude that we have prepared effective develop biopolymer based multiparticulate system or a nano system for effective management of colonic disorder. Therefore, we propose a nanopharmaceutical would have a high potential to being a bench to bedside research. Thank you. And these are the references that was used.
Yes, thank you, Mr. Wasim Akram. You may stop sharing your screen. We are coming back to Mr. Rajendra Chauhan. Mr. Rajendra Chauhan, if your presentation is ready and if it is possible for you to share, you may share your screen now. Mr. Rajendra, are you able to hear us?
Is not there.
Mr. Rajendra, can you please start your presentation? Dr. Subhash, sir,
Yeah.
as we are running short of time,
We can go for.
moving on to the track 3, that is drug targeting and drug formulation.
Yeah, but we have a break, no? Lunch break?
We can skip that if participants who are already joined, we can move on quickly.
We have in forum, there is a keynote forum also in 2:15.
Sorry, sir?
By 2:15, there is a keynote.
Yes, yes. So before the
And
keynote,
before that, there is some lunch break. And then 2:45 should start the presentation.
Yes, sir. Before the keynote, maybe we can take 2 presentations.
One presentation we can opt to because we have to go for a little fresh.
Okay, fine, sir. So we will just move on to the track 3, that is drug targeting and drug formulation. So our first presenter will be Isabel Ann Ocampo from University of Santo Thomas Graduate School from Philippines. Ms. Isabel will be discussing about the evaluation of anti-inflammatory activity of sulfated polysaccharide from Sargassum polysystem, C. agarth, on carrageen-induced paw edema in rats. Ms. Isabel?
Hi, good afternoon.
Good afternoon.
Share my screen first. Is my slide visible?
Yes, yes, it's visible.
Okay. So let me begin my presentation by saying thank you for this opportunity to present our study entitled Evaluation of Anti-Inflammatory Activity of Sulfated Polysaccharide from Sargassum Polysystem C. agarth on carrageenan-induced paw edema in rats. Inflammation is a protective defense of the body. It involves a series of vascular and cellular events that allow tissues to respond to injury, infections, and hypersensitivity reactions. However, excess inflammation is known to increase the progression of certain diseases, including cancer, cardiovascular diseases, respiratory diseases, diabetes, and Alzheimer's disease. These are some of the leading causes of death globally, according to the World Health Organization in 2019. More recently, it has been proven that pre-heightened inflammatory levels for patients who are susceptible to COVID-19 can make them more prone to uncontrolled inflammation or cytokine storm when they are infected by the virus. And ultimately, this can cause a more severe COVID-19 case. Inflammation is commonly treated with NSAIDs or non-steroidal anti-inflammatory drugs, which act by inhibiting the cyclooxygenase enzymes or COX-1 and 2, thereby inhibiting the synthesis of prostaglandins and thereby preventing inflammation. NSAIDs are widely used worldwide by over 30 million people every day. However, severe side effects are associated with the prolonged use and high doses of these drugs, including gastrointestinal side effects that can occur to 10 to 60% of patients, and cardiovascular side effects, particularly to COX-2 inhibitors. In fact, the use of these drugs can increase the risk of major vascular events by a third. For this reason, interest has been redirected to developing alternative treatments from natural resources with better safety profile. Thus, this study aims to utilize Sargassum polysystem, which is an edible brown seaweed that grows abundantly in the coasts and shallow ocean waters in the Philippines. Other than being a food source and a contributor in the biodiversity of ecosystems, recent studies show the importance of marine environment as an untapped source of bioactive compounds. Particularly, different species of the genus Sargassum have been studied for their biological activities, which are usually associated with their sulfated polysaccharide contents. Ultimately, the general objective of this study is to evaluate the anti-inflammatory activity of sulfated polysaccharide extracted from Sargassum polysystem on carrageenan-induced paw edema in rats. To achieve this, the following methods were performed. Beginning with the preparation of the extract using hot water extraction, followed by chemical chemical analysis using qualitative and quantitative techniques, and in vivo tests, which include acute oral toxicity test and anti-inflammatory test. Again, to evaluate the anti-inflammatory activity of the sample, carrageenan-induced rat poedema assay was used. This allows for the quantification of acute and local inflammatory response by increase in pus size. So this begins after a 7-day acclimation of test animals under standard conditions. On day 8, test substances were administered once orally with the test substances. The volume of the right hind paw was measured using a plethysmometer before the injection of carrageenan and 1 hour after. Measurement was repeated hourly until the 6th hour. Results obtained are as follows. This slide shows the dehydrated seaweed material as well as the crude drug, so that is prepared after air drying and grinding prior to extraction, as well as the sulfated the Sargassum polycystum sulfated polysaccharide. So the percent yield after extraction was 4.17%, which is comparable to other studies. For the preliminary screening of the sample, IR spectra analysis was done, and based on the IR spectra, peaks corresponding to the sugar backbone of polysaccharides and sulfate esters were observed. A peak corresponding to the methyl group of the fucose monosaccharide was also observed, and with this result, it is suggestive that the sample is indeed a fucose-containing sulfated polysaccharide. To further investigate the chemical composition of the sample, chemical quantification of sulfate, carbohydrate, uronic acid, and protein was performed. The results obtained further confirmed that the sample is a sulfated polysaccharide. Recent studies show that sulfated polysaccharide, again, that are extracted from marine seaweeds and other marine products exhibit different pharmacological activities. For acute oral toxicity, limit test according to the OECD guidelines was performed, for there is evidence that the substance is safe as it is used as a food source. After 14 days, there was no treatment-related clinical signs of toxicity and all test animals survived. So we deemed that the sample is safe for use. Results of the anti-inflammatory test are as follows. This graph shows the change in paw volume from the 1st hour up to the 6th hour of observation. The percent edema rate was calculated with respect to the baseline measurements as control. This suggests that all doses of sulfated polysaccharide from Sargassum polycystum was able to decrease the edema rate in a dose-dependent manner and without significant difference to positive control, which was diclofenac sodium, during the 1st up to the 6th hour of observation. Also, since the decrease in edema rate was observed starting from the 3rd hour, which marks the 2nd phase of inflammation, this could be indicative that the mechanism of action is similar to that of diclofenac, which inhibits prostaglandin secretion also, which is involved in the 2nd phase of carrageenan-induced inflammation and edema formation. In conclusion, and based on the gathered results, the sulfated polysaccharide from Sargassum polycystum C. agar has potential as an anti-inflammatory agent. Lastly, the researcher has the following recommendations. And I would like to acknowledge my advisor, Dr. Mary Jo-Ann Corpus, for her support in the accomplishment of this study, as well as the following institutions. Thank you very much for your kind attention.
Thank you for your nice presentation.
Thank you, Isabel, for your presentation.
Thank you very
So
much.
with this, we'll be moving on to our lunch break. We'll be taking a short, quick lunch break of 15 minutes. I request all the participants to be back within 15 minutes. So soon after the lunch break, we'll be moving on to our keynote presentation by Dr. Animesh Acharjee. So please be back within 15 minutes. Thank you so much.
You there?
Hello everyone, welcome back to the 8th World Conference on Pharmaceutical Science and Drug Manufacturing. So we are back after our lunch break. So next we'll be moving on to our keynote forum by Dr. Animesh Acharjee, who is a senior scientist from University of Brigham and NHS from United Kingdom. Dr. Animesh, am I audible?
Can
Hello,
you hear
sir.
me?
Good afternoon. How are you?
I'm good. I'm good. Thank you very much. I will share my screen.
Sure.
Okay. I hope you can see my screen.
Yes, sir.
Okay, great. So first of all, thank you very much for inviting me and inviting me to talk about the work I'm doing, and mostly in multiple different frameworks. So my name is Animesh, and I've been working with University of Birmingham in England and also NHS. I have a couple of other roles. So I'm a visiting scientist with the University of Cambridge. I've been working in Cambridge for 3.5 years. And then I moved in Birmingham. I recently set up my own company called Omics Marker Limited, where we are doing quite a lot of diagnostics and prognostic research, research-based organization. So, so that's why you can see my title. My title is the Big Data-Driven Medicine, a first step towards the diagnostics. So I would like to talk about how we can discover the markers, non-invasive markers for multiple different samples. It may be the urine samples and or it may be the blood samples. So let's see what is the in agenda. So I plan a couple of things, and I'm very much cautious about the time. If time doesn't allow me, so I'll cut it down a bit. But what I'm planning to do is introducing the big data. So what do you mean by nowadays the big data? The second point is identification of the marker, how you do the marker identification in different settings. And what I did from the multiple studies, I have taken into account different case studies. So my case studies are varied from the gastroenterology, trauma, and multiple different diseases. So very general, and I have taken intentionally those case studies just to show the diversity of the approach. And then finally, the interdisciplinary research. I want to emphasize how interdisciplinary research can be possible, and it's very much required in nowadays in terms of the pharmaceutical science, in terms of the molecular biology, computer science. Everything has to be integrated together to accomplish a common goal, like, for example, disease diagnosis and all. So let me talk about the big data. What do you mean by big data actually in that case? There are certain properties of the big data. So number one properties of the big data is volume, okay? So you will see and the massive amount of volume, and there are, I'm coming back, I'll be giving some examples what I mean by volume. Velocity, for example, the way the data is coming into picture in your table, the velocity. Veracity means different types of data. Variety, types of multiple different variety, and the value. They might have different sort of value, and those values may be multifactorial in a way. Now, the people who are already in the bioinformatics, or people are working on the high throughput data, they have already seen the big data in approximately 2001-2000 time frame, when the first human genome sequence has been established. So you can see this is the Nature publication, came out in 2000 after entire human genome was sequenced. And then not only in the Nature, but in the Science, they have published, you can see in the cover page, just to decode all the sequences from human body. Now, as a consequence, what happened is many different sequences were available, and people face many difficulties. People face difficulties how to store the data, how to retrieve the data, how to analyze the data. Okay, so it's not that you might have heard many of the terminologies nowadays called data science or big data analytics. So in current scenario, it has more pronounced because of the internet booming, because of the social networking, and because of many other cases. But the people who are in the life sciences domain, they have seen those sort of data many years ago. So that is my point. So as a consequence, what happened, you need a databases. So people developed different databases. So you can think about the protein database and many other databases. So that's one of the sources of big data. Now, if you look at the explosion, and you can see in the cartoon in the left hand side, in the x-axis is the time frame. This is the old data. This is not data is until 2012. So 2012 until 2012. However, you can see the terabyte in the y-axis, how data is being generated across. And then it has keep going from the kilobyte, megabyte, gigabyte, and zettabyte, and the many other dimensions. And as I was telling in the previous slide, that every 6 seconds, for example, in the social networking like Twitter or Facebook or many other real time, for example, if you're talking about the satellite communication, you will see every second they are generating many different data sets, and so on and so forth. And then, for example, in the agriculture, you can see, so he is my collaborator in Colombia in South America. And what they're doing automated plant phenotyping. So they are using the drone. So from the drone, they are taking pictures, and then pictures has been analyzed in the data. So they minimize the manual labor. Okay, so they minimize the manual labor or costing of the labor. And nonetheless, in the medical domain, we see in many different, like MRI, CT scan, X-rays, and those sort of data intensive, so that we can make a better decision for diagnosis of the patients. Okay, now if you look at the literature, what people are saying in the medical domain. So if you look at, for example, I have just picked up a couple of papers. These are quite recent, last year. Some of them are from last year. You can see many papers, or many of the in journals like Nature Medicine or eLife or even Nature. Actually, I mean, it's they've been talking about how you can how the data can be used, how data can help a clinician, a medic, to make a proper decision. So these are all about, so there are challenges, but there are also some sort of, the helping attitude for the clinical domain as well. Now let's talk about a little bit on the analytics. I mean, what are the many topics on the analytics? I mean, if you talk about the analytics, you can see they are coming from multiple different domains. You can see here knowledge representation, image analysis, natural language processing, artificial intelligence, machine learning, statistics, and mathematics. This is the analytics part. So either these are intersection between statistics, computer science, and the mathematics, but also big component is on the clinical side. Okay, so this is a kind of a tool, and these tools have been applied in multiple clinical domains. So this is a brief introduction, and then I am moving to my research group. So here I've been affiliated with multiple different groups. Here you can see University Hospital in NHS, National Health Service in England. This is centralized, so I'm placed in the Birmingham. From London, it is an approximately 1 hour train journey. Alan Turing Institutes. Alan Turing Institutes majority of them are working on mainly on the novel algorithm development and computational part of the Center for Computational Biology here in the department, and recently developed HDR UK. So we call it Health Data Research UK. So we have in our group the clinical domain people and also non-clinical domain people. So we have divided in our group in multiple different aspects, and one of the things is comparative phenomics, large-scale representation, actionable data analytics, network medicine, translational medicine, and many different other subdomains. So I will touch upon briefly a couple of them, but not all of them because of the time limitation. So we call it the phenotype-based patient profile. So what does it mean? So these are the phenotypes. So for example, you can see here, if you look at the lower part, you can see the genome, transcriptome, microbiome, metabolome. So these are mostly the molecular trait, and in the hierarchy. So in the hierarchy, upper level, these are the different phenotypes. It's either you do MRI, you do the clinical letters, retina scans, and all these things. So we can definitely define as a two parts. One is the omics analytics. So omics in the sense high throughput data. So omics, it may be transcriptomics, it may be proteomics, which is coming from on the protein data. It may be metabolomics, LCMS or GCMS. It also can be other like SNPs or molecular markers. So these are the molecular trait, whereas the phenotypic based analytics is driven to the clinical thing. Now, if you understand, if you go back to the basics, I mean, what is happening? So if you think about the diseases and the definition of the diseases, we can represent the disease and thing in a very systematic way. So for example, we are getting some input, stimulus or perturbation. So those stimulus or perturbation can be in cellular level, those perturbations can be in the tissue level or in the hierarchical level, okay? And then something else is happening, some sort of communication, and then you get output, okay? And then the body want to maintain this output for, doesn't matter what the changes you are getting, and then you want to make equilibrium point condition. Then what they do, they give a response or in the input, and then body try to stabilize. So when I say body, that means either it may be the cellular level, it might be in the tissue level, or it might be in the organ level, okay? So for example, if you represent the input as a genotype or X and phenotype as Y, this is always a challenge how you link genotype to phenotype, yeah? So people showed many different ways. People showed literature, they do a QTL mapping, they do a GWAS, they do many different ways to link them, yeah? So eventually, this is not a very easy task to do. So because it is not that the genotype to phenotype, but also there are many other layers of molecular organization, okay? So different genes, you need to cross multiple different metabolite, protein-protein interactions, and then it is a manifestations of your phenotype, yeah? So, so the idea is it's a very complex, and you need a lot of assumptions because you wanted to solve some sort of the equations in this area. But one of the important thing is if you control one parameter, for example, if you're talking about some molecular communication or cellular communication, like for example, PPAR alpha gamma signaling, for example. And then if you see, as because they are master regulator, they are multifactorial functions in our body. So if you control one, it's possible that it is manifested in other disease. So there is a lot of change, like cancer. If you think this is genetically, it can be genetically, it can be environmental, genetics, and all other things. So before you control, you need to identify which one to control and how to control. Okay. So if you say these are the genes are important, but then which genes and how do you target those genes? So that's the key. Okay. Now, and that is the main intention here. What the marker, definition of the marker, can you identify particular marker which can be targeted? And can those marker can be useful for a diagnostics purpose? Okay. So non-invasive. So for example, you take the blood and then you say because of concentration of the particular metabolite, and you can say like if you were suffering from diabetes, so you look at the HB1C. So if your HB1C is higher, you tend to get more diabetic. Okay, so this sort of thing. So but then what are the different properties of the biomarker? So you cannot just call it as a biomarker if it is just indicative. Okay, so it has to be some sort of a properties of the biomarkers. Now, one of the thing is it has to be measurable. Okay, now measurable, what does it mean? If it is a protein marker or if it is a gene expression, for example, you should measure it. Okay, so you should be able to quantify the value in terms of the control or healthy patients versus non-healthy patients. Yeah. It has to be indicative. It should say something about your disease state. So either it is early stage, or it is late stage, or it is middle stage. Like in cancer, stage 1, stage 2, stage 3. It should tell you where it is and what is happening. Predictive. So what does it mean? Predictive means it is not only for example, if I do a trial and the clinical trial, and I am including only the Asian population. So Asian population, including India, Pakistan, Bangladesh, Sri Lanka, and all these countries, South Asian countries. But then it has been seen the marker is only affecting in the Asian population, not affecting in the African population. Okay, so that is not useful. Okay, you wanted to make a diagnostics marker which can be more or less can be so powerful that it can be go across geographically. Okay, robust. So what does robust means? Robust means that it has to be reproducible all the time. Okay, and it is robust in the nature, and then you claim this is the diagnostic marker. Okay. Now, in the literature, there are many different names, so people get confused. What is the definition of the diagnostics marker? Okay, so sometimes people say translational biomarker. Okay, drug response. Okay, genomic medicine, precision medicine, personalized medicine, population-based medicine, or stratification predictive medicine. They sometimes use terminologies in a similar way. Okay. I wanted to tell you this one because when you are looking at the literature, please be careful what you are reading because maybe they are meaning the same thing. Okay. So this is a brief introduction of the biomarker. Now, what I'm going to do, I'm going to do some sort of a real data. I'm going to do some sort of a real data where I've been working a couple of years, and we have been able to produce some of the papers and also some of the grant based on those. So this is on the trauma. Okay. Now, before I'm going to the trauma, I am giving you some background information. So so we are in Birmingham, in the Europe. We are largest clinical trial center. We have developed largest clinical trial center entire Europe. Okay, and we are also center for Defense Academy of the England. So what does it mean? Defense Academy. It's not as such academic, but it is more kind of when people get blasted. So from the England, people soldiers are going to fight in Afghanistan and other Iraq and other countries. They get injured, they come to the England, and if you come to the England after that, they send to the Birmingham Center. Okay, so what do we do? So these are trauma. Okay, so this trauma has caused by the blasting, shooting, and all these kind of things. So these are the soldiers' trauma. There are another type of trauma is if you're falling over from a high building or sleep. This is or road accident. This is also another sort of trauma. Okay. So we have been studying the multi-organ dysfunction. So multi-organ dysfunction means that once one of your organ is not working, and it processes multiple different organs sequentially. Okay? So it's kind of a sequential or organ failure or sequential organ dysfunction. So for example, your liver is not working, your pancreas is not working, and then failure. There is no treatment for these except the transplantation. So we wanted to identify based on the profiling, metabolic profiling or immune profiling, can we identify some of the soldiers or some of the patients who are going in this trajectory. Okay, so the trajectory means if they are following the multi-organ function dysfunction pathology, or they are not. Is there any way to know it? Okay, so what we did a cytokine profiling. Cytokine is inflammatory immune inflammation, and we measured the peripheral blood. We have shown earlier in the PLOS medicine. This is the Hazeldine. So Hazeldine is my colleague. He showed earlier in 2017 in PLOS medicine. He published and he showed how to take early blood samples. So you got a trauma in the road accident, within 1 hour he can take the blood. He developed a technology for that. Okay, and then as you can see, this is, I'm talking about less than 1 hour sampling. We have done some sampling 4 to 12 hour sampling. We have done 48 to 72 hour sampling. So longitudinal trial design, okay, and then we have done some sort of then used some sort of a AI, artificial intelligence related methodology, and then we identified some of the marker which can be important for the patients. Okay, so what we did, I'm showing you, this is the result section. Actually, I'm showing you here is you can see there are the probability values. So for example, I'm showing that each of the time point we identified some of the markers. For example, first time point we found monocytes. Those are important. In the second time point we did not find anything. In the third time point we found a CD63 is important. And these are more kind of a statistical analysis where we have shown the data makes sense, and we are now doing the trial targeting only those monocytes in the larger population. So selected markers is linked to neutrophil, and we found the neutrophil is one of the important function in the early stage trauma. So if you're getting trauma, if you can control, if you know which of the neutrophils are behaving, so that we can make the patient management in a better way. So we published this paper 2 years back, and this is the Nature scientific data. So we are able to publish and say that these are the maybe important for the trauma patients. If you want to know, please go through this article and you can get more information. Okay, so now I'm moving to the second case study where a fusion of multi-omics data. Okay, so what does it mean? It means that you have developed a cohort. You have a cohort of diabetes, for example, and you have a 50 control and 50 diabetic patients. And you have generated high throughput RNA sequencing data. You have developed metabolomics data, transcriptomics data, and multiple different data sets. Now you are asking, okay, how do I make sense of this data so that I can make a translational lead, so that I can identify the marker which can be useful for my further study? So if you look at the literature, what is happening, these are the overview of the literature. These are very high impact journals. And then if you look, for example, Nature Immunology, they use one of the example on the multi-omics data with the deep learning artificial intelligence tool. Nature Communication, they have identified the gut microbiota with some specific design aspects. So each of the paper here is representing some sort of an integration. And that's the motivation. That's motivated us. Okay, can we integrate multi-omics? Okay, but before that we need the data. Okay. So this is another example where I'm working with the hospital here with the gastroenterology people, the gastroenterology consultants, and one of the diseases is called primary sclerosing cholangitis, and people often confused with primary sclerosing cholangitis versus ulcerative colitis. Okay, so that means, what do you mean by confused? Confused means if you are suffering from PSC, you go to the doctor, and there will be misdiagnosis saying you are suffering from ulcerative colitis, okay? Because, and we are wanting to prove the ulcerative colitis is really different from PSC. As they are different disease, you cannot treat from the same drug. That is the point, okay? So we developed here multiple different different technologies, multiple different data sets. So you can see we have a healthy control, number of samples are 10, PSC samples 10, ulcerative colitis patients are 10. Number of patients are less. Nonetheless, we have collected the microbiome data, because you might be aware, microbiome design or analysis now boom in the field of diagnosis, because people can use microbiome to treat the patients, okay? Immunology, so we have done 3 different technologies, and we have generated data like transcriptomics data, cytokine data, microbiome data. We have done some sort of a machine learning and biomarker, and then we try to integrate everything, try to understand what is happening in the molecular level, what is exactly happening, what is different ulcerative colitis patients versus the control patients. Okay? So this is a general omics fusion I have developed when I was doing my postdoc in and PhD in Netherlands and some part in Cambridge. Here it doesn't matter how many matrices, how many data you do experiment, but I have found a generalization of the matrices fusion still can make out of the biological sense. So here we have given a matrix, I reduce the dimension, I integrate, and then I link with the pathways and biological, and then we follow up experiments to validate those results. Okay. So this is just an example for the transcriptomics data. Here, I try to optimize the number of features because when we do a transcriptomics or RNA sequencing, initial experiments gives you around 40,000 genes, and 40,000 genes are, you don't know which one to target. You wanted to see, and then it's kind of an optimization function. And based on optimization function, for example, I have selected around 10 genes because it's showing more predictability in terms of the sensitivity and specificity. Okay. And then here it looks like a very complicated, but it is not. What I have done, you can see first of all 3 different colors. 3 different colors means 3 different types of data. You can see the connections either green or maybe the red. The green ones are positive correlation, the red ones are negative correlation. You also see some of the clusters. Okay, so clusters are, these are the G, starting from G, these are the genes which are interesting because we found those genes are always together, and there must be some reason why they're together. And then when we did a pathway-based analysis, we found those genes are involved in the bile acid regulation. So we found that bile acid regulation is one of the mechanistic way when the PSC patients are suffering from, when the patients are suffering from PSC, there is something to do with the bile acid regulation. Okay, so we reported this this paper. We published this paper last year, and it's a quite high impact paper in Journal of Crohn and Colitis. And now we also got a grant. We are doing an intervention with a drug. So we are treating the PSC patient with particular drug. We are interested where if we apply the drug, what are the changes is been going in terms of the microbiome profiling, in terms of the gene profiling. Are those changing, are similar? Okay, so that's the thing we are doing. So that's my second one, and it's connected with. So this is case study 3. There is a typo. It's not 2. It should be 3. But as you can see, the microbiome research is coming into boom. So this is the paper published in 2 weeks. So 26th March it published. And I propose, if you're interested, this published in Science. So you know there's Nature and Science, how much impact. If you get one paper in Science, so that is your career is done actually. I mean, you can get anywhere in the world. So this is a review paper from one of the famous lab from California, from Rob Knight lab. Rob Knight is actively working on the microbiome. He developed many of the tools for microbiome research. Now, what he's talking about in this review, that the cancer is many of the cancer cells are driven by the microbiome, and we can target the microbiome, which are very specific to the cancer. For example, if you're talking about the colorectal cancer or pancreatic cancer, some of the microbiomes create a tumor microenvironment, and because of the tumor microenvironment, this is very unique. So we need to target those microbiota, and probably then we can understand better the cancer, because the cancer still is in the process. We don't have any like cancer has not cured. So there might be one of the reasons microbiomes are playing a role in this process. Also, there are some other papers like microbiome-based diagnostics, radio application laboratory medicine. There are very nice clinical chemistry. They publish very high impact papers. Microbiome-based companion diagnostic, no longer science or fiction. So they're saying, okay, people are doing it like we are doing here in FMT, fecal microbiome transplant. What does it mean? So we are taking stool samples from the healthy control. We culture the cell, and then the person who is suffering from IBD means either ulcerative colitis or Crohn's disease, we're transferring those microbiome to their body and then see what are the changes. So we in the Europe, we got a very license to do FMT. So this data is coming into picture and will be very high impact paper as well from those data sets. I'm quickly going here. So this is the pipeline we have developed in our group to handle the microbiome diagnostics. So it is kind of a neurofuzzy or artificial intelligence based pipeline. This paper is under review now, and we hope to get this out soon. But what I'm saying, it's a kind of a combination of the biology and combination of science. Like a couple of case study on the COVID-19. You know the COVID, nothing, everybody is working on COVID-19. Any decent papers, you can see like Lancet or Nature paper, you can see people are talking about how AI-based approaches can be using for COVID-19, okay? So what we are doing in the Birmingham is we are collaborating with the Global Search Unit. So it's called NIHR. NIHR is National Institute of Health Research. And we have a global surgery unit, and global surgery, just to give you, this global surgery is spreaded across six different domains across the world. In India, we have in Punjab, in Ludhiana, we have a center. Also Rwanda in Africa, one of the center, Nigeria, Ghana, and in Mexico. So we have the centers. We are getting the patients, those who's been done in the surgery. Okay. So this is a web link. If you're interested, you can know more. What we did here, so patients are coming in our hospitals, and we collected many of the data sets. Okay, we have collected like if the patient is smoking, what is their sex, what is their urgency, what is if how much anesthesia given, what is the age group, and all the clinical data we have collected, and then we developed an app. So if you look at the app and you give the data, and then it will show you what is happening there. And then this is the pipeline. You can see there are a lot of modeling has been done in this case, and nonetheless, what we did in summary is you give the data will show some analysis, and it will give you some sort of an indication why this patient what is the lacking in this patient. Is it due to age group, or is it due to infection if they have other complications earlier? And we have done quite a lot of these kind of things. So this is kind of a result. So we have shown how like age, ASA2, and respiratory, if you're suffering from respiratory earlier, and we know because if it's a SARS-CoV is a respiratory disease mainly. So if you are already suffering from some sort of problem, then your infection rate goes high in this case. So this is kind of a shiny app we have developed, and the paper is under review in the moment. Now this is a couple of slides, and I would like to go through very quickly the need for interdisciplinary research. Okay. Now this is one of the slides where you can see I've been talking to many different I'm sorry, I think something happened and it goes off. But what I'm trying to say, I've been working with many different domain, and in our group, what we did is kind of making a balance between multiple different domain expertise. So here you can see the colorectal cancer, sarcopenia, burn injury, trauma, IBD, PSC, FMT, and all these kind of things. So we are tightly working with the multiple different domains, and we are sharing our complementary expertise with them. Also in multiple other countries, and I've been collaborating with Harvard Medical School for some time. It's already more 4 to 5 years time, and Italy for polyphenols, University of Canada in Manitoba in diabetes, type 1 and type 2 diabetes, University of Amsterdam Academic Medical Center on the colon cancer, Cambridge, liver, obesity, nutrition, NFLD, and all this kind of things. So what we do, we not only just talking with them, we produce the papers, we produce the grant, and all these kind of things. So that is very fascinating. And one of the fascinating part is people are coming from multiple domains. By the way, my educational degree is very diverse. I am electrical engineer by my profession. I did undergrad, but I did master's in biology. So and then bioinformatics. After that, I did my PhD in statistical machine. I understand a little bit of biology, a little bit of other things as well. So I strongly believe the interdisciplinary research. So these are the key points here. So big data, feature selection, biomarker, and then it goes to the diagnostic research, interdisciplinary team, collaboration matters, really matters. Yeah. And then finally, these are my collaborators, and I am reporting to my director. So he's the director of SDRUK. I'm reporting to the director. I have a group here. I'm leading a couple of PhD students and postdoctoral fellows here, and they are across multiple different countries. And finally, I'll be very happy to take any questions. And yeah, thank you very much for your attention.
Thank you, Dr. Anmesh, for your presentation, for your keynote presentation. That was amazing. And it's an absolute pleasure to have you here. Presenters, if you have any queries, you may write down to us in the chat box, or you may write down to Dr. Anmesh directly to his email address. So thank you so much once again for joining us. We'll be moving on to our sessions, technical sessions. Would you like to share anything with us, sir?
No, thank you very much for giving me the opportunity. But if anyone can contact me for anything, I mean, in future, if they find anything I can help or whatever, university wise or the students, anything, I'm pretty open to any of the stuff. Yeah. Thank you.
Thank you so much for your help, and thank you once again for joining us, sir. So next, I would like to welcome Mr. Hemant Maurya.
Yes, ma'am.
From Jivaji University from India, who will be discussing about the formulate and evaluate of sustained release bilayer tablets of propanol hydrochloride.
Good afternoon, good afternoon, all of you. I am Hemant Maurya. Hello. Hello.
Yeah, you're audible. You can
yes, you're audible.
Yes, sir. Yes, sir. Yes, sir. Good afternoon. Good afternoon, all of you. I am Hemant Maurya, and my topic is formulate and evaluate of sustained release bilayer tablet of propanol hydrochloride. So first of all, we will talk about that what is bilayer tablet. Bilayer tablet is a novel technology for the development of controlled release formulation, developing a combination of two or more active pharmaceutical ingredient is a single dose from is known as bilayer tablet. So we can see in the diagram that with the combination of immediate release layer and sustained release layer with compression, this process is carried out easily. Further, we will talk about the drug which I had taken for preparing bilayer tablet, which are propanolol hydrochloride. Propanolol hydrochloride is an antihypertensive drug which is non-selective beta-energetic blocking agent that is mostly used for treating angina pectoris and many other cardiovascular disorder. This drug has certain properties that it is highly soluble in water with 3 to 5 hours of biological half-life. The purpose for preparing propanolol hydrochloride as a bilayer tablet to get prolonged effect due to its sustained release, because it's usually done is 40 mg twice daily, which leads increased drug concentration of drug in GIT tract, which affect it severely. The main objective of study is design and evaluate of propanolol hydrochloride bilayer tablet for reduced frequency of administration and increase the efficacy of oral bioavailability of the drug. For the result, I had prepared bilayer tablet by direct compression method with with the ingredient of immediate release like this, and for the sustained release ingredient are This for the tablet of 8 formulation F1 to F8 were made, and I'm using a different polymer for immediate release like iron T314 and sustained release HPMC and ethyl cellulose. The dose in the formulation for immediate release layer was 22 mg and the maintenance dose 58 mg of propranolol hydrochloride were calculated. So now we'll discuss that total 8 formulation from F1 to F8 were prepared with different concentration of polymer, amongst which the optimized for formulation F4 shows post compression parameters are weight variation, hardness, friability, thickness, and drug content. This graph. Sorry, sir. Sorry, sir. The graph represent a cumulative percent drug release with respect to time to all the formulation, among with F4 shows better sustained release with 98.36% than other formulation. It can be concluded that the optimized formulation F4 shows a higher rate of drug release up to 12 hours as compared to other formulation due to the combined effect of HPMC due to hydrophilic effect and ethyl cellulose with hydrophobic effect to get an improved rate of drug release. This modified release of bilayer tablet gives using frequency, increase bioavailability, and provide better patient compliance. And this is the references, and that's all of you. That's all from my side. Thank you so much.
Mr. Hemant. Thank you, Hemant, for your presentation.
Thank you, ma'am. Thank you so much.
So next I invite Ms. Gomati AR from JKKM Institute of Health Sciences, College of Pharmacy, India, who will be discussing on the formulation and in vitro evaluation of colon-targeted indomethacin tablet using tamarind seed polysaccharides by 32 factorial in design. Ms. Gomati?
Yes, ma'am. I will present.
Yeah.
Is my screen is visible?
Yes, it's visible. You may begin your presentation.
Okay, thank you. So myself is Gomati here. So my work is formulation and in vitro evaluation of colon-targeted indomethacin tablet using tamarind seed polysaccharide by the 3-2 factorial design. Here I have chosen the colon drug delivery system. So it has gained a more importance, not only for delivering the drug, but also for delivering the proteins and therapeutic peptides. Here I have chosen the indomethacin drug. It is act as a anti-NSAID. It is most commonly used for the treatment of inflammation and the pain resulting from the rheumatoid arthritis, and also it has used for the anti-colorectal cancer activities through the COX independent mechanisms. And here I have chosen the natural polysaccharide. It was mostly used for the development of solid dosage form for delivering the drug to the colons. Here I have chosen the tamarind seed polysaccharide. It was combined with the ethyl cellulose. It was used for preparing the matrix tablets, and also that matrix tablets were coated with the Utrazit L100. So it can the coating integrity can withstand over the pH values of stomach and the small intestine, but can dissolve at the neutral pH at the terminal ileum. Thus, the significant plan of my study is to develop the indomethacin entry-coated matrix tablet for colon delivery by combining and time-dependent pH-sensitive approach. Here I have chosen 2-factorial design, in which the amount of TSP and ethyl cellulose ratio was taken as the independent variables, while the Cumulative drug release at the 4th hour and cumulative drug release at 12th hour were taken as dependent variables. Totally 9 formulations were made. The experiment trial was performed at all the 9 formulations. The formulation layout for the factorial design is shown in this table. And formulated tablets were evaluated for the different evaluation studies like weight variations and also the hardness test, drug uniformity, and also the disintegration time. The disintegration time was found to be in the range of 25 minutes to 44 minutes at the pH of 7.4, the colon pH medium. And in the dissolution study, at the pH 1.2, none of the formulations showed any release of drug due to the entry coatings, while some batches started releasing the drug at 6.8. The release of drug was a maximum of 90% from batch of F4 and the near colon site. Also, the kinetic studies was performed for the all the batches. The N value was found to be in the range of 0.5 to 0.6, and also the data analysis for the responses were shown in this table, and also here the graph is representing cumulative release of totally 9 formulations. And in the discussion part, the weight variation was performed for the result formulated tablet. It was shown that the weight was increased below the 3%, and also the weight gain was in the range of 3 to 7%. The disintegration test. In the disintegration test, all the tablets remain intact in the pH of 1.2, and they were shown no cracks or the fractures. So some of tablets disintegrated within 45 minutes in pH 7.4. This indicating the disintegration time.
Thank you, Ms. Gomati, for your presentation. I would like to welcome Kavita KV from of ethanol extract. Yes, Ms. Kavita, you may begin your presentation.
Yes, ma'am. Good afternoon to one and all. Myself, Kavita KV from Day1 College of Pharmacy and Research Center, Trivandrum. My poster presentation is about the topic. Yes, ma'am.
Excuse me, Kavita, can you please share your screen?
Yeah, I shared, ma'am.
Sorry, it's not visible.
Okay, I will do once again, ma'am.
Her name is not included. She is a new entry, madam.
Sir, name is
Yes,
included,
sir.
sir. It is visible, sir, madam, now?
Yeah.
No. Please share your screen. You have option below, right in the middle. Share screen.
Yes, ma'am, I done that.
Mustafa, madam, she is not included in this list, or
Okay, you may begin your presentation then.
Yes, ma'am, now it's visible.
Sorry, no. Or so shall we move on to our next presenter? Meanwhile, Kavita can see what is the problem and resolve and come back.
yeah, we can move
Okay,
on to.
1 minute. So we'll share from our side then. Kavita, is that okay?
Yes, ma'am.
Just a minute.
Okay.
We'll share it from our side. then.
Okay, ma'am.
Yes, Kavita, please give the presentation.
Yes, ma'am. Is it audible?
Yes, audible.
Okay. Good afternoon to one and all. Myself, Kavita KV, from Day1 College of Pharmacy and Research Center, Trivandrum. My poster presentation is about the topic anti-tumor activity of ethanol extract of Lepidogathis cuspidata against Ehrlich ascites carcinoma in mice. Lepidogathis genus belongs to the family Acanthaceae. It is represented by 100 species. This family is known to possess broad spectrum biological activities such as cytotoxic, antifungal, anti-inflammatory, antioxidant, and insecticidal activity, etc. The major isolated chemical constituents of this genus include sesquiterpenes, alkaloids, flavonoids, saponins, essential oils, and inorganic minerals, etc. The species of Lepidogathis cuspidata is a spiny shrub. It is found in tropical Himalayas and Western Ghats. Traditionally, the whole plant is used for the cure of inflammation, itchy infections, mouth ulcer, etc. The in vitro antioxidant and antifungal activities of these plants are also reported. Based on the traditional usage, chemical constituents, and the scientific report, the ethanol extract of Lepidogathis cuspidata was selected for the present study. Regarding the extraction, the whole plants were collected and identified by the taxonomist, and it was dried and powdered and extracted with ethanol using SOX-like apparatus. The study procedure, the studies were carried out using male Swiss albino mice weighing about 20 to 25 grams. The EAC cells were obtained from Amala Cancer Center, Thrissur, Kerala. The male Swiss albino mice was divided into 6 groups, and each group containing 12 animals. The first group were treated with a normal saline, and the second group were EAC control group. The third group, fourth group, and the fifth group were treated with different doses of ethanolic extract of Lepidogathis cuspidata, that is 100 milligram per kg, 200 milligram per kg, and 300 milligram per kg respectively. And the sixth dose were treated with the standard drug 5-fluorouracil. The total study duration was 21 days. Then after the administration of last dose, followed by 18th hour fasting, the 6 mice from each group were sacrificed for the study of antitumor activity for the estimation of hematological and biochemical parameters, and the remaining animals in each of these groups were kept to check the mean survival time and increasing lifespan of tumor-bearing host. The result and discussion. The present investigation indicates the ethanol extract of Lepidogathis cuspidata showed significant antitumor activity in EAC-bearing mice, and the results are discussed. As shown in the table, the EAC control group, the mean survival time was almost 19 days, but it is increased with drug-treated groups in a dose-dependent manner. Treatment with ethanolic extract of Lepidogathis cuspidata at different doses significantly reduced the ascites volume, packed cell volume, and viable tumor count as compared to EAC control group. Regarding the hematological parameters, the hemoglobin content and RBC content in EAC control group were significantly decreased in the normal group, and the WBC count was found to be increased. Anemia was observed in tumor-bearing mice. It is mainly due to reduction in the RBC and hemoglobin percentage. The treatment with ethanolic extract of Lepidogathis cuspidata brought back the normal hemoglobin content, RBC level, and the WBC level to the normal. The next, the level of serum marker enzymes, as it is shown in the table, SGOT, SGPT, then alkaline phosphatase. Yes, ma'am?
Yeah, you can wind up in 1 minute.
Try to conclude. It is 5 minutes only.
Okay, ma'am. Okay, sir. Okay. The conclusion was the inhibition of tumor cell growth and was observed after treatment with the different doses of ethanolic extract of Lepidogathis cuspidata, and enhanced survival time was also reported, which suggests the anti-tumor activity of this particular plant against EAC bearing mice. From the above results, it can further concluded that the presence of the important phytochemicals are responsible for its potent anti-tumor and antioxidant activities. Thank you, ma'am.
Thank you. Remove your slide.
Okay, ma'am. Okay, sir.
Thank you, Kavita, for your presentation.
Thank you, ma'am.
You may stop sharing. Yeah.
Next candidate.
Yes. Thank you. With this, we are moving to our next presenter, Munusamy Jambulingam. Mr. Munusamy from Erode College of Pharmacy, India, who will be discussing about the in silico docking analysis of Mycobacterium tuberculosis. Mr. Munusamy, can you hear us?
Yeah, yes, madam. I can hear you. Whether it is audible now, madam?
Yes, yes, audible. Can you please share your screen?
Yes, madam. Madam, whether it is clear, madam?
Yes, yes, it's visible. You may begin.
Okay, madam. Madam, myself, Munusamy Jambulingam, and working in the Department of Pharmaceutical Analysis, Erode College of Pharmacy, Erode from Tamil Nadu. And my topic today's session is in silico docking analysis of Mycobacterium tuberculosis, a potential target of enol CoA, enol ACP reductase with some transition metals ligands, what I have synthesized. So if you see the introduction, generally we know tuberculosis is a communicable diseases that may cause ill health, and it is the one of the major diseases when compared to HIV and AIDS. And this tuberculosis mainly caused by this organism Mycobacterium tuberculosis, which will be spread when the people who are sick with the TB bacteria into the air. And we know the dose regimen, that is, it requires accurate diagnosis, screening of drug resistance, and an effective direct observed therapy treatment regimen for the duration of 6 months and followed by support. So in order to develop a new drug with some transition metals, I have carried out in silico designing of the compound of interest by using ChemDraw 12.2, and all the structures has been evaluated by using Autodoc Vina. And the metal complex, what I synthesized is been characterized by mass, IR, C13 NMR, and H proton NMR. And the metal complexes has been evaluated for anti-tubercular activity. And these are my proposed structures for metal complex ligands. So the starting of the synthesis is sulfaldehyde react with an amino acid such as histidine to form a skip base along with acetic acid as a catalyst. Now the skip base which is being formed is being treated with various metal chlorides such as iron chloride, cobalt chloride, zinc chloride, and manganese chloride to obtain the proposed structure of my synthesis. So after completing the synthesis, all these all the synthesis metal complex derivatives were being screened for anti-tubercular activity against H37RV strain. And in silico design was being carried out, and all the compounds synthesized were being in agreement with the Lipin's rule of 5. Further, the enzymatic assay aided by molecular docking of H37RV strain and enol CoA was studied. And the tabular column indicate the 5 compound which I synthesized. The first compound.
Munusamy, your time is up. You may wind up.
Okay, madam. Yeah. Only the result and discussion I will complete it, madam. So after doing the molecular docking analysis through Autodoc, and it is been validated through Autodoc Vina software, And we can see the binding sites where it is going to bind with the receptors, and all the parameters has been validated through Drug Discovery Studio. And here I can promise that all the transition metals which I have synthesized have good antitubular activity, and I have done with LJ medium also, which is shown at the bottom along with the bottle. Some metal ligands completely decolorizes the H37 RV strain. So with this, I think I can stop, madam.
Yes, thank you so much for your presentation.
Thank you, madam.
Next, I invite Divya Das from JIS University.
Good afternoon, ma'am.
India.
Yeah. Good afternoon, ma'am. Am I audible?
Yes, you're audible. You may begin.
Yes, ma'am. Just previous speaker can remove that screen.
Yes.
Just a minute.
Yes, Mr. Divya Das, you may begin.
Yes. Ma'am, it is visible?
Yes, it's visible. You
Okay.
can put it on full screen, slide mode.
Yes, just a minute, ma'am. Is it okay, ma'am?
Yes, it's visible. Begin.
Okay. Yeah. Good afternoon, everyone. Today's my topic is method development and validation of methylprednisolone by LCSI MSMS in human plasma and its application in bioavailability and bioequivalence studies. So, in introduction part, the methylprednisolone is a corticosteroid. It relieves inflammation and it is used to treat the certain forms of arthritis, skin, thyroid, colitis, allergies, and asthma. So here I may prepare the bioanalytical method because in previous bioanalytical methods are having some drawbacks. So we tried to overcome the problems. Next, this quantification method is reliable. The extraction procedure was very simple and cost effective. So to the determinant of therapeutic efficacy of the drug, this method is very useful and accurate, and it is applicable in BAB studies with prior permission from ethics committee. So now the experimental part. So here I prepare a simple, sensitive, and rapid high performance LC electron spray ionization MSMS method and validated as per the US FDA and EMA guidelines. And here I use propanolol as a internal standard. So the parent and the product and transaction of for the methylprednisolone is given, and the IS is already propanolol, and were monitored on triple quadruple mass spectrometer, operating in the multiple reaction monitoring MRM, and it is in its positive mode. The linearity range was in between 12.50 nanogram per ml to 800 nanogram per ml. And already I mentioned that the total chromatographic time is taken 7 minutes, and the method was validated as per US FDA and EMA guidelines. So now materials and method. So the API of methylprednisolone, IS, and blank human plasma samples are obtained from MS Tab Biostudy Services, Jadavpur, Kolkata, as well as the chemical and equipments also. So here I used the methanol, milky water, and formic acid as a chemical. And it is the methods, the HPLC and ESI MSMS method are used. Here the picture of my LCMSMS. So now LCMS condition. The column we used there, the C18 phenomena kinetics column. And in here, the mobile phase I use in pump A 0.1% formic acid in milky water, and in pump P I used 0.1% formic acid in acetonitrile. And the flow rate is 0.5 ml per minute, and the injection volume is 10 microliter. And previously I mentioned the runtime is 7 minutes. So here the gas and source parameters. So here the parent ion of methylprednisolone is 375.4 and the propanolol is 260. The daughter ion means the product ion is 161, and for the propanolol is 161.1. So the DP, the declustering potential is both 46 and 25. Focusing potential is 377 and 393. The collision energy is 44 and 25. And the entrance potential is 4.60. And for propanolol, it is 8.50. And the exit potential, CXP is the exit potential. It is 11 for methylprednisolone and 2.00 for propanolol. All are I found as per the trial and error method. So here the carton gas is 38 and the cat gas is 5, the ice is 5500, the temperature is 400 degree, and the gas 1 is 45 and gas 2 is 45. So now here I prepare the plasma extraction procedure and the sample preparation. So at first we draw the spike blank human plasma samples from the.
Sorry to interrupt. You have 3 minutes left to conclude.
Yes, ma'am. Okay. Withdraw the spike blank plasma sample from the freeze, and then we use the 100 micrometer blank plasma and 50 microliter drug and 50 microliter ice, and we centrifuge in 12,000 RPM, and then separate the upper natant solution, and then give the LCMS method. So here the concentration, the 800 nanogram per ml to 12.50 nanogram. So here the figure, the Q1 scan, MS2 scan, and the calibration curve, and here the peak. So here you see, as per the US FDA guideline, that LLOQ accuracy is 80% to 120%, and LQC, MQC, HQC is 85% to 115%. So you see in interday and interday, all the accuracy is within the range. So my method development is. So here the pharmacokinetics parameters. So in the test formulation and the reference formulation, it is that showing the 91.86%. And here the mean plasma concentration time profile for methylprednisolone. So here the conclusion, the bioanalytical method development for methylprednisolone is highly specific, scientific sensitive, and the found very simple and reproducible. And the method involves a simple specific sample preparation, only 100 microliter plasma is needed. The overall analysis time is time promising. The method was validated as per US FDA and EMA. This method was successfully applied for pharmacokinetic studies and BAB studies. This is the acknowledgement, the tab by study services, and thank you.
Thank you for nice presentation.
Mr. Devyadas for your presentation.
Thank you, sir. Thank you, ma'am. Am I stopped sharing, ma'am?
Yes, we have stopped.
Okay, thank you, ma'am.
Thank you so much. So next, I would like to welcome our next presenter, Shanmuga Priya V from Vinayaka Missions Medical College and Hospital from India, who will be discussing about the role of aromatase in the pathogenesis of uterine fibroid. Ms. Shanmuga Priya, yes, you may begin.
Kita aja tuh 6 tambah 2 lah kan. Enggak dong. Kasih Pak.
Screen visible, ma'am?
Yes.
Yes, thank you, ma'am. Shall I start, ma'am?
Yes, you may begin.
Yeah. Very good afternoon to one and all. I am Dr. Shanmuga Priya, Professor Biochemistry, Department of Biochemistry, Vinayaka Mission Medical College, Karaikal. My topic for presentation today is the role of aromatase in the pathogenesis of uterine fibroids. So to start with, uterine fibroids are the commonest benign tumors of the uterine smooth muscle cells, commonest cause of gynecological disturbances and in women, and to get hospitalized and undergo hysterectomy. It is the commonest cause of hysterectomy even in this modern era. The prevalence of the disease is 20 to 50%, or 1 in 20 women will have fibroids in their reproductive age group. The fibroid tissue is found to consist of extracellular matrix tissues like collagen, fibronectin, proteoglycans, and they are prone to undergo degeneration sometimes in the postpartum period with calcifications also. Studies shows that the reproductive hormones, estrogen and progesterone, proliferate the tumor growth. Depending on the location, the fibroid tissue can be classified as subserous, submucous, and intramural types. They rarely appear before menarche, and they usually regresses after menopause as they are hormonal dependent. 1 in 1,000 fibroids may turn into malignancy at the later stages of age. They may simply remain asymptomatic. If symptomatic, they produce wide range of symptoms starting from dysmenorrhea, abnormal or irregular bleeding called as menorrhagia, non-cyclical pain, pelvic pressure symptoms, abdominal distension, bladder and bowel disturbances. They are known for their reproductive problems, including infertility, miscarriages, preterm labor, and postpartum hemorrhages also. So the aims and objectives of this study is to analyze the uterine steroid hormones, estrogen and progesterone in fibroid patients, and to compare the same with healthy study subjects, to evaluate serum aromatase enzyme status in patients with uterine fibroids. The study was conducted in Department of Biochemistry, Vinayaka Mission Medical College, Karaikal. A total of 105 study subjects, including 65 cases and 40 controls in the age group of 30 to 45 were included and investigated in the study. A detailed history of the patient collected, including clinical, personal, and family history. Uterine fibroid patients in the age group of 30 to 45 years were included in the study, whereas diabetic, hypertensive, Postmenopausal women, patients on hormonal treatment, jaundice patients, and patients on other long-term treatments were excluded from this study. The parameters tested were fasting blood sugar, urea creatinine, liver function test, lipid profile, calcium and phosphorus. They were done by standardized kit methods. Vitamin D was done by ELISA method. Oxidative stress marker MDA by Estrabov and Steinberg method. Also, we have done the complete blood clone, blood grouping and typing by gel agglutination method. Under hormone analysis, we did estrogen, progesterone, and testosterone by chemiluminescence method in Maglomi 2000 TurboChem autoanalyzer. Serum aromatase was done by ELISA method. The results, demographic characteristics of the study subjects. Patients with uterine fibroids had earlier age of menarche as well as earlier age of their first childbirth when compared to the controls. BMI was little elevated in patients, but that was not significant. The mean age of cases of 40.8, whereas in controls it was 38.7. Coming to the blood group, 42 out of 65 patients, that is 64% of the patients were found to have B positive blood group. Whereas when we analyze the control blood groups, 13 out of 40 were O positive. O positive was the major blood group type among the controls. The next groups were A positive and B positive, and 2 of them were O negative. Biochemical characteristics, as expected, hemoglobin was 8.8 in cases when compared to the control it was 10.8. Alkaline phosphatase among liver function tests was significantly elevated in cases as is used to increase in normal pregnancy, any pathogenesis of uterine fibroid and uterine muscle tissues. Serum calcium was significantly lower in cases, so also vitamin D. Vitamin D is actually half of that which was found in that of the controls. The oxidative stress marker was doubled in cases with uterine fibroids when compared to that of controls. Hormonal analysis. Serum estrogen was significantly elevated in cases with uterine fibroids compared to the controls. There was not much difference in the progesterone level, but coming to testosterone level, again there were significant levels, statistically significant increase in the level of testosterone in uterine fibroid cases when compared to the other controls. The serum enzyme aromatase was actually doubled. The values were 13.8 plus or minus 3.9, whereas in controls it was 6.2 plus or minus 1.9 in the levels were significantly elevated in cases with the uterine fibroids. Vitamin D level is decreased in patients who also serum calcium and phosphorus, which are dependent on vitamin D. Vitamin D3 calcitriol functions as a strong antifibrotic factor by inducing a remarkable reduction in the expression of collagen and other tumor growth factor beta-3 dependent key pro-fibrotic factors in human fibroid cells. So decreased vitamin D3 has caused a decreased antifibrotic effect of this vitamin D3 result in uterine fibroids in these patients. Oxidative stress marker, myomatous uterus accommodates large number of enlarged arterioles and annules. It is a angiogenesis in uterine fibroids accommodates this large number of arterioles and venules. So cyclical contraction during menstruation, traumatic injury by hypoxia, myometrial cell hypoxia, fibrosis may lead to oxidative stress resulting in free radical formation. And estrogen is a known prone oxidant, so all these factors will result in an anemia. In addition to this, makes patients more prone to develop oxidative stress. Coming to serum aromatase enzyme, it is a naturally occurring enzyme located in multiple tissues like brain, muscles, testicles, ovaries, placenta, lining of uterus. It is an enzyme that converts testosterone and estrogen in the liver tissues. Higher amount of aromatase in circulation, the more the amount of testosterone being converted into estrogen. So in testosterone, the methyl group is oxidized and it is hydroxylated to form estradiol. Aromatase activity increases with age in line with testosterone that usually decreases with age. Aromatase is found to increase with the amount of fatty tissue present.
Dr. Shankar Priya,
Yes, ma'am.
we have about 3 minutes left. You may conclude.
Yes, ma'am, I'll finish it off. And subcutaneous fat is one of the source substrate for this action of aromatase enzyme. Poor nutrition, weight gain, high stress, and lack of exercise also known to increase this level. So sufficient circulating level of estrone can be produced from this extraglandular aromatization of androstenedione. So subsequently, such estrogen reduced to the biologically active estradiol in peripheral tissues to cause uterine bleeding and endometrial hypoplasia in obese anovulatory or postmenopausal women. So aromatase activity appears to be enhanced in certain estrogen-dependent local tissues next to breast tissues like endometrium in endometriosis as well as in uterine fibroids. There are many studies shows that aromatase inhibitors are effective in the treatment of myomatous uterus as well as in postmenopausal bleeding. So to conclude, symptomatic women typically suffer from menstrual disorders affecting the quality of our life, causing a negative impact. So it also imposes mental, social, and economic burden, resulting in work loss and productivity. So identification of risk factors and timely management with medical treatment could preserve the uterus so that it doesn't affect their fertility in reproductive women. Serum aromatase enzyme can be used as an early marker for screening in the age group of 30 to 45 years for the women who are at risk of developing uterine fibroids. And vitamin D antioxidants can be supplemented in the medical management. Earlier age of menarche and first childbirth are known risk factors, so such patients can be followed up and can be screened for the development of uterine fibroids in future. Thank you so much.
Thank you, Shanmuga Priya.
Thank you, ma'am.
Next, I invite K. Amudavalli from JKKM Institute of Health Sciences College of Pharmacy from India, who will be presenting a review article of anticonvulsant activity of herbal drugs. Amudavalli, can you please share your screen and begin the presentation?
Okay, ma'am.
We are running short of time. Can you please fasten?
Okay, ma'am.
Amudavalli, sorry, we have to move on to our next presenter. You may resolve your issue and come back to us. So next I invite Mr. Deepak Ganesh Joshi. Mr. Deepak, are you ready with your presentation? Mr. Deepak, can you hear us? Presenters requesting you all to be present as per the agenda with your slide presentations. We will be not waiting for the presenters to come and join. Our next presenter is Abhimanyu. Amudavalli, it's ready?
I'm not yet ready.
Yes, then you may begin your presentation. Amudavali, we have to move on to the next presenter. Next, I invite Saravanan Muniappan to begin his presentation. Thank you, Saravanan, for joining us. Saravanan Muniappan from Vinayaka Missions Research Foundation, India. Your audio is not audible. Can you begin the presentation? Mr. Saravanan, are you able to hear us?
Yeah, can you hear me, madam, now? Hello? Hello?
Can you hear me?
Can you hear me, madam? I can hear you.
Yes, please begin your presentation.
Sure, madam. 2 minutes. 1 minute, 1 minute. Can you see the slide, madam?
Yes.
Okay, fine. Thank you very much, madam. So myself, I am Saravanan. I'm a full-time research scholar doing my full-time PhD under the guidance of Arul Balasubramanian. He is my guide, and Kotha Ramalingam and Dr. J. Bandrakar, both were my DC members. So my title is In Silico Docking Studies on Fucodens and derived from brown seaweed Sarcospermum marginatum, twitching as a dengue virus NC2B and NC3 protease inhibitors. So this is my title. So as you know that dengue virus infection costs around 50,000, around 5 lakh deaths every year all over the world. So my focus was to develop a novel molecule to combat this dengue viral disease. So the major aim is to target this viral protease enzyme that is shortly called as NS2B, NS3 Pro. And my target enzyme is this enzyme, and the plant which I've selected, this is my work plant called Sarcospermum marginatus, a brown seaweed widely available in Tamil Nadu. It comes under the Phaeophyceae family, found rich in fucodens. So fucoden is a macromolecule coming under sulfate polysaccharide, have repeated units of galacturonic acid and maluronic acid. So these fucodens were essentially studied for antiviral activity. It was proven antiviral activity was already evident with their activity against HIV, HPV, flu, and SARS viral strains. So this has given the motivation to go for this study. So my work is to perform in silico docking studies, and my objective were to select viral enzymes, then go for the selection of fucoden derivatives, and in silico docking studies and finding of the lead fucoden molecule. So by the you can see the picture, the various enzymes which is having the which covers the entire section of that virus. So my target is on NS2A, NS2B, and NS3 virus. So the methodology goes like this. I've taken Marvin sketch for drawing the structures and protein data bank for deriving the components, and for simulation software, as some software I used Discovery Studio, PyRX, and PyMol, and the fucoden ligand molecule I've taken from PubMed. So the target enzymes were protease helicase, particularly NS2B, NS3 protease. The other two enzymes were methyl transferase and polymerase. So these are the viral target enzymes you can see. I have quoted only 6 images over here. The first 3 were the dengue viral protease from dengue virus 1, 2, 3. Because dengue virus got 4 different strains, I have targeted all the 3 viral enzymes of dengue virus 1, dengue virus 2, and dengue virus 3. All were NS2B and NS3. So the target biomolecules, what I have done is, fucoden is, even though it is a polysaccharide, by converting that into a monomer, then I'm going for the synthetic derivatives. So my target is to go for acetylation, amination, sulfonation, and phosphorylation. So this is how the compound finally comes out. Acetyl derivative, it's monoacetyl and diacetyl, 3,4-diacetyl. The same way we got aminated fucodens, sulfated fucodens, and phosphated fucodens. So it's a disulfated, diphosphated, diaminated, diacetylated fucodens. So how I'm planning to go for that is go for acetylation, whereby I get acetyl fucodens, amination, sulfonation, phosphorylation. These are the short scheme which I've quoted over here. So this particular enzyme will be targeted by these 12 molecules against this fucotin molecule, this totally 4 actually, acetylfucodens, aminofucodens, sulfated and polyphosphated. This will be targeting on this NS3, NS2. But to begin with the study, I've tried with all the enzymes, totally around 144 compounds we have docked. Out of this, I've selected 3 compounds that falls under NS2B and NS3. So out of this, total 36 compounds were tried. Particularly, as I told you earlier, protease NS2B, NS3 Pro was used. And when I combined the results, so these were the docked figures of my molecule. This is acetylfucoden with a binding affinity of -7.8. When we go for the aminofucodens on 3-LSP, this is about dengue virus 1. This gives a higher binding affinity of -10.8. This is against dengue virus 1 protease inhibitor. Then coming to sulfated, little bit okay, -9.9, and finally for the phosphated fucodens it is -1.4. So 3-LSP, dengue virus 1, NS2B, NS3 protease has shown good antiviral activity. These are the binding sites and their corresponding amino acids of the acetylfucodens, amino, sulfated and phosphated fucodens against 3L6P, dengue virus 1, NS2B, NS3 protease. And coming to the second viral protease, that is 2-FON. This is derived from the protein database, the crystal structure. So this also has given least binding affinity acetyl derivative -5.9. But when compared to acetyl derivative, amino derivative has given good binding affinity of -9.9 when compared to other two amino derivatives. And sulfated, it was a little bit less in this case, -6.9. And but phosphated derivative has shown good activity on 2-FOM, that is dengue virus 2, NS2B, NS3 protease. And these are the binding scores which I've got and the corresponding amino acids for dengue virus 2 against NS2B, NS3 protease inhibitors. The third enzyme which I've targeted is 4M9K, dengue virus 3, that is strain number 3. Against acetylfucodens. Again, here acetylfucoden has shown the least binding affinity, but aminofucoden has shown a good affinity of -10.8, which was very much promising. So again, this sulfated fucoden got a little bit less of -6.9, and phosphated was a little bit okay, -7.4. So this are the binding scores of all the 4 derivatives like acetyl, amino, sulfated, and phosphated fucodens. So coming to the discussion, so total 36 compounds I have taken. Out of all these 36 compounds were tried against all the amino acids, all the target enzymes like protease, helicase, methyltransferase, and RP, that is RNA dependent RNA polymerase. Out of this 144 compounds, I have cut shorted into 36 compounds. Out of these 36 compounds, I have targeted on one particular enzyme, that is NS2B, NS3 protease, shortly called as NS2B, NS3 Pro, and it was targeted against 3 different viral enzymes, viral strains, that is dengue virus 1, 2, and 3. So all the 4 derivatives were tested, and ligand binding affinity was found to be more in case of amino fucodens for dengue virus 1, 2, and for 3 it was sulfur-phosphated fucodens. But the range of binding affinity which I have studied, which I have noticed, was -10.8 to -9.9. So aminofucodens and over-sulfated fucodens are active against dengue virus 3, that is 4M9K viral strains, where compared with the other 2 strains, dengue virus 1 strain, that is 3-LSP, and dengue virus 2 strain, 2-FOM. So out of this, we can conclude that amino derivatives and got more binding affinity. So an aminated fucoden may be a lead target molecule for the future studies. So with this short presentation, because the time was running very short. So with this short presentation, I conclude that amino and sulfated fucodens are good lead molecules which can be developed to combat this dreadful disease called dengue virus. Because now we are in the field of bio. My work was started with the dengue virus at that time. Now the corona has come. So my next target was trying the same molecule for coronavirus also. So I thank the coordinators giving this opportunity. Once again, I thank my guide and my DC members for motivating me in all such means to complete my research work. Thank you very much. Thank you for your.
Thank you so much, Mr. Saravanan, for your valuable presentation. From now on behalf of Jessila, I will be taking care and I will be the host for the respective conference. So I would like to call the next participant, K. Amudavalli, from JKKM Institute of Health Sciences, College of Pharmacy, India, on a topic of a review article of anticonvulsant activity of herbal drugs. Ms. Amudavalli.
Yes, sir.
Can you please share the screen and present?
இந்த மேடம் கடுத்த தாட்டம் கரெக்டா 4 மணிக்கு சொன்னாரு 4 மணிக்கு மீட்டிங்கா
Ma'am, can you start sharing the screen and present?
உங்க அப்பா பேச போறாரு உங்க அப்பா பாக்குறியா
Mr. Mudavali?
They're not visible in the they're not visible.
Ma'am, your voice is not audible, ma'am. It's the network problem, it seems. Please check your network connection once.
Okay, sir.
I request you to correct your network correction and please reconnect again. Meanwhile, we'll go with the next participants.
So okay, sir. The next participant.
Thank you so much, ma'am. Next, I would like to call Mr. Deepak Ganesh Joshi from Sri JJTU, Rajasthan, India, on topic of investigation of in vitro anti-arthritic activity of aqueous extracts of leaves of some medical plants. Mr. Deepak Ganesh Joshi. Mr. Deepak Ganesh Joshi, are you available? Okay, we will go with the next participant, Mr. Abhimanyu Nepal from Jadavpur University, India, on topic in vitro anti-diabetic activity of Asianathanathus sikimnes staph. Mr. Abhimanyu. Mr. Abhimanyu Nepal. Okay, it seems Mr. Abhimanyu Nepal is also not available. So we will go with the next track of topics, which is on drug design and development. The participant is Dr. V. Kishore Kumar from JKK Nataraja College of Pharmacy, India, on topic of evaluation of ethanolic extract of Calamus rotan L for anti-diabetic activity. So Dr. V. Kishore Kumar.
Yes, sir.
Yes, you can please share the screen and proceed.
Sir, slide is visible, sir?
Yes, it's visible and your time is exactly 10 minutes. You can proceed.
Okay. Thank you, sir. Good afternoon to all. I am Dr. V. Kishore Kumar from JKK Nataraja College of Pharmacy. I am going to present the presentation evaluation of ethanolic extract of Calamus rotan for anti-diabetic activity. So this plant is medicinally used as a anti-diabetic purpose. Traditionally, the raw fruit 1 to 2 taken for the daily 6 to 8 weeks to control diabetes. So that's why we selected this plant. Unfortunately, we didn't collect the raw fruit, but we studied only in the leaves. The aim of the study, the present study was aimed to evaluate anti-diabetic activity of Calamus rotan leaves. The objectives, the collecting and configuration of confirming the plant material by authentication, preparation of extracts, and doing phytochemical studies, screening of toxicity studies, screening of anti-diabetic effect by in vivo. The plan of work, plant collection and authentication, preparation of extract, preliminary phytochemical analysis, toxicity studies by acute oral toxicity studies OECD 425, pharmacological studies by in vivo. Methodology, plant collection, extraction, analysis, phytochemical analysis, acute study, anti-diabetic activity. This is the plant profile, Calamus rotan. It should be called as Pirambu in Tamil, Choral in Malayalam. Parts used whole plant. We selected only leaves for our study. Plant collected in Malappuram district, Kerala. The air-dried, coarsely powdered leaves were extracted with ethanol in percolation process by using Soxhlet apparatus. Then the extract are screened for various phytoconstituents. Then acute oral toxicity studies was performed by OECD guidelines 425. So anti-diabetic activity, for this we selected Richter albino rats. In that rats we induced diabetes by streptozotocin and nicotinamide. Then the diabetic rats were grouped 5 groups, each groups 6 rats. Then the leaves extract 100 and 200 mg per kg were induced daily for 28 days. In the meantime, we evaluated blood glucose level and body weight per weekly basis. Finally, we estimated various biochemical parameters. Likewise, glycosylated hemoglobin, total cholesterol, triglycerides, proteins, and liver enzyme markers, hemoglobins. The result and discussions. The phytochemical study shows the presence of carbohydrate, alkaloids, saponin, flavonoids, tannins, and phenolic compounds. In acute studies, the extract, ethanolic extract, must be greater than 2000 mg per kg, does not produce any sign of toxicity. Biochemical estimation, the fasting blood glucose level. The fasting blood glucose level was shows 66.9% in 200 mg per kg dose, more or less near than glibenclamide. Then the changes of body weight also, we got excellent improvement of body weight in dose of 100 and 200 mg per kg when compared to diabetic control. Then HbA1c and hemoglobin level also, we got very excellent effect by using ethanolic extract of calamus rotanglin with the dose of 100 and 200 mg per kg when compared to diabetic control. Then the different cholesterol levels and triglycerides, we observed the dose of 200 mg per kg are more or less near than glibenclamide treated groups, like the percentage 51.35, 52, then 56.95, 58, 72, 70, 56.93, 58, 46, 48. So this 200 mg per kg treated groups are more or less near than standard treated groups when compared to diabetic control. The same time, the liver markers are AST, ALT, ALP also increased in diabetic rats. After the treatment, we got very much great effect in the treatment of calamus rotanglin ethanolic extract. Histopathology also, in diabetic rat, we seen the reduction of beta cells after the treatment with 2 dose and standard drugs. We got excellent regeneration of marked normal regenerated beta cells. Finally, we can conclude this present finding clearly demonstrate the ethanolic extract of calamus rotang exhibited excellent anti-diabetic and also anti-hyperlipidemic activity in STZ dicotinamide induced diabetic model. Indeed, this study was undoubtedly provide scientific confirmation and evidence for the safety and use of leaves of calamus rotang by traditionally. However, further studies, we can go for isolation and purification of bioactive compounds from the leaves extract for the molecular mechanism can be carried out in diabetic and diabetic complications. Acknowledgements, reference. Thank you. Thank you for your.
Thank you so much, Mr. Kishore Kumar.
Thank you.
Please can proceed with the chat box session. If they want to raise, they can raise queries within the chat box. I would like to call the next presenter, Ms. Pallavi K. Wahal from PES Modern College of Pharmacy, Savitribai Phule, Pune University, India, on topic of synthesis and biological evaluation of novel curamine derivatives for its anti-diabetic activity. Ms. Pallavi?
Yes, sir, I'm here.
You can start sharing the screen and present.
Yeah. Yes, can you see my screen, sir?
Yes, we can. And the time is exactly 10 minutes. You can proceed.
Hello, good afternoon, everybody. My name is Pallavi. I'm from Modern College of Pharmacy, Savitribai Phule, Pune University. And today I'm going to present my topic that is thesis and biological evaluation of novel coumarin derivatives for their anti-diabetic activity. So as we all know that coumarin is a very well-known compound, and it is having various biological activities like anticoagulant, antimicrobial, anti-inflammatory, analgesic activity, antioxidant activity, and many more. So in this study, what we have done is we just fuse coumarin with other heterocyclic compound to give and we evaluate that coumarin derivatives for their biological activities. Various biological activities. Here I am presenting only anti-diabetic activity. So first what we did is the docking. After selecting pharmacophore and depending on structure, after selecting pharmacophore, we did docking studies, and from that docking studies we have selected the compound which are having good binding energy with that of ligand. So after that we have decided the synthetic pathways for that molecules. So two synthetic pathways we have selected. So the first one, some of the steps are common in the synthesis. The first is 3-chlorocoumarin. Sorry.
Excuse me. I request all the participants to keep on mute. The respective session participant is struggling. Sorry, ma'am, you can proceed.
So the step one is the synthesis of 3-chlorocoumarin from 2-hydroxyaldehydes or ketones. So in this step, what we did is we are using we had used 2-hydroxyaldehydes or ketones reacted with triethylamine in presence of chloroacetyl chloride and chlorosulfonic acid to give 3-chlorocoumarin. After that 3-chlorocoumarin we converted into 6-sulfonyl chloride. And that 6-sulfonyl chloride in the third step we converted into 6-sulfonamide. Then in this 6-sulfonamide, now these are the 3 steps which are common for both the pathways. After that, the fourth step in the first pathway is the synthesis of substituted isocyanate we have prepared simultaneously by using different anilines. And these substituted isocyanates then we treated with 6-sulfonamide to form sulfonyl ureas, coumarin sulfonyl ureas. And in the second pathway, we have used different indole derivatives to form indole coumarin derivatives. So these indole coumarin derivatives and sulfonyl ureas coumarin, these we have. Sorry, is there any question? Okay, so these derivatives we have first characterized by using different spectroscopical methods like IR, NMR mass. Then CH analysis also we have performed for these different derivatives. And after that we have biologically evaluated this derivative for its in vitro antidiabetic activity and in vivo antidiabetic activity. In case of in vitro antidiabetic activity, we have used rosiglitazone as a standard compound. We have compared rosiglitazone as a standard, and in vivo activity we have induced the diabetic activity in rats by using streptozotocin and nicotinamide. Here what we have in result and discussion, what we have found is some of the derivatives are active. We have actually optimized the synthesis so that we can get the compounds which are having optimum molecular weight to give the good activity. So 2 compounds are active. 30 derivatives we have prepared. The total 30 derivatives we have prepared from 2 synthetic pathways. From these 30 derivatives, 2 are active. Then 1 possess moderate activity, and another 2 molecules are having comparable activity with that of rosiglitazone. Conclusion of my work is in present study, novel derivative coumarin derivatives synthesized and evaluated for their antidiabetic activity. Derivatives were screened for their DPP-4 inhibitor activity because DPP-4 inhibitors are the most novel compounds, most preferred method for this particular thing. And we have used sulfonylurea along with coumarin, so that because of that SO2 group, that might give the good activity for longer period of time. So our focus is that only, that we have to take for diabetic patient have to take daily dose of their anti-diabetic drug. So we want to prepare a drug which will give a longer action. So that might be possible with this drug. And these are some references which we have used. Thank you.
Thank you so much, ma'am, for your valuable presentation. Next participant is Subhash Sharma from Jivaji University, India, on topic of development and characterization of natural polymers based gastroenteric tablets for the effective management of diabetes. So Subhash Sharma.
Miss Pallavi, remove your slide.
Yeah.
It seems Mr. Subhash Sharma is not available. We'll go to the next presenter, Ms. Ukti J. Bhatt from National Institute of Pharmaceutical Education and Research, India, on theoretical and experimental validation of praziquantel with different polymers, miscibility, phase diagram, and estimation of Florey-Huggins interaction parameter to select appropriate matrix for hot melt extrusion.
Hello.
Yes, you can share the screen
yes.
and start presenting.
Yeah, sir. Am I audible? Can I share my screen?
You can.
Yeah.
Is my screen visible?
Yes, ma'am, it's visible. The time is exactly 10 minutes. You can proceed.
Good afternoon, one and all. I'm Ukti Bhatt from Department of Pharmaceutics, National Institute of Pharmaceutical Education and Research. I would like to thank, first of all, I would like to thank Bioleagues and APR for giving me wonderful opportunity. My today's topic is the validation and theoretical experimentation of praziquantel with different polymers, and we will focus. We have focused on miscibility, phase diagram, estimation of Florey-Huggins interaction parameter, followed by hot melt extrusion. Coming to the hot melt extrusion, so the industrial application of hot melt extrusion has been established since 1930s. Thus, we can consider the melt extrusion as a well-developed manufacturing technology in vast field. In HME, the drug, polymer, and required quantity of plasticizer is premixed and feed into feeder. We can see on the right-hand side, schematic of a typical extruder, and on our left-hand side, thermoscientific hack mini lab 2 that we have in our pharmacoengineering lab. So the premixed mixture is further thoroughly blended under given temperature and desired rotation with a single or twin screw rotation, and amorphous solid dispersion is formed through hot melt extruded. This filament is further we get of a desired diameter. Coming to the introduction of 3D printing, that is additive manufacturing. So the filaments that are formed from the hot melt extruder is fused into FDM, mediated single or dual nozzle extruded 3D printer to generate fascinating prototypes, thereby creating a versatile method of prototyping numerous drug delivery system with additive manufacturing approach. Additive manufacturing is one of the industrial manufacturing process where product is formed in parts and combined together to form the final product. In the category of manufacturing, 3D printer is a major class of additive manufacturing. So here we have formed a filament through hot melt extrusion. The filament that we have formed is drug-loaded filaments. In the right-hand side, we can see fuse deposition modeling, Ultimaker 3 of our lab, pharmacoengineering lab, and the rapid prototyping of layer by layer formation through FDM. So in previous studies, we have seen that the marketed filaments that are available were typically immersed for 24 hours in API organic solvent solution, and they were then over-dried for the drug loading, and the impregnant drug loaded was very less. So there was no approach for a theoretical and validation formation of a drug loaded filament with a stable which can be processed at high temperature with a stability, and for that theoretical and practical validation has to be done. So coming to the aims and objective, so here theoretical and experimental validation of praziquintal we have done on the selection of 3 different polymers. We have done the miscibility study through Hansen solubility parameter and Fleury-Huggins interaction parameters melting point depression approach. Further, we have formed a phase diagram and estimated the interaction parameter. On the basis of that, a process parameters for hot melt extrusion was formed. The HME drug-loaded filament were further characterized for stability, DAC, ATR, PXRD, and the filament were further coupled with FDM to form a desired shaped personalized medicines 3D printed caplet. So these are the objectives. Coming to the rationale for selection of polymers, over here we can see from HPLC force degradation study that we have performed, the praziquintal showed high degradation in alkaline pH. So on the basis of that, 3 polymers with praziquintal were evaluated: Eudragit EPO, polyvinyl alcohol, and Collicott IR. Further, miscibility studies were done. So, difference in the solubility parameters between the drug and polymer are indication of drug polymer miscibility, and it has been said that it should be less than 10 millipascal by the Green-Gall experimentation. So further, a positive interaction value, if these are the results where we have estimated the interaction values. So for a positive interaction value indicates immiscibility, negative values indicates miscibility, and the delta value should be less than 10. So here we can see Eudragit EPO having the least delta value with the praziquintal, showing the high miscibility when it will be form a solid dispersion, the high miscibility will be obtained, and a stable solid dispersion with hot melt extrusion will be formed with no use of organic solvent, which is another big advantage of hot melt extrusion. Coming to the melting point depression that we did on the basis of Fleury-Huggins interaction parameter. So a better understanding of drug polymer miscibility and their mutual interaction in solid dispersion helps to avoid phase separation or crystallization of API, which is driven by the supersaturation at drug loading beyond the solubility or miscibility limit of specific polymers. So here we have evaluated drug polymer miscibility via melting point depression approach using DAC to obtain high precision results. Physical mixers of praziquintal with EPO, praziquintal with PVA, praziquintal with Collicott IR in the ratio of 90:50 were made, and further DAC analysis on Mettler Toledo was performed, and melting point depression graphs were obtained as such. These 3 melting point depression graphs is praziquintal with EPO the first, praziquintal with PVA second, praziquintal with Collicott IR is the third. Further, the values were taken and to investigate the effects of temperature and free energy on miscibility between praziquintal and the 3 polymers. We next constructed phase diagram using the results that we have obtained from DAC, and the values, the interaction parameters were calculated with each and every temperature point, and the values obtained from interaction parameters were then plotted against the 1/Tm, and the graph of phase diagram were obtained. So from these, constants were obtained that each x calculated using the constant A and B in each systems has subsequently subsequently affected in formation of liquid solid miscibility curve and Fleury-Huggins T5 phase diagram was formed. So here, by the judicious approach of T5 phase diagram, we have identified an optimal HME processing temperature for praziquintal at a predefined drug loading with three different polymers, and through the evolution under fixed drug loading, and three levels of temperature were selected. And next, the free energy drug loading diagram was formed. So from here, the filaments of theoretically and experimentally validated filament in which the BCS class 2 drug praziquintal is stable, solid dispersion has been formed with the processing temperature that further degradation will not be done due to the higher processing temperature, and the praziquintal filaments were formed. These filaments were further characterized for DSC analysis, PXRD and ATR. We have found some wonderful results for hydrazine EPO and some really interesting results for polyvinyl alcohol, and Collicoat are same. Coming to the main conclusion part, if I say hot melt extrusion was taken, the hot melt extrusion hack Minilab CTV, and further filament was produced through proper validation of phase diagram through DSC, and filaments were characterized. And these filaments, this is the part one. The study has been, the whole research has been divided into three parts. I have represented here the one part of my research. I would like to give the scope of the further research which has been going on right now in our pharmacoengineering lab. So these filaments is currently going under the further evolution of filament in terms of flexibility and mechanical strength, and we are using no plasticizer over here or no any other additives we are using, and which and we are making a filament which is further integrated into fused deposition modeling. The model, the FDM machine that we are using is Ultimaker 3, and the personalized dosage form that is 3D printed caplets are formed. Here, the second phase of study in which we are performing in-depth study of the filament that has been formed through validation of phase diagram, and in-depth flexibility and mechanical strength, and in-depth study through Repka-Zhang filament test for further integration into the FDM. And here the further scope of study that we have done is we have optimized each and every process parameters of fused deposition modeling 3D printing, which could affect this final dosage form. So this here by here, I conclude my presentation. Thank you so much. Thank you for showing the patience. Thank you.
Thank you so much, Ms. Yukti Bhatt, for your valuable presentation. Session chairs or participants can raise any queries if they have. The next participant is Ms. Devashi Kar Mahapatra from.
Sir, I'm here.
Yes, sir. Yes, you can raise the query, sir.
No, so I'm the next.
Okay, you are the next participant.
Presenter.
Okay, you are Mr. Devashi.
Yeah,
Yes,
so
you can
can
share
I
the screen
stop sharing my screen?
Yes, definitely. You can stop sharing your screen, ma'am.
Thank you so much.
Devashi will share the screen now.
Yeah, sure.
Yes,
Very
your
warm
screen is visible.
Yeah.
It's exactly 10 minutes. You can proceed.
Yeah, so welcome everybody. This is Debashika Mahapatra. I'm ICMI SRF Fellow at Department of Pharmaceutical Sciences in Dr. Hari Singh Vishwavidyalaya in Sagar, Madhya Pradesh. So this is one of the work which we have done in this lockdown period, and as an alternative for the lab work, where we have worked on using the Design Expert software assisted optimization of catechin soya phosphatidylcholine complex ratio, which can be further, which is having perspective in formulating phytosome formulations. Well, it is already known that there are several natural products which are having multiple pharmacological activities, and in our daily life we use a lot of natural products. But somewhere it's been a myth, or sometime it proved that the synthetic compounds, they work very well as compared to natural. But while going to the fault, we can see the main issue rises with the bioavailability and its metabolism, its high rate of metabolism. So therefore, one of the best ways of protecting this type of compound from metabolism as well as to enhance the bioavailability, the best part is to make phosphoryl complexes as well as making them into phytosome formulations. So here, where we worked on the formation of catechin. You know that catechin is one of the most important phytoconstituent present in everyday life, that is in tea. That you have all catechin components as well as whatever food you eat, majority are having the catechin. But how, although they are known as antioxidant, but whether this antioxidant property you are enjoying or not, this is a real question mark, because majority of the components get degraded once it reaches the GIT. So therefore, this is one of the approach where we made a sulfosylcholine complex with the catechin, where it forms an envelope. So now we tried for 14 batches with the different drug ratio, that is the catechin as well as the sulfosylcholine ratio. And here, one thing I just want to draw attention, that instead of doing in the laboratory, as you know that it's very difficult nowadays to work in laboratory due to this lockdown and all. So we have another thing, that is we have statistical tools and computational aids, which are equally effective in proving or at least optimizing the formulations, where a design expert as well as JMP, and there are a lot of number of files, lot of number of softwares, they are into application nowadays. Now here we used one design expresses optimization, where we went for a quadratic equation of 3-level factorial having 13 runs, using the software which we obtained from a design experts for a 30-day trial. And here it's very difficult to get a real conclusion, because everything is a numerical. So here you can just see that we got some factorials where the coding level was produced from -1 to +1. That means a 0 means a mediocre expression, a plus means a higher, as well as -1 refers to a lower expression. So here the power calculation, we went for a delta as well as a sigma, where the delta signifies 2 power and the sigma signifies 1 power. Here you can see the mean values. Ideally, we achieved a mean of 0, which means the components are highly correlated. And however, the multicollinearity was prevented to some extent. We have also achieved the standard deviation in a very optimized level, that is less than 2. And in the next part, you can also see the response. Now we applied the factors, now we are getting the response. Here we are getting the quadratic models, where you can see the quadratic model summary is given, where the adjusted R-square and the predicted R-square have been in a significant level. And in the polynomial analysis, we got the highest order polynomial, which said that the model is not linearized. So here, then you can see the degree of freedom, which is one of the most important parameter, where we get at least 2 lack of it, which is we got a value of 3. So these we at least we have reached the things which are beyond the critical limits. We have achieved beyond the critical limits, and you can see even the pure errors is more than 3, the value of 3. We obtained a value of 4. That it's a borderline. Now we studied 3 parameters particularly. One is the complexation rate, second one is the entrapment ratio, and the third one is the drug release. So these 3 important aspects are required for any formulation. So here you can see that the standard error was in it shows that it's a very balanced design. However, much lower standard errors are preferred, but we obtained in the level of 3 to 5. Particularly, we use prediction-based matrices for developing the equation that is a plus b plus ab plus a square plus b square. And another thing is we got an ideal VIF value that is within one. So which shows that the parameters are absolutely perfect, model is optimized, and the design can be used for practical evaluation. Now you can see the equation that what we have obtained. You can read the equation from the values, where you can see from that equation we can get the complexation rate. We can at least predict the complexation rate. The leverage, you can see it is very close to 1, and even it's below 1 with an average which is very impressive, that is even less than the half of the value, which shows that the system is highly robust. Now we have one I value. However, if we got a very small I value, usually it's less than less than 0.4 is desired. We also receive, and a smaller d-optimity value we got here that helped that showed that the system is not unicollinearity. It was reframed. So now, secondly, we got the ANOVA applications, where we got the sum of square having the type 3 partial coding, as well as the factorial coding we got is particularly AB plus A square plus B square. And you can see one of the most important statistical parameter that the p values are less than 0.05, which is most important part. And that is your precision part is also greater than 4, which also signifies that the model is robust. Now we obtain several plots based on the normal plot of residues as well as residual versus predicted. And we also got, we see there are the total number of runs are 13, and you can see that all are in the higher range. You can see up to 94.26% it can have the prediction. You can see from the colors that which points they have been the highest expression, as well as the points having the lowest expression. Now here the DFF ITS with the differences fits and the Crookes distance, it also helped in predicting the collinearity of this model. Now, secondly, we have another parameter that is the influential parameter, and we have also got a box-cost power transformation, which also supported the same. Now, this is the report, all over report. Further, we also obtain the perturbation and interaction plots, which shows the difference in both the models between A and B points, which is also within the limits. And secondly, we also gain the same type of results in the entrapment efficacy, as well as we also obtained the graph, and the limit was 87.97%, which is the highest limit. Same we received for it. And further, we got a drug release,
to conclude.
and where, yeah, sure, sir. Now the same type of graphs we also received for all the three components. Now here it is the final part where we received a desirability of 1 with a prediction value of 77.45% for the complexation rate. For entrapment is 76.38%, and for the drug release it is 80.86%. So this shows that the formulation has all the 3 good characteristics, and you can see from this part also that we got the desirability of 1, as well as with all parameters for our drug, that is catechin. Now we can see the overlay plot where you can see the x1 and x2 values.
Sorry to interrupt,
Showing.
It's almost 10 minutes. I request you to close as is possible.
Yeah, sure. There's a standard error, and that's it. In the conclusion, you can say that we obtained the 3 equations that helped in forming the formulation and developing the formulation, that too using in silico tools, that is Design Expert software. And we also obtained one ratio, that is the 1:1, which is the optimized ratio we obtained from this equation. So thank you very much for patient listening to it. Thank you very much.
Thank you so much, Devashi. Queries can raise by session participants and the session chair through the chat box. The next participant is Najwa Nabila from Bandung Institute of Technology, Indonesia, on topic of exploration of biological activities of the light subunit of mushroom tyranase from mushroom Agaricus disrobus. Ms. Najwa, am I audible? Ms. Najwa, ma'am, please release the mute and you can proceed. I request Ms. Najwa Nabila, ma'am, to proceed.
Good evening.
Yes, may I know to whom I am talking to?
Yes, he is there. You can share your screen and start presenting your paper.
Okay, please wait for 1 minute. Can we move to the next presenter first?
No, it's not visible, ma'am.
Share your screen.
Okay, please wait for 1 minute.
Kindly share your screen and start presenting.
Is there any problem?
It seems there is some difficulty while sharing the screen, sir. We'll go with the next presenter.
Yeah, you can go for next presenter because we are running shortage of time.
Absolutely, sir. The next presenter is Fauzia Zara.
Yes, sir. Can you hear me?
Yes, ma'am. You can share the screen and start presenting.
Okay, can you see my presentation?
Yes, ma'am. Time is exactly 10 minutes and you can proceed.
Okay. Good afternoon. My name is Fauzia Zahra. Yes? Excuse me?
Poster presentation is 5 minutes, I think so.
Yes, it's 5 minutes.
Yeah.
Okay, so in this section, I will explain my poster presentation with study anti-cancer activity from the light subunit of mushroom tyrosinase or LSMT in breast cancer model MDAMB2 cell line and development of spheroid as tumor model. Recombinant-like subunit of mushroom tyrosinase or RLSMT has anti-proliferative activity on MDAMB2 cell line. Aberrant signaling pathway occurs in cancer such as Wnt signaling. Activity of RLSMT on regulation of Wnt signaling pathway in MDAMB2 cell line was observed due to its role in regulation of proliferation, differentiation, and self-renewal. The aim of this research is to explore the potential activity of RLSMT as an anti-cancer through modulating the Wnt signaling pathway and to develop three-dimensional culture or spheroid as an advanced in vitro model for the solid tumor. From the SDS batch analysis, the molecular weight of RLSMT is approximately 18 kilodalton, which is slightly larger than mature LSMT at approximately 14 kilodalton. The additional weight may originate from the presence of amino acid residues and the additional hexes stacked with its linker. The cytotoxic assays show that RLSMT has anti-proliferative activity on MDAMB2 cell line with the IC50 value is 579.3 micrograms per milliliter. Next, dysregulation of Wnt signaling is marked by accumulation of beta-catenin and also the accumulation of cyclin D1 and exin 2, which are the downstream targets of the Wnt signaling. Cyclin D1 has role in cell cycles and its overexpression occurs in cancer. From this experiment, treatment with 579.3 micrograms per milliliter shows slightly decrease the expression of CTNNB1 and cyclin D1 gene, and also shows slightly increase the expression of exin 2 probe. Spheroid was generated by culturing MDAMB2 cell line in serum-free medium, supplemented with EGF, basic FGF, and B27 on ultra-low attachment plate. The MDAMB2 spheroid is formed on the 7 days of culture with grape-like shape, and they have size between 50 to 400 micrometers. Then, after 27 hours incubation of RLSMT, the reduction size of MDAMB 2-to-1 spheroid is negligible compared to the negative control. Overall, we conclude that RLSMT inhibits proliferation of MDAMB 2-to-1 cell line in monolayer culture with the value of IC50 is 579.3 micrograms per milliliter. Treatment with IC50 value of RLSMT on monolayer culture of MDAMB 2-to-1 tend to reduce the expression of beta-catenin and cyclin D1 gene, although Wnt signaling was not the major mechanism of RLSMT. Given 350 micrograms per milliliter of RLSMT on MDAMB 2-to-1 spheroid shows negligible reduction size of spheroid. So further investigation on mechanism of anti-cancer activity of RLSMT and the concentration for MDAMB 2-to-1 spheroid are needed. Thank you for your attention.
Thank you so much for your presentation. The next presenter is, she's an oral presentation by Shakuntala NM from JVM's Mehta Degree College, India on topic organic photosynthesis for biomedical applications. Ms. Shakuntala. Ms. Shakuntala. Okay. We'll go to the next presenter.
Yeah.
Oral presentation by Smriti C. Shinde from AISSMS College of Pharmacy, India on topic of development and validation of stability indicating HPTLC method of estimation of dolutegravir. Ms. Smriti, you can start presenting. Ms. Smriti, please release the mute and you can proceed.
Now am I audible, sir?
Yes, you're audible now. You can start sharing and present.
Okay, to all the respected dignitaries and presenters, a very good afternoon to all of you. I'm Smriti Shinde from AISSMS College of Pharmacy, India, presenting my research work entitled Development and Validation of Stability Indicating HPTLC Method for Estimation of Dolutegravir. The contents of the presentation are as follows: aim and objectives, drug profile, experimental and chromatographic conditions, validation parameters, stress degradation studies, conclusion, and references. The main objective of this study was to develop and validate a simple, precise, sensitive, and accurate HPTLC method for estimation of dolutegravir, and to study the stress degradation behavior of dolutegravir as per ICH guidelines. Drug profile of dolutegravir. The following figure shows the structure of dolutegravir and its IUPAC name. The drug belongs to antiretroviral category and it is sold under the trade name Tibican Instra. Mechanism of action: the drug works by decreasing the amount of virus in the blood and increasing the number of immune cells that helps to fight infections in the body. Experimental conditions: chromatographic separation of the drug was carried out on aluminum plates pre-coated with silica gel 60 GF254 using a CAMAG 100 microliter syringe and a Linomat 5 applicator. Densitometric scanning was performed using CAMAG TLC scanner operated by WinCat software. The radiation source used was deuterium lamp. Chromatographic conditions. The following table shows the chromatographic conditions used for the study. Band length of 6 mm was applied on TLC plate. Mobile phase. Initially, the trials were carried out using various solvents in various proportions. After a few trials, ethyl acetate and methanol in the ratio of 9:1 was chosen as mobile phase, which gave acceptable peak parameters. The densitometric scanning was done at 258 nanometer, and the RF value of the drug was found to be 0.48. Validation parameters. The method was validated as per ICH Q2R1 guideline. The following table shows the summary of validation parameters. The R-square value of the drug was found to be 0.9976, and it was found to be that the method is linear in the range of 200 to 1200 nanograms per band. Precision studies were carried out for both intraday and interday. The percent RSD value was found to be less than 2%, which shows that the method is precise, whereas percent assay was found in the range of 99.87% to 101.38. LOD and LOQ values were found to be 34.04 nanograms per band and 103.16 nanograms per band. The method was found to be both specific and robust. Stress degradation studies, stability studies were carried out on drug to provide evidence on how the quality of drug varies under the influence of various stress conditions like acidic, alkaline, oxidation, heat, and photodegradation. The following table shows the summary of degradation parameters. Figure 1 and 2 shows densitogram of standard drug dolutegravir and acid-induced degradation with one peak of degradant in acid-induced degradation. Figure 3 and 4 shows densitogram of alkali-induced and peroxide-induced degradation, both with one peak of degradant. Similarly, figure 5 and 6 shows densitogram of dry heat and photolytic degradation, wherein the densitogram of dry heat degradation shows 2 peaks of degradant and photolytic doesn't show any peak of degradant. Degradation pathway. Given figure is the probable degradation pathway of the drug under acidic and alkaline hydrolysis. The mass to charge ratio of the drug was 419. Upon acidic and alkaline hydrolysis, the mass to charge ratio was reduced to 294 for acidic and 96 and 107 for basic hydrolysis. Figure 8 shows degradation pathway for oxidative degradation, and the mass to charge ratio was found to be 307. Conclusion: a rapid, simple, and specific HPTLC method has been developed for determination of dolutegravir. The method can be used for the routine analysis of the drug in presence of degradation products, if any. These are the references. That's all from my side. Thank you.
Thank you so much for your valuable presentation. And I would like to call the previous presenter, Ms. Najwa Nabila from Bandung Institute of Technology, Indonesia, to share her presentation.
Okay, I will start share my screen.
Yes, please. Yes, it is shared. You can proceed. The time is 5 minutes.
Okay, I will start. The title of my research is Exploration of Biological Activities of the Light Subunit of Mushroom Tyrosinaceae from Mushroom Agaricus bisporus. Actually, I will call this protein as LSMT, and LSMT shares structural homology to the hemagglutinin component of HA33 and resin B-like protein. Based on that structural homology, we address LSMT as a lectin-like protein. As we know, lectins are protein with capability to recognize and bind carbohydrate and typically able to agglutinate erythrocytes. That's why the purpose of this research is to study the binding specificity of LSMT to sugar molecule and to study the agglutination activity of LSMT. This is in this research we use two methods. First is in vitro cytotoxicity assay using CCK8 reagent and the agglutination assay with traditional method. And based on the result, we can see that we can conclude that LSMT is a mannose binding protein based on analysis with SPR instrument, because the differentiate in the peak is only happen in mannose, and then LSMT specifically inhibit the proliferation of MCF7 and MDA-MB231, because using the other cell line, although the normal cell line, there are no inhibition of proliferation by LSMT. And also LSMT is not an agglutinating protein until the concentration of 2000 ppm using four types of blood, human, and then mice, rat, and chicken blood. So the conclusion of this research is LSMT was found to bind specifically to mannose upon analysis with SPR instrument, and based on our in vitro study, LSMT has ability to inhibit proliferation of the MCF7 and MDA-MB231 cell line, and that two cell line is a breast cancer cell line. LSMT also crucial for its future development as pharmaceutical excipient of active ingredients, and it there be safely used because it wouldn't cause blood coagulation upon its introduction to the internal blood circulation system. So this property places LSMT ahead of the lectin for potential application in the therapy and medicine. That's all for me. That's all from me. Thank you.
Thank you so much for your valuable presentation. And the next participant is Namrata G. Bhavaskar from AISS College on topic of development and validation of stability indicating HPLC method for estimation of Vagansin-Slow wear.
Dr. Najwa,
Ms.
please
Namrata?
remove your slide.
Sir, please allow me to share my screen.
Definitely, ma'am. Ms. Najwa, can you please stop sharing the screen?
I already stopped.
Yes, you can proceed. Ms. Namrata, you can share the screen.
Yes, sir. Sir, is my screen visible?
Just a minute. It's still yes, now it's visible and you can proceed. The time is 5 minutes.
First of all, good afternoon to all delegates. Myself, Namrata Bhavaskar. My topic for the presentation is development and validation of stability indicating HPLC method for estimation of algan cyclovir under the collaborative cooperation of AISSMS College of Pharmacy and Mir Maharashtra Institute of Pharmacy, Pune, Maharashtra, India. Content for the today's presentation is aim and objective, drug profile, chromatographic conditions, validation parameter, result and discussion, and references. The main aim of this study is to develop and validate a stability indicating HPLC method for the estimation of valganciclovir in the bulk and the tablet dosage form. The main objective of the study is to develop and validate a simple, specific, precise, sensitive, accurate, and robust HPLC method for the estimation of valganciclovir in dosage form and the bulk drug. And second is to study the stress degradation behavior of the valganciclovir as per the ICH guideline, Q1AR2 guidelines. Then going to the drug profile of the valganciclovir. The present slide contains the chemical structure of the drug with the molecular formula. Then the IUPAC name. The therapeutic category of valganciclovir is antiviral drug. It is used to treat the cytomegalovirus infection. The valganciclovir is hygroscopic in nature, which is storage temperature is -20 degrees Celsius freezer under the inert atmosphere. The trade or the marketed formulation used in the analysis was V Gravate 450 mg tablet. Under the various trials and error, the finalized chromatographic conditions are given in this table. For the analysis of sample, the instrument where uses a Glint 1260 series HPLC system. For the separation, Waters C18 column was used with a measurement of 250 into 4.6 mm with the particle size 5 micrometer. The mobile phase was used as ammonium phosphate buffer, pH 3. The pH was adjusted by the glacial acetic acid used to methanol 55 to 45 volume by volume. The oven temperature was maintained at 50 degrees Celsius. The analyte was analyzed at the flow rate of 1 ml per minute. The total runtime was given for 12 minutes and the retention time for the standard was found to be 6.5 minutes at the detection wavelength of 254 nanometer. And in the result and discussion, the developed method was validated as per the ICH guideline Q2R1 and the results are given in the table. The linearity, for the linearity, the R-square was found to be 0.837 in the range of 5 to 30 microgram per ml. The precision was carried out at the interday and intraday level where the percent RSD value was found to be in within range, that is less than 2%. The percent SA was found to be 101.14%. The accuracy was carried out by using the standard addition method at the where the addition of standard drug at the three different level 50%, 100% and 150%. where the percent recovery was determined from the linearity of equation. Then the LOD was found to be 0.52 microgram per ml, and LOQ was found to be 1.58 microgram per ml, and the developed method was found to be specific and robust. In the developed method, the post degradation studies was carried out for the bulk drug. The present slide contains the figure 1, which is the chromatogram of standard valganciclovir 10 microgram per ml, and the second figure is of the chromatogram of valganciclovir stress sample under the UV light, in which the sample is exposed to the UV light, providing the illumination not less than 200 watt hour per meter square. And figure C is the chromatogram of valganciclovir stress under the 0.1 normal HCl. Figure D is chromatographic valganciclovir studies stress sample under the 0.5 normal NaOH. Figure E is the chromatogram of valganciclovir oxidative stress sample, in which the sample is exposed to the 10% of hydrogen peroxide. Figure F is the chromatogram of valganciclovir thermal stress sample, in which the drug is exposed to the 80 degrees Celsius temperature in the oven. Here is the summary of degradation parameter. Here we can conclude that the major degradation of sample is found in alkaline condition, where the drug is exposed to the 0.5 normal NaOH, where the percentage of valganciclovir is found to be 9.13%. Further study was carried out for the identification, characterization, and prediction of the degradation product by the LCMS, where the separation of major degradation product of alkaline condition of valganciclovir was carried out by the HPLC, and identification and characterization was carried out by LCSIMS.
I'm sorry to interrupt. It's already more than 5 minutes. I request you to close as early as possible.
Yes, sir. The degradation there are the 3 degradation products were found in the alkali condition at the 3 different RTs, and the rest of the integration list is given in the table. Here are the LCMS spectrum of the valganciclovir standard, and rest of the 3 are the degradation product LCMS spectrum. From this spectrum, we can conclude we can probably enable to predict the degradation pathway of the valganciclovir. The conclusion is the values of accuracy, precision, robustness, and linearity, and LOD and LOQ were found within limits. So the developed method can be used for the estimation of valganciclovir in bulk and pharmaceutical dosage form. And as per the post-degradation study, the valganciclovir is found to be sensitive to alkaline, oxidative, and thermal condition. So the recommended storage condition are to store the drug in cool and dry place. Here the references. That's all from my side. Thank you.
Thank you so much, Ms. Namrata, for your presentation. We are glad to receive your presentation. And we will move on. If any queries to be raised, they can raise in the chat box. We'll go to the next track, which is on pharmaceutical formulations. I'm going to present by the first presenter, Santosh Badrikaria from Jivaji University, India, on study on binding effect of extracted mucilage from Bombax saber plant flower petals for tablet formulation. Mr. Santosh? Mr. Santosh, am I audible? Mr. Santosh, can you hear me? Okay, it seems the presenter is not available. We'll go to the next one. Dr. B. Bandhu C. Chatale from MET Institute of Pharmacy on synthesis of the prodrug to mask bitter taste and its taste evaluation. Dr. Bandhu C. Chatale. Mr. Bandhu. Mr. Bandhu, can you hear me? Okay, we'll go to the next presenter by Dheri Damhuri from IPB University, Indonesia, on formulation of nanogel boswellic acid and its antibacterial activities against infection causing bacteria and
Yes, I am.
Yes, you can start sharing the screen, present.
Okay. Wait a minute. Can you see my PowerPoint?
Yes. The time is 5 minutes. You can proceed.
Okay. Okay, now I will present the result of our research entitled Formulation of Nanogel Boswellic Acid and its Antibacterial Activities Against Infection-Causing Bacteria in vulgaris. Introduction. Acne vulgaris is a chronic inflammation of the pilosebaceous follicle with a multi-factor cause. Pratama et al. 2017 said that 100% of respondents had experienced acne vulgaris, where 20.2% started occurring at the age of 12 and 19.2% at the age of 15. This disease attacked the face, shoulder, the upper part of superior limb, chest, and back. Negative impact of this disease are depression and anxiety, lack of confidence, stress, and up to planning suicide. There are many causes of the acne vulgaris, but pathologically caused by keratinocyte or hyperproliferation, sebocyte hyperproliferation, inflammation and immune response, and pathogenic microbial infection. And this research is focused on the pathogenic microbial infection. The main bacteria causing infection of acne vulgaris are Propionibacterium acne, Staphylococcus aureus, and Staphylococcus epidermidis. Clinical manifestation of this disease are non-inflammation lesion, just like whitehead and blackhead comedones, and inflammatory lesion, just like papule, pustule, nodule, or case. Infection of bacteria in the acne vulgaris can be treated using antibiotic, but overuse the antibiotic can reduce the overuse of the antibiotic can cause of side effects just like irritation, skin redness, itching, immune hypersensitivity, and bacterial resistance. Alternative method that can be used to reduce the side effect is herbal medicine treatment with boswellic acid. Boswellic acid is the sap of Boswellia serrata. Boswellic acid, based on the several studies, has antibacterial properties against Propionibacterium acne, Staphylococcus aureus, and Staphylococcus epidermidis. But in the research of Saptut et al. 2018, said that the essential oil of the Boswellia serrata have better antibacterial ability than boswellic acid. Changing the particle size of changing the particle size of boswellic acid to be nano is the best way to increase the ability of boswellic acid compound. Imanto et al. 2019 found that the smaller the particle size, the better the treatment effectiveness. Drug particle in nano size make the process of solubility, absorption, and distribution gets better. This research will make boswellic acid will made to be gel. Gel is chosen because of gel is the most widely used to treat acne vulgaris disease. The advantage of gel preparation are do not contain fat, easy to remove, spread quickly, moisturize the skin, not sticky, transparent, and stable. This research aimed to find the best formulation for nanogel boswellic acid and to analyze the antibacterial activities of nanoboswellic acid against infection-causing bacteria in acne vulgaris. Result and discussion. This is the formulation of nanogel boswellic acid. We have 3 formula, and the differences of each formula in the amount of nanoboswellic acid used. Formula 1 used 9 milli, Formula 2 used 18 milli, and Formula 3 used 27 milli. This is the result of qualification test of nanogel boswellic acid. In the first parameter are organoleptic with subparameter color, shape, and smell. Swastika et al. 2013 state that the good gel have a semi-solid shape, clear in color, and doesn't smell rancid. Based on the result, we can see that in the subparameter, in the color parameter, Formula 1, Formula 2, and Formula 3 doesn't meet the criteria as a good gel. But because of Formula 1 have faded chocolate color. Formula 2 have chocolate color, and Formula 3 have dark chocolate color. But in the gel base, the color is meet the good criteria as a good gel because the color is slim or clear. The next parameter is spreadability, dry power, homogeneity, pH, and viscosity. And this result is compared to the commercial gel criteria. In the commercial gel criteria have to spreadability 5 until 7 centimeter, dry power 10 until 20 seconds, homogeneous, pH 4.2 until 6.2, and viscosity 2000 until 4000 CPa. Based on the result, we can see that spreadability and dry power parameter doesn't meet the criteria as with commercial gel. But in the parameter of homogeneity, pH, and viscosity are met the commercial gel criteria. This is the result of antibacterial activity of nanogel boswellic acid. Based on the result, we can see that the best on the highest inhibition zone diameter was found in the formula 3 of nanogel boswellic acid, and the lowest antibacterial or inhibition zone diameter was found in the gel base. The results that all the formula of nanogel boswellic acid have better antibacterial activity compared to the control. Control is commercial anti-acne gel and gel base. Nanoparticles have low surface energy and surface tension, make it easier for particles to penetrate the bacterial cell membrane. In the treatment, we can see that the best inhibition zone diameter, the highest, was found in the Formula 3, and the lowest was found in the Formula 1. This is because of the Formula 3 have concentration of boswellic acid in the Formula 3 is higher than Formula 1. The higher the concentration of boswellic acid, the higher the diameter of the resulting inhibition zone. This is the mechanism of antibacterial of boswellic acid. The first one is disruption of cell membrane, modulation of efflux pumps, inhibition of oxygen uptake, alteration in oxidative.
Damuri, it's more than 5 minutes. I request you to proceed as soon as possible to close.
Okay, this is the conclusion of my my research. All formula of nanogel boswellic acid have a good quality with result that match with criteria for a good gel preparation. All formula of nanogel boswellic acid have better ability to inhibit the growth of P. acne, S. aureus, and S. epidermidis than commercial anti-acne gels. And Formula 3 of nanogel boswellic acid is the best formulation as an alternative drug to topical acne vulgaris gel formulation. That is my presentation. Thank you so much.
Thank you so much, Mr. Devi, for your valuable presentation. The next participant is Anita Thomas from Government Medical College, India, on topic of co-crystallization and emerging technique for physiochemical property modulation of pharmaceuticals. Ms. Anita Thomas. Ms. Anita Thomas, can you hear? Okay. I would like to proceed with the next participant, presentation by Roshni Singh from Jivaji University, India, on a topic of a review microsphere. Mr. Roshni Singh. Mr. Roshni Singh, can you hear? So I would like to call the next presenter, Mr. Kamala Kannan, D from JKK Nataraja College of Pharmacy. Okay, prior to that, I would like to call Mr. Bandhu. Mr. Bandhu Chatale.
Yes, sir.
Can you please share the screen and proceed?
Yes. Thank you, sir. Just I am sharing the screen.
Yes, please make it fast, and the time is 5 minutes. You can start.
Okay, sir. Okay, my screen is visible.
Yes, you can proceed.
Okay. Myself, Dr. Bandu Chatale from Met Institute of Pharmacy, Mumbai, India. I am going to present my research work, which is entitled as Synthesis of Product to Mass Bitter Taste and its Taste Evolution. Okay. So in this, my flow of content, introduction, literature survey, objective of research work, and finally conclusion. So my research is mainly taste masking of bitter drugs. So for that purpose, we will understand what is taste. Okay, so taste is the sensation produced when substance in the mouth react chemically with taste receptor cell located on taste bud in the oral cavity, mostly on the tongue. Okay, so there are different tastes: sweet, salty, sour, bitter, and umami. In that, we have mainly targeted bitter taste. Okay, so to design product and synthesis, we did brief literature review. In that, we found that numerous pharmaceutically important drugs belong to different therapeutic class are bitter. Okay, to unpleasant taste of most of the drug is masked by physical method. Okay, so but we have planned to mask the taste by chemical method, because by chemical means there is a more effective masking of taste. And that's why there is reason behind is that which alter the ability of drug to interact with taste receptor. This is logic. For that purpose, there are mainly two group or three. Mainly there are different functionality like hydroxy group, amino group. They are interact with taste receptor, and because of there is a bitter taste. Okay, so we have planned to make ester of that functionality so that we can effectively reduce the bitterness. So for that purpose, there are one important thing to design product, that we require to mask that functionality without modifying essential pharmacophoric feature required for therapeutic activity. That is important thing. So for that purpose, in literature we found that there are two type of functionality masks for bitterness, that is aliphatic and aliphatic function OH group type or amino group type, and another type, second type, that is the benzylic type. So from taking that literature in mind, there are so many drug in literature like naloxone piolate as a product for taste masking, naloxone piolate. So such type of drug is designed by piolate, making piolate ester. Okay, so such type of ester is we plan to design. So for this purpose, we selected one important drug from non-steroid anti-inflammatory drug class, that is meloxicam. So it is mainly used for management of arthritis and pain. Okay, so for this meloxicam, there are different formulation that are designed by physical method, that like oral dissolving film, oral disintegrating tablet. Okay, but there is no any chemical method. So for we decided to method. Okay, we know that market, some are to reduce the its our drug is for reduce the bitterness. So for this purpose, we design, make scheme, we make reaction. In this reaction, we did this reaction by two methods. First method, we did, we dissolve this taken meloxicam drug, and in method first, in 10% sodium hydroxide solution, using phase transfer catalyst like tetrabutyl ammonium bromide in MDC solvent at 0 degree. We did this reaction, we get product, but there is a yield that is less yield, that is only 75% yield. Then we go for another method. So in that method, we use triethylamine as a base, and we use anhydrous THF solvent, and we carried out this reaction at 0 degree, and we get very quite good yield. Then after that, we did its characterization by FTIR. So we did FTIR for meloxicam as well as Easter drug. So by we get that there is a disappearance of hydroxy group in that characterization, and there is also we get ester peak, carbonyl ester. From that we confirm that it is our ester is synthesized. Then we also done NMR for this. Okay, so from NMR, just it is next slide. Okay, my
So
voice
although
is
it's
clear
more
till
than
right?
5 minutes, I request you to finish as early as possible.
Okay, just give me 3 minutes. Okay, thank you. Then okay, my slide is not going ahead. Just a second. Okay, then we did NMR. Then after that, we did its HPLC also for purity characterization, and then we did its test evolution by rat palatability test. And in that, we found that as compared to meloxicam, there is quite improvement in palatability. So we did this rat palatability test for test evolution purpose. Okay, so in this way we found that there is a very drastic improvement in the palatability of this meloxicam privilege as compared to meloxicam. So in this way we can from Easter successfully reduce the bitterness of the drug. Thank you.
Thank you so much, Mr. Bandhu, for your valuable presentation.
Okay, thank you, sir.
And I'd like to call Mr. Kamalakannan D from JKK, Nataraja College of Pharmacy, India, on a novel method development, validation, and study of force degradation behavior on prazosin and polythiazide in combined pharmaceutical dosage form by UPLC method. Mr. Kamalakannan D. Mr. Kamalakannan, can you hear? We would like to call the next participant, Ms. Padalavani Sujata from Mallareddy Institute of Pharmaceutical Sciences, India, on drug utilization and evaluation of anticoagulants in tertiary care hospital. Ms. Padalavani Sujata. Sujata. Sujata, can you hear? Okay, I would like to call the next presenter, Ms. Hilal Basak Erol from Ankara University, Turkey, on susceptibility of ESBL positive E. coli strains to commercially available and laboratory isolated bacteriophage. Mr. Hilal Basak Erol. Mr. Hilal. Mr. Kamalakannan. Mr. Kamalakannan, can you hear?
Sir, I'm audible, sir?
Yes, you are audible now. Please start this, sharing the screen and proceed.
Shall I start presentation, sir?
Yes, you can start sharing the screen and proceed, Mr. Kamalakannan.
Just a minute, sir.
Your voice is not audible. Your network is not stable. No, Mr. Kamalakannan, I request you to be in a network place and kindly proceed later. So we will go with the next presenter, Dr. Divya Dilip Kumar, MDS, SRM India, on top evaluation of efficacy of Pudina extract on Candida albicans and Lactobacillus acidophilus, an in vitro study. Dr. Divya Dilip Kumar. Dr. Divya Dilip Kumar, can you hear? Seems the presenters are not available. So I would like to call the next presenter, Mr. Shivendra Verma from SRK University, India, on topic of azithromycin and ofloxacin antimicrobial resistance of isolated from raw vegetables Listeria spp. So Shivendra Verma. So Shivendra Verma, can you hear? So I would like to call Ms. Shalini Singh from SRK University on phytochemical constituents, antimicrobial activity of Chenminelia cebula fruit extracts of against E. coli and AM Pseudomonas pathogens. Ms. Shalini Singh,
Yes.
can you start sharing the screen and proceed? Ms. Shalini Singh, can you please share the screen?
My topic is phytochemical constitution and antimicrobial activity of Terminalia cibula, fruit extracts of against E. coli and Pseudomonas pathogens. Department of Microbiology of SRK University, Bhopal. Urinary tract infection. UTI are serious health problem and its second most common types of infection in the body, affecting millions of people each year. Infection of any part of the urinary system, including kidneys, ureterus, bladder, and urethra. Acute kidney injury. Bacteria continues to cascade up to the kidney, leading to acute kidney injury. Phytic infection of renal parenchyma, cause of inflammatory response called phyllopancreas association. Ureterus. Bacteria starts towards the kidney via the ureterus. Bladder. Urophytelium pathogen pancreas, bladder and bacteria replicates parenchyma and biofilms. Urethra. Pathogen colonize the urethra and escapes towards the bladder. Symptoms vary between different types of UTIs. Some of the commonly noted symptoms include burning sensations during urination, frequent urinate, but incomplete voiding, pain or pressure in the back of lower abdominal, pain, pelvic pain, blood and urine, cloudy dark stains of strong smelling urine mixed with the blood in some cases, tiredness, fever and chills, nausea and vomiting, vaginismus. UTI by E. coli and Pseudomonas. E. coli, or better known as E. coli, is the bacterium responsible for over 80% of common UTI cases. Other bacteria can also cause UTI, and they include Pseudomonas, Staphylococcus, Streptococcus, and Klebsiella pneumoniae. Coli 63% and 5% Pseudomonas. What are phytochemicals? Phytochemical free chemicals of plants, phytochemicals of phytonutrients and the biological activity, natural occurrence substance in plants that have protective and disease preventing properties. They act of natural defense mechanism in their host plants. They are also provided pigments to the plants. They are not essential nutrients, meaning that they are not required by the human body for sustaining life. So, for scientific knowledge of 3,000 different phytochemicals with possible health benefits.
Phytochemicals and Terminalia cibola. Terminalia cibola is called of the king of medicine in the Tibet and is one of the prime Ayurvedic herbs from India because of its extraordinary healing powder. Terminalia cibola for daily health vitamins. In addition, it is the digestive benefit. The fruit is also good source of vitamin C and the minerals selenium, potassium, magnesium, iron, and copper. Terminalia cibola fruit is also considered to have antibacterial and anti-inflammatory properties. At the same time, it is known to promote tissues growth and health. This plant in the management of the sore throat, high cough, asthma, ulcer, heartburn, vomiting, diarrhea, dysentery, and bleeding, piles, and bladder disease. Our study bacteria collect UTI positive urine sample pathology from the Bhopal MP People's Hospital, isolate and observation sample, identify two bacteria E. coli and Pseudomonas for further work. Plant collected Terminalia cibola fruit, make powder and salt in 4 degrees Celsius refrigerator for further chemical screening of Terminalia cibola alcohol test for cobalt, chloride, burate test protein, then steroid sulfate test, anti-quinone glycosides, alkali test, Mayer and Wagner test, then positive ferric chloride, chemical acetic acid, mercuric chloride, potassium iodine, mercuric nitric acid, alpha methyl butanol, hydrochloric acid, tannic acid, sodium chloride, sulfuric acid, cobalt chloride, sodium hydroxide, copper sulfate, lead acid, glacial acetic acid, gelatin, ferrous sulfone, 6 mm filter, paper discs, Mueller-Hunter agar, TPC and TFC. Total phenolic content 703.119 plus minus 0.0015. Tannic acid standard curve जो absorption and concentration Rx value 0.9869. Total phenolate contains acid TFC 100 plus minus 0.0056. Rx value 0.9968. Antimicrobial activity. The disc diffusion method was used to test antimicrobial activity in the extracts against test microorganism solvent, dyed and sterilized filter paper 6 mm were then impregnated with the known amount of the last test substance using micropipette and the resistant solvent were complementary evaluated. This plant where the kept of low temperature 4 degrees Celsius for 24 hours allowed maximum diffusion. This bears gradual change of test material concentration in the media surrounding the disc. The plates were the incubated 37 degrees Celsius for 24 hours allow maximum growth of the organism. The test material having antimicrobial activity inhibited the
I'm
growth
sorry
of
to
the
interrupt you. It's
microorganism.
more than 5 minutes. I request you to finish as early as possible.
Oh, okay, sir. Antimicrobial activity in each line 25, 50, 50 compared to different concentration in best 50 concentration zone activity in E. coli, then Pseudomonas, so 25 concentration zone activity in Pseudomonas. Conclusion. In the present study, we found the most biological activity phytochemical present in the plant extracts such as protein, steroid, flavonoids, alkaloids, glycosides, and carbonate, etc. Terminalia cibola plant hydroalcoholic extract. In this research showed bacteria activity with UTI pathogens, E. coli and Pseudomonas. Plant extracts showed good antimicrobial activity against this pathogen. A large segment of population in the world relies upon traditional system of medicine. This study useful in future further herbal medicine importance in the pharmaceutical industry for the different types of medicinal use. Thank you.
Thank you so much, Ms. Shalini. I would like to call the previous presenters, Mr. Kamala Kannan, to proceed if he is available. Mr. Kamala Kannan. Hope your network is stable now. You can start sharing the screen.
Now it's okay, sir. My poster is visible, sir.
No, it's not visible.
Now, sir?
No, it is not.
No, share your screen.
Please share your screen.
Yeah, just a minute.
Yes, now your screen is visible. Please.
Yes, sir.
Open the presentation and you can start. Time is 5 minutes.
Yes, sir. Good evening to all. Today I'm going to present my topic in the analytical department. The novel method development, validation, study of force degradation, behavior on prazosin and polythiazide in combined pharmaceutical dosage form by UPLC TUV method. The main aim of this my method is to develop a novel, accurate, precise, and effective analytical methods by RPHP UPLC for the selected drugs, that is the prazosin and polythiazide combination. Here, the prazosin is an antihypertensive used for the treatment of hypertension, and the polythiazide is a diuretic drug. The main aim and objective is to develop a stability indicating assay method and to perform a validation parameters as per the ICH guidelines and to conduct a post degradation studies for a single and combined formulated drug by RPUPLC. There are n number of benefits over the HPLC when compared to the UPLC. By using the latest technique of UPLC, ultra performance liquid chromatography, the method has been developed, validated as per the ICH guidelines and the post degradation studies performed. Initially, the preparation of standard and sample solution. We have prepared both standard and sample solution in the dilution of 5 microgram per ml of polythiazide and 20 microgram per ml of prazosin. The optimized chromatography condition is the mobile phase ratios 55 orthophosphoric acid buffer and 45% of acetonitrile. The flow rate is 0.8 ml per minute, and the column we have used is BEC18 column, and the detector wavelength at 260 nanometer. And the column temperature is 19 to 30 degrees centigrade, and the injection volume is 10 ml. So hence, there are n number of trials has been conducted. The finally fixed mobile phase is 55 ratio with orthophosphoric acid and 40% of acetonitrile with the faster retention time and good linearity and degradation coefficient is very good. And the resultant discussion clearly shows that the linearity range is 5 to 30 microgram, and with, for prazosin and for polythiazide 1.25 to 7 microgram per ml. The regression coefficient we found that 0.999 for polythiazide and both prazosin. Hence, all the parameters are within the limit. And the assay we found by using the formulated drugs of 99.94% by using these above proposed methods. And the system precision, method precision, and accuracy, LOD and LOQ, robustness, all the parameters has been performed and all are shows within the limit. And finally, the conclusion, the RPUPLC method were developed and validated for the estimation of the selected drug in bulk and pharmaceutical dosage form. To check the stability indicating nature of the developed RPUPLC method, the first degradation of these drugs were carried out under the acidic, basic, oxidative, thermal, and photolytic hydrolytic conditions. Newly developed stability indicating RPUPLC method were successfully applied for the determination of the drug content from the pharmaceutical dosage form in the presence of the degradation product. From the result obtained in the first degradation studies, it may help to facilitate the pharmaceutical development, such as the formulation development, manufacturing, and packaging of the drug products, as well as in the storage of the active pharmaceutical ingredient, in which the knowledge of the chemical behavior can be used to improve a drug product. Right? This is my presentation for the topic, the combination of polythiazide and prazosin combinations. So thank you, one and all. Thank you.
Thank you so much, Mr. Kamalakannan. I'd like to call Ms. Padala Vani Sujata.
Yes, sir.
I request Kamalakannan to stop sharing his screen.
Yes, sir.
Yes, thank you so much. Please, Sujata, you can start sharing the screen present. Ms. Sujata.
Okay, sir. Yes, sir. Sure. One second. Is it visible, my screen?
Yes, it's visible. You can start sharing the screen. Time is 5 minutes.
Sure, sir. Good evening, everyone. I'm Vani, here to present my research work on anticoagulant utilization evaluation in healthcare center under the guidance of Dr. Shubham Babu Gupta. Anticoagulant drugs have narrow therapeutic range. Its utilization is very utilization and its evaluation is very essential. And drug evaluation and um, anticoagulants, commonly known as blood thinners, are chemical substances that prevent coagulation of blood and prolonging clotting time. Anticoagulants are used in the therapies for both in treatment and prevention of cardiac diseases and cerebral vascular accidents and thromboembolism. And the ADRs or the adverse events related to regarding anticoagulants are seen mostly in medical and surgical units and rehabilitation and emergency room outpatient clinics. Types of errors associated with anticoagulant prescriptions are of dose omission, underdose, extra dose, and prescription refill delay, wrong dose, etc. And the aims of and objectives aim of the research work is to monitor drug utilization pattern of anticoagulants, identify and assess drug interactions and its management. Objectives to observe prescribing patterns of anticoagulants and to monitor the incidence of ADRs. And the study methodology, this is an observational descriptive study, and this is to carry out the evaluation of prescribing or administering patterns of anticoagulants. And the study period is of 6 months, like from Jan to June, and the sample size of patients 350 to 380 were taken for the significant outcome. And the inclusion criteria were patients aged from 18 to 70 years under the department's cardiology, dialysis, orthopedics, and the ICU and medicine unit. And exclusion criteria patients not under anticoagulant therapy, infants and pregnancy patients, pregnancy people. And patient a well developed pro forma was made for the case collection based on the monitoring parameters. And the results here shown are like the risk of thromboembolism is if the risk is under 10%, there is no specific or the other option or the alternative therapy. If the risk and as due to the risk is between 10 to 40%, and the patients are given with a low molecular heparin. Yes.
You can proceed. The time is going up. You can proceed.
Sure, sir. Okay. And the alternative therapy was given enoxaparin as well as the fondaxaparin. And the patients with high risk, like 40 to 80%, were actually given for other oral vitamin K antagonist, which and the control of INR was done. And the reasons for the hospital admission is due to acute coronary syndrome and coronary artery angiography and congestive heart failure and myocardial infarction as well. And the other risks are DVT and VTE, DVT deep vein thrombosis and venous thromboembolism. As the graph is shown, it was due to as you can see the graph, the major cases were of acute coronary syndrome as well as coronary artery angiography. And the therapeutic goals based on the indication of anticoagulants are in the prophylactic condition, the heparin was given, and in the therapeutic condition, mostly the warfarin combination with enoxaparin was used. And areas associated with heparin were majorly bleeding and minor and in the therapeutic as well as in the therapeutic and the prophylactic, bleeding was a major cause. And ideas associated with enoxaparin are bleeding is not there in the prophylactic, but in the therapeutic, if the enoxaparin is given, the major is bleeding as well as the allergic condition. ADR associated, and ADR associated with warfarin is majorly bleeding and as well as GI distress. And The conclusion of this is, despite the existence of comprehensive guidelines for prevention and treatment of venous thromboembolism, it is not performed accurately, and the lack of acquaintance of the prescribers with the guidelines may be of a major potential risk for not administering anticoagulants properly in the hospital. And furthermore, for every general hospital, it is recommended to have local thromboprophylaxis strategy in a form of written form as like thromboprophylaxis protocol. Therefore, arranging a meeting for preparing such an agreed protocol with describing the proper implementation and the potential impact on the system with the relevant healthcare professionals. Thank you.
Thank you so much, Ms. Sujata, for your presentation. And I would like to call the next presenter, Mr. Hilal Basak. Mr. Hilal Basak, can you please start sharing the screen? Ms. Sujata, ma'am, can you please stop sharing the screen? Thank you. Mr. Hilal Basak.
Yes, I'm here.
From Turkey. Mr. Hilal, can you hear?
Yes, I will start the document. Yeah.
Yes, thank you, ma'am. You can start sharing the screen. The time is exact 5 minutes. You can proceed.
Do you show my presentation?
No, it's not visible.
Share your screen.
Yes, it's visible.
No, it is not. Please share your screen.
Yes. Share again.
Yes, now it is shared. You can start.
It's visible?
Yes.
It's visible?
Yes, ma'am. You can proceed.
Okay. My topic name is suspected for ESBL positive E. coli strain is commercially available and laboratory isolated bacteriophage. And E. coli, the key problem of antibiotic usage is resistance increase. The most serious pathogen is E. coli within the gram-negative bacteria. As an alternative to antimicrobials, as a promising tool, demand for bacteriophage therapy has increased significantly over the years. Bacteriophage entered the literature as a bacteria eating viruses in early 19th century. Bacteriophages are defined as bacterial viruses, and they offer a selective effect as they can kill their specific target bacteria. Bacteriophages can be found wherever bacteria are found. It's estimated that their approximate number in nature is 10 to 32. Bacteriophage consists of heat, neck, tail, and tail fibers. The heat part has the form of icosahedron containing nucleic acid surrounded by a protein sheet called capsid. Nucleic acid can consist of double or single-strand DNA or RNA. And they have some areas of usage, identification and typing of bacteria, using phage as vectors in recombinant DNA technology, vector in working preparation in order to extend the life of food, disinfection of farming, and especially they can use for phage therapy. Bacteriophage show specific effect. They have the advantage of reproduction at the site of infection and therefore being available where it's needed most. They don't cause serious side effects. Phage resistance bacteria may be sensitive to other phage with a similar target range. And selection of new phage is a relatively fast process and can often be carried out within days or weeks. The objective of this work was to isolate and characterize potential therapeutic phage active against E. coli and compare the efficacy with commercial phage preparation in test bacteriophage on ESBL positive E. coli. And our methods. 9 E. coli strains were used for phage isolation, and additionally 50 clinical ESBL E. coli isolates from the Chicago hospital and 8 laboratory isolates were used for host range analysis. All bacteria strains were routinely cultured in military agar at agar. And intestinal bacteriophage was brought from George Eliava Institute of Pharmacy in Georgia. It's a mix of sterile filtratophage lizard, active through life. Our study took place in 3 stages to obtain the ESP positive strain, taking a 1 water sample and phage isolation. For ESP E. coli strains were used for as an identical host for phage isolation, and 5 water samples were collected from river in the different regions of Ankara. Phage enrichment was performed first phage isolation, and spot test method was applied to determine the possible presence of phage. To confirm the presence of lytic phage in the filtratable layer agar method was applied to spot test positive sample, and single phage isolation was made for purification in petri dishes with phage plates. And we determined the titer of phage and the first suspected because of the suspected of the harvested phage was determined using a spot test. 50 clinical E. coli strain and 8 laboratory strain were used for this analysis, and all strain was spread evenly on the LB agar plate. After drying, 10 microliter of phage filtrates were dropped on the overlay top agar, and intestinal bacteriophage also added on the layer of agar. Therefore, the exact after the cultured, the presence or absence of lysosome was observed, and each experiment was performed in 2 days. And our results. A total of 4 E. coli were used to find lytic phage from the collection of water and from the river water. Among all 9 filtrate produced clear zone against the host bacteria. The isolated phage produced visible plaque on the bacterial loam in the range of 1 and 3 millimeter diameter in the initial sequencing using plaque assay. And the susceptibility intestinal bacteriophage and newly isolated phage were determined against.
Ms. Hilal, it's more than 5 minutes. I request you to finish as early as possible.
It is the positive, the lysosome is in here. And if there's a lysosome, there is a positive. There is
it's oral presentation. You can take 2-3 minutes more, no problem.
Yeah, okay. The results of activity revealed the lytic spectra ranging from 20 to 97%. Overall strain was susceptible to 1 or more phage from the panel. It was proved the intestinal is very effective in a wide variety of strain of E. coli. Input test strain also show that the newly isolated E6 phage is effective as a commercial phage, and as overall strain was susceptible to 1 and or more phage from the panel. It was proved the intestinal phage is very effective in a wide variety of strain of E. coli, including the test strain. Also showed that the newly isolated E. coli phage is effective as commercial phage. And these are results of bacteriophage susceptibility of E. coli strain. And thanks for listening.
Thank you so much, Ms. Hilal, for your valuable presentation. The next participant is Mr. Shivendra Verma. Mr. Shivendra Verma, can you please, Ms. Hilal, you can stop sharing your screen. Mr. Shivendra Verma, can you hear?
I think he is not there. We can proceed for next.
Just a minute, sir. He's sharing the screen now.
Okay, then okay.
Mr. Shivendra, please release your mute.
Hello.
Yes, you can start sharing the screen and please proceed.
Just 1 second, sir. Can you listen me, sir, properly?
Yes.
You are audible. You are audible. Share your screen and start presenting.
Just 1 second, sir. Some technical problem. So, sir. Hello. Visible my side?
No. Yeah,
Your screen is shared, Mr. Shivendra. Please open the presentation and proceed.
Just sir, 1 second. Sorry. Please, sir, invite to other candidates. So I will. Hello.
Do you want me to proceed with the next candidate?
No, sir. Okay.
Just open presentation. Click on the presentation. Yeah.
Yes, sir. Done. Have you visible, sir, my side?
No, not yet open.
Presentation is not open. Still, it is opening. Trying to open.
Please, sir, invite to other candidate. So
Okay, please rectify your problem. I'll call the next candidates.
So stop sharing.
I request you to stop sharing your screen so that we can call the next participant. So I would like to call the next participant, Mr. Simachal Panda from Lincoln University College, Malaysia.
Hello.
On the topic of research on enhancement of solubility. Yes, Mr. Shivendra Verma.
Yes, sir. Okay. It's visible, sir?
No, it's a blank presentation.
No, 1 second. Just is loading, sir. Okay, sir.
Yes.
Can you visible, sir?
Yes, it is visible now. Time is too short. Please, I request you to finish as early as possible within 5 minutes. Proceed.
Okay, 1 second, sir. Okay, sir. Myself, Shivan Verma, sir. I'm PhD scholar from Bhopal, SRK University. It's my hello.
Yeah. Continue.
Yeah. Okay. My works are presented especially foodborne listeriosis and foodborne disease because the foodborne pathogen listeriosis can food poisoning cause Listeria monocytes, which is 30% rate as mortality rate patient worldwide. And generally, Listeria monocytis is the main cause of the foodborne pathogen. It is a gram-positive bacteria, soil, water, and some animals, including its incubation period 3 to 17 days duration in days of week to week. Then it's our aims and objective of this study is a collected raw vegetable different district of Madhya Pradesh, isolated sample using ISO standard method 11292 method and identify Listeria species and then isolated pathogens antimicrobial activity of with using standard antibiotic azithromycin and ofloxacin. We have all collected 312 samples of raw vegetables distributed 10 types in MP district, especially cabbage, tomato, potato, spinach, regular etc. different types, and it's more than 10 districts choosing MP for sampling. The microbial analysis of presentation Listeria monocytes and all Listeria species of fresh vegetable sample were done by standard method and this pre-enrichment with talcum using and DHI agar. Then we have observed our results in general observation of 147 sample positive out of 312 sample, around 47.11 vegetable sample were positive Listeria species. Then phytochemicals test such as hemolysis, catalysis, oxide. We found 4 types, especially Listeria species: Listeria monocytogenes, Listeria innovi, Listeria siligere, and Listeria nucleus. One suspected Listeria species then is Listeria species. That is some pictures of isolated Listeria agar plates, and that is a microscopic Listeria pic. For antimicrobial method using for disk diffusion method for step-by-step and this is my antimicrobial Patrick plates for azithromycin and ofloxacin. It is our antimicrobial zone inhibition for Listeria monocytogenes, Listeria inocual, Listeria siligeri, and other Listeria species. That is the results mentioned show Listeria monocytogenes. That is Listeria, ofloxacin is a much better than result than compared to azithromycin. In this our study we have found five Listeria species, and then after this diffusion method antimicrobial activity ofloxacin is much better than compared to azithromycin. It is good for Listeria disease for foodborne around the world and medical field. It is final, sir. Thank you. Hello.
Thank you so much, Mr. Shivendra Verma, for your valuable presentation.
Thank you, and sorry for late, sir.
No problem. You can.
Okay.
You have,
remove your slides. So
okay, sir. Thank you so much, all of you.
Thank you so much. You can proceed by stopping the sharing. And I want to proceed with the next participant, Mr. Simachal Panda from Lincoln University College, Malaysia. Mr. Simachal Panda, can you hear? Are you available, Mr. Simachal Panda? Seems he's not available. We'll go to the next presenter, Mr. Parkavi M. from JKK Nataraja College of Pharmacy, India, in silico studies, synthesis and characterization of 4-methyl 9-to-10 hydro 2H, 8H, chemeno 8-to-7, oxazin 1. Ms. Parkavi M.
Are you present, Parkavi M?
Ms. Parkavi M, are you present?
He is also not there, I think.
Yes, sir. We'll go to the next presenter, Mr. Alifia Yusuf Lahari from Prim KM Kundani College of Pharmacy, India, on topic of Shilajit, a Himalayan herbo-mineral wonder. Ms. Alifia. Are you present, Ms. Alifia? Okay, we will go with the next presenter, Mr. Ishtiaq Jilani from University of Toyama, Japan, on total synthesis of biologically active natural products with 6H dibenzo 621 using palladium catalyzed Beryl coupling reactions. Mr. Ishtiaq Jilani.
Yes, he is there.
Yes.
He is online.
Mr. Ishtiaq Jilani, can you please respond? I request Mr. Ishtiaq Jilani to respond. Okay, I would like to call the next participant, Dr. Sudha from Lincoln University, Malaysia, on pharmaceutical synthesis of pyrazopyrimidine derivatives along with its biological activity. Dr. Sudha.
Dr. Sudha, please. Are you able to hear us?
Dr. Sudha, ma'am, can you please respond?
Yes, is there.
I request you to unmute Dr. Sudha, ma'am. Dr. Sudha, can you please respond? Dr. Sudha, can you please respond? Ma'am, can you hear? Madam Dr. Sudha, ma'am.
Dr. Sudha, will you be able to share your screen? Yeah.
Can you hear Ms. Sudha? Can you please respond?
Good evening, sir. Hello, good evening, sir.
Ms. Sudha, can you please respond? I request you to talk.
Presentation is open. I think voice is not coming.
Yes, sir. She's in unmute only, but still she's not responding at all.
Yeah. Let us see. Dr. Sudha, start presenting. We are shortage of time, running with shortage of time.
I would like to call Miss Alefia Lahari. Ma'am, can you present?
Yes, sir, I can. Just
Yes.
give me a second and I will try and share.
I request you to share your screen and proceed.
Yes, sir. Just give me a second, please.
Yes.
I apologize for not being able to present earlier.
First, you can share the screen, ma'am. On the middle of your screen, you can see an option called share screen with the green color option.
Yes, I am on that, but it says that Zoom requires me to sign in with my email again.
Absolutely. You have to go through with your email, then only your Zoom will open. I mean, you can go through with the link and you can proceed sharing the
I am
screen.
with the link, sir, but
Once again, you may log in. You go out and log in. Meantime,
Yes.
we can go for next.
Ma'am, you please disconnect and relog in. Rejoin again.
okay, sir, I will definitely do that.
Yes, we will call you the next participant. So I would like to call Mr. Ishtiaq Jilani. Mr. Ishtiaq Jilani, can you hear? Mr. Ishtiaq Jilani, can you please unmute and respond? Okay, he's not responding, so we'll go with Dr. Sudha. And still, she is also not available.
We can invite openly those who have not presented and with us, they can share.
So I can is it all right if I try again?
Yeah, your voice is coming.
Okay.
I'm talking to Miss. May I know your name, please?
Yes, this is Alifia.
Yeah.
Okay, I request you to please
share,
share.
madam.
Does this show? It says
No,
the
it is
file
not,
will be
ma'am.
shared.
No, it's not. Your screen was not shared.
Does
Yes, now it is trying to share. Just a minute.
Is my
Yes, ma'am.
screen
It's shared
visible,
now.
sir?
Yes, you can proceed.
So, my my
Yes, you can
research
proceed.
is basically on Shilajit. Traditional systems of medicine have been important for centuries, and they can still be used as a supplement to modern medicine. The drug that formed the basis of my research is Shilajit, which is a herbomineral drug that is abundantly found in mountainous regions in various parts of the world. Shilajit shows a wide range of pharmacological activity, right from antioxidant and anti-inflammatory activity to cardioprotective, hepatoprotective activity, anti-cancer activity, anti-diabetic activity, and the list just goes on. But the main focus in this research was lying on its anti-cancer profile, and an attempt was made to increase the bioavailability of Shilajit by formulating it as a phytosome or or a phytophospholipid complex, as this would decrease the dose of the drug required while increasing its bioavailability. So by reducing the dose, we're also conserving priceless mineral resources. Now, Shilajit was also standardized, which is difficult considering it's an unorganized drug. So a method was developed for the HPLC and HPTLC analysis. And as part of pre-formulation, even UV spectroscopy and FTIR analysis were performed. And Shilajit-loaded phytosomes were prepared through the solvent injection technique using a predetermined drug-lipid ratio. After formulation, when the phytosomes were evaluated, they were evaluated for various characterizations such as their percent entrapment and parameters such as particle size analysis and zeta potential. With the in vitro release studies and an SRB assay was performed to evaluate its anti-cancer activity. And I've included the results of my study. The percent entrapment was found to be 62%. And the percent released by diffusion method was found to be 73%. The particle size and zeta potential were also found to be within an acceptable range. And the results from the SRB assay suggest that the phytosome showed optimum activity against MCF7 cancer cell lines. This is followed closely by the extract. As you can see, the IC50 values for the phytosome is 8.39 micrograms, whereas it is 82.23 microgram per ml for the extract. So you can see that when you're comparing the phytosome and the extract, the phytosome is superior in terms of its activity. Shilajit, being a safe and natural drug, can be used to treat cancer patients. Shilajit is an adaptogen and it's a rejuvenator and can be used to promote longevity. If we combine a traditional drug such as Shilajit with modern formulation techniques, it will help us unfold avenues of cancer treatment. And that is what my entire focus was on. That is all. Thank you, sir.
Thank you so much, Ms. Alifia, for your valuable presentation. I would like to call the next participant, Dr. Sudha, to share her screen and proceed with the presentation. Ms. Sudha, ma'am. Dr. Sudha, ma'am, can you hear? It's in mute, but it's not audible. Your voice is not audible to us. I request you to use a microphone or increase the volume. Dr. Sudha. Dr. Sudha, can you hear us? Ma'am, your voice is not audible, ma'am. We cannot hear you. Sorry, ma'am, please kindly check the settings from your side. And I would love to call next presenter, Sonali Bhagat from Arid Forest Research Institute, India, on
Yes, sir.
phytochemical investigation and quantitative estimation of some flavonoids in the fruits of Capparis decuada, Edge, from Arid region of Rajasthan, India. Ms. Sonali,
Yes, sir. Am I
kindly share
audible?
the screen. Yes, you are audible.
You
Kindly
are
share the screen.
audible. Share your screen.
Sir, is the screen visible?
Screen is visible. Please open your presentation and proceed.
Click on your presentation, open your slide,
Yes, sir.
then, yeah.
Yes, sir. Good evening, everyone. Today I'm going to give my presentation on the topic phytochemical investigation and quantitative estimation of some flavonoids in the fruits of Capparis decidua force edu from arid region of Rajasthan, India. First of all, introduction of Capparis decidua.
Ma'am,
It is
your
an
presentation is not shared yet. We cannot see your presentation.
it is showing that you are screen sharing.
Yes, the screen is shared, ma'am. It's showing your folder, but it's not, your presentation is not visible for us.
Actually, presentation is not open yet.
We can see your folder. If you are on the presentation, please click the presentation on the folder and you open it.
Sir, I have already opened it.
I'm sorry, ma'am, but it's not visible for us. Professor, sir, can you see? Subhash Chandra, sir, can you see?
No, sir, it is not
No,
visible.
it's not
It's not
visible, sir.
It's not visible, ma'am. Please open it.
Just she has to click. I think she is not identifying this. She has to click on the PPT.
Yes, sir, I have already clicked and the PPT is open in front of my screen.
Once again, close and open otherwise.
Okay, sir, I'm trying.
Share screen, through share screen, you open. First open, then share screen, then click on your presentation. Yeah, click. Yes, click on that.
Is the presentation visible now?
Yeah, presentation, click on that. Let it be open.
Right, it is actually open in front of my screen. I don't know what is the technicality.
That means you have not shared, I think. You didn't share it. You didn't share it, that's why.
It seems there's a problem with your presentation.
You once again go out and again log in otherwise. Yeah, it's coming.
Is it coming now?
Yeah, now visible.
Yes, sir. Shall I start?
Yeah.
Okay. Today, I'm going to present on the topic phytochemical investigation and quantitative estimation of some flavonoids in the fruits of Capparis decidua force edu from the arid region of Rajasthan, India. I'm a research scholar from Arid Forest Research Institute, Jodhpur, Rajasthan. First of all, the introduction of Capparis decidua. It is an important arid zone shrub, and it is basically used as folk and herbal medicinal plant since ancient times. It has variety of edible uses, such as it is used as a vegetable preparation in Panchkota. It is a delicacy of Rajasthan, and it is also used to make pickles. Several phytochemical studies have also have already been carried out, like phenols, flavonoids, sterols, glycosides, etc. They have been determined in different extracts of fruits, stems, flowers of Capillus residua. The flowers, fruits, stem, and seeds, they contain N-pentacosane, N-tricontain, ascorbic acid, proteins, carbohydrates, and other minerals. Now, the medicinal importance of Capillus decidua, the various reported medicinal properties it has, like it is used to cure asthma, cough, toothache, malaria, inflammation, fevers, rheumatism, etc. And these are the medicinal properties which are found in its stem, roots, and leaves. And anti-arthritic, similarly anti-arthritic potential, and it is also found in the stem, roots, and leaves. And going to the fruits have anti-hemolytic effect, anti-diabetic, hypolipidemic, anti-atherosclerotic, antibacterial, and antifungal, antioxidant, anti-helminthic. Now my experimental section went to like this. I collected the fruits and then shade dried and then powdered it, and then they were exhaustively extracted in methanol using exoxylate apparatus, and then went to the qualitative analysis part using the standard qualitative methods to test the phytochemicals. That is phytochemical screening, and then antioxidant activity using the DBPH free radical scavenging method. This was used, and then determined the total phenol content and total flavonoid content. And then finally, the quantitative analysis using reverse phase HPLC. Now, phytochemical screening was performed. Following standard qualitative tests were done for confirming the presence of secondary metabolites. These secondary metabolites like steroids, tannins, alkaloids, flavonoids, and phenols, they showed their presence in my fruit extract. Now, estimation of antioxidant activity using DPPH free radical scavenging activity. The present study, it was determined it determined the antioxidant properties of Capillus decidua fruits, as well as the composition of phenolics and flavonoids that may contribute to the antioxidant activity. That's why after antioxidant activity, I moved to the estimation of phenolics and flavonoids. And the values of IC50 inhibition concentration, the IC50 concentration value in micrograms per ml was found to be 67.5 plus minus the standard deviation 3.43, and this result shows that these fruits are quite rich in their antioxidant activity. The quantitative estimation for finding out the total phenol content, it was estimated in this extract using the method used by Donald et al., Folinsio-Calci method, and it was depicted in terms of milligram gallic acid equivalent per gram on the dry weight basis. Standard curve was prepared by making the following concentrations of gallic acid standard, which of high purity, that is more than 95% pure, and taking the absorption readings as at 750 nanometers, and the regression obtained was more than 99%. Quantitative estimation of total flavonoid content. It was done using a method given by Olajire et al. 2011, aluminum chloride colorimetric method, and it was depicted in terms of milligram quercetin per gram on dry weight basis. And the standard curve was prepared by making concentrations of high purity quercetin standard, and the absorption readings were taken at 510 nanometers, and the regression obtained was greater than 99%. Now, the values for total phenol content and the total flavonoid content in the fruit extract were like these: that 38.25% total phenolic content and total flavonoid content to 18.58 milligrams quercetin equivalent per gram. Now, the HPLC was used for quantitative estimation. Hello? Hello? Sir, am I audible?
Yes, you are audible. Ms. Sonali, you can proceed.
Yes, yes. Quantitative estimation using the reverse phase HPLC was done. First of all, this method was standardized. The binary grade HPLC system of make waters was used, and the reverse phase column sphericol was used for this analysis work. Solvent system used was methanol and orthophosphoric acid plus TEA plus water for a total runtime of 45 minutes. The retention time obtained of rutin was at 24 minutes, and retention time of quercetin was found at 36 minutes. Quantitative estimation by using the peak area measurement value was done, and this revealed that rutin was found 0.32 ppm, and quercetin was 0.16 ppm in our study. These were the chromatograms that shows the first chromatogram shows the peaks of rutin and quercetin at 24 and 36 minutes respectively, and in my sample I obtained it for also at 24 minutes and 36 minutes. Based on this only, the rutin concentration and quercetin concentration was found. Now, on concluding, a result from our above work on antioxidant activity, phenols and flavonoids opens up a way for further phytochemical studies on these fruits that can be beneficial to the pharmaceutical industry for the herbal preparations. And my further work aims at screening and determining the key bioactive principles, marker compounds present in these fruits, which are mainly responsible for their medicinal activities. Thank you.
Thank you so much, Ms. Sonali. So now I would like to call Dr. Sudha.
Yes, sir.
Yes, ma'am. Now you can start sharing the screen and present.
Okay, sir. May I start now, sir?
Yes, you can proceed.
Good evening to all of you. Today I am here to give you a presentation on the topic pharmaceutical synthesis of pharmaceutical pyrimidine derivatives under the kind guidance of Dr. Mohammad Gausuddin. Contents include introduction, aim and objective, literature survey, reaction scheme, detail of synthesized compound, analytical evaluation, pharmacological screening, result and discussion, and references. Now, introduction. Introduction includes medicinal chemistry. Medical chemistry is a discipline at the intersection of chemistry. It involves the synthesis, development, and new chemical entities suitable for therapeutical use. Now come to the main point is pyrazole-pyrimidine derivative. It is a ring of fused or condensed pyrazole or pyrimidine rings. Both are heterocyclic ring. In pyrazole, this is a 5-membered ring, and pyrimidine, these are 6-membered heterocyclic ring. They are fused together and give a formation of pyrazole-pyrimidine derivative. Now, aim and objective is to develop a new series of pyrazole-pyrimidine derivatives to evaluate derivatives for different biological activities and to find out derivatives having potent activity and lesser side effects. Now, literature review. Basgir et al. Synthesized pyrazole-pyrimidine derivatives against standard drug streptomycin. Manthi et al. Synthesized pyrazole-pyrimidine derivatives against gram-positive bacteria Staphylococcus aureus and gram-negative bacteria E. coli. Now, Rashid et al. Synthesized pyrazole-pyrimidine derivatives active against Candida albicans and Aspergillus niger. Now, last, anti-cancer activity. Mustafa et al. and Taylor et al. Synthesized pyrazole-pyrimidine activity against EAC cell line. Now, anti-inflammatory activity. Synthesized pyrazole-pyrimidine derivatives for mice and rodents. Now, reaction scheme. Reaction scheme include phenol hydrazine derivatives react with ethoxymethylene nitrile in the presence of ethanol. It gives intermediate pyrazole derivative which will further react with formic acid, acetic acid, and formamide. It gives pyrazole-pyrimidine derivatives. These are the intermediate compound in which some compounds show molecular formula, molecular weight, RF value, and percentage it. Now these are for the synthesized substituted pyrazole-pyrimidine derivatives. These are the characterization in which I have done NMR, mass, and IR of 5 pyrazolopyrimidine derivatives. Now for anti-inflammatory activity, I have done anti-inflammatory activity and analgesic activity by carrageenan-induced rat cow method. In this, I have done by using 0.1% CMC solution and carrageenan injected was subcutaneously in left hand paw of rats. By formula, percentage inhibition of edema is equal to volume of edema for control minus volume of edema for test and standard, and 200 upon volume of edema for control. These are the percentage inhibition of synthesized compound. Analgesic activity. It was done by the tail immersion method was used to determine the analgesic activity. Here I was used rats of viscous strain by randomly used 6 groups of 6 animals in each. And the temperature of organ bath was set as 55 plus minus 0.5 degrees Celsius. The time taken, observation time of interval was 0, 30, 60 and 120 minutes. These are the data. I have done allergenic activity for confirming the toxicity of the pyrazolopyrimidine derivatives. Here I was used albino rats of 3 groups, and here making 3 groups, group 1, group 2, group 3, in which we have taken 0.5 ml of CMC as a vehicle, controlled the diclofenac solution, and after that all sacrificed and examined by means of magnifying lens 10 into 10x. These are the synthesized compound. Now conclusion, in which all of the compound PP3B, PP2C and PP5C is active against anti-inflammatory activity, analgesic activity, PP5A, PP4C and PP2A. Now the references. Thank you, sir.
Thank you so much, ma'am, for your valuable presentation, Dr. Sudha. So I would like to call Divya Dilip Kumar to share the screen and proceed with your presentation. Ms. Divya?
Yes, sir. Are you able to hear me, sir?
Yes, I can hear you. Please start sharing the screen.
1 second, sir. So are you able to see the screen?
Yes.
Yeah, it's visible.
Yes, sir.
Yeah, it's visible.
Yes, sir. Good evening, everyone. I am Dr. Divya Dilip Kumar, PhD scholar and a professor at SRM Kattanguruthur Dental College, Tamil Nadu, India. My presentation today is about the efficacy of Pudina Mentha arvensis.
இது வந்து எனக்கு வந்து எனக்கு போஸ்ட் பிரசன்டேஷன் இருக்கு போஸ்ட் பிரசன்டேஷன்.
Hello.
Sorry, ma'am, you can proceed.
Yes, sir. My presentation is about the efficacy of Pudina Mentha arvensis on Candida albicans and Lactobacillus acidophilus, which is an in vitro study. Orthodontic appliances act as an active site for the attachment of food particles and subsequently microorganisms which later cause cavity and periodontal disease. And the patients with orthodontic brackets often have a large number of retention sites for plaque accumulation. Many plants exhibit potent antimicrobial activity against various microorganisms, and they have no side effects and act against and modulate the factors that are crucial for microbial survival or the microbial activity. Pudina, which is the Mentha arvensis, a perennial aromatic herb belonging to the family Labiata and genus Mentha, is an important culinary plant with immense medicinal use. The antibacterial effect of Pudina has already been proven against many microorganisms, but the anti-candle effect of Pudina has not been studied yet. So my aim of the study was to evaluate the efficacy of methanolic extract of Pudina on Candida albicans, which is most common microorganisms that is seen in orthodontic patients, and Lactobacillus acidophilus. The significance of the study is that Lundstrom et al. Has shown that there's a significant increase in colonization of Streptococcus mutans and Lactobacillus in orthodontic patients. Ibino et al. Studied and found that there's a significant increase in Candida albicans level in oral cavity of orthodontic patients. Paisam et al. Has shown that there's an increase in Streptococcus mutans, Lactobacillus, and Candida albicans in orthodontic patients. The antibacterial effect of Pudina has been proved against Streptococcus mutans. Hence, this study is to evaluate the antibacterial effect and the anti-candid effect of Pudina and on Lactobacillus and Candida albicans. Pudina powder was obtained from pudina leaves, which was then air-dried, and the different concentration of pudina extract was prepared. 100 grams of finely powdered pudina was then macerated with 500 ml of 100% of methanol. Methanolic extract was then prepared and subjected to filtration with Wattman filter paper to obtain a clear filtrate. The filtrate so obtained was reduced to low temperature of less than 60 degrees to obtain a solid residue of the pudina extract. From 100 grams of pudina powder dissolved in 500 ml of methanol, 5 grams of residue was obtained, so the yield was 5% by weight. 0.5 grams of extract was then dissolved in 10 ml of dimethyl formamide to obtain 5% of concentration of extract. Similarly, 1 gram of extract was dissolved in 10 ml to obtain 10% concentration, and the same procedure was made for 50% of concentration. 0.2% of chlorhexidine was used as a positive control, and dimethyl formamide was used as a negative control. Now, the cell suspensions with 10^8 cells were prepared using Lactobacillus acidophilus and Candida albicans. Using sterile microtips, wells were made, which was about 7 to 8 millimeters in diameter. Lawn culture was made on appropriate media. Then the preparation with different concentrations were added on to the wells and observed for zone of inhibition after incubation at 37 degrees Celsius for Lactobacillus acidophilus and 25 degrees Celsius for Candida albicans. The results obtained were that for the Candida albicans zone of inhibition, for 5%, there was no zones of inhibition. At 10% concentration, there was 14.5 millimeters of zone of inhibition. And for 50% concentration, there was 16.25 millimeters of zone of inhibition. Whereas for Lactobacillus acidophilus, there was no zone of inhibition seen in any of the 3 concentrations. So this study has confirmed that the pudina has antimicrobial activity against Candida albicans with the presence of inhibitory zone. And it does not have any effect on lactobacillus. The study is the first of its kind. Further, the composition of pudina extract at 20% might be such that it has a minimum antimicrobial potential. This study can be further extended by increasing the concentration of pudina. I conclude by saying that the pudina extract demonstrated an antimicrobial activity against Candida albicans. Hence, pudina can be used as a mouth rinse for orthodontic patients instead of using chlorhexidine, which is a chemical agent, and which also has a disadvantage of discoloration of the teeth. Further studies by increasing the concentration can also be done to see whether there's an increase in the zones of inhibition. My references. Thank you.
Thank you so much, Ms. Divya Dilip Kumar. Session chairs or participants can raise any queries to the chat box. So I would like to request the previous participants one more time. Ms. Anitha Thomas.
Yes, sir.
Can you please start sharing the screen and present?
Okay, sir. Thank you, sir. Sir, am I audible, sir?
You're audible, ma'am. Please share the screen.
Sir, I have already shared my screen. It's visible, sir?
No, it's not. You have not shared the screen, ma'am. Click on the green color share screen, then click the tab which you want to share and then share.
Okay, sir.
Just move your cursor. There will be share screen option. You click on that green option.
Yes, sir.
And whatever the screen you want to share, click on that, and then you can see an option called share. Click that. Yes, sir, now it is.
Sir, is it visible, sir?
Yes, visible, ma'am. You can proceed.
Okay, thanks, sir.
Make it full screen mode. Yeah.
Sir, is it visible, sir?
Yeah,
Shall I
yeah. You can
Okay,
continue.
thanks. Good evening, everybody. I am Anita Thomas, Assistant Professor of Pharmacy, Government Medical College, Thiruvananthapuram, Kerala. And welcome to my presentation on co-crystallization, an emerging technique for physicochemical property modulation of pharmaceuticals. The drug discovery has changed dramatically during the last decade. The development of combinatorial chemistry has introduced methods to synthesize massive numbers of new diverse compounds. This, along with high throughput screening techniques, make it possible to examine the potency of new compounds. But this kind of approach is highly successful in the discovery of new structures with a super in vitro potency, but often ignores issues of physicochemical properties like solubility, bioavailability, stability, hygroscopicity, etc. Poor aqueous solubility is likely to give rise to increased formulation difficulties. About 60 to 70% of newly discovered compounds have low solubility, and these poorly soluble drugs have limited absorption and low bioavailability. Enhancement of solubility is a major challenge among pharmaceutical researchers. Traditionally, drugs have been classified into 4 by BCS classification system. Among these, BCS class 2 and 4 have low solubility. And BCS class 2 drugs, even though they are highly permeable, their low solubility leads to decrease in low bioavailability. So there are some methods. There are some common methods to improve solubility. They are by physical modification like particle size reduction, modification of crystal habits, drug dispersion in carriers, complexation by using complex agents, solubilization by surfactants, and there are chemical modifications like salt formation, derivatization, and there are other methods like co-crystallization, hydrotrophy, etc. Among each technique has its own merits and demerits. Among all these, co-crystallization approach is unique as it does not affect pharmacological properties of the drug. Co-crystallization can also be used as an alternative approach based on crystal engineering to enhance specific physicochemical and biopharmaceutical properties of active pharmaceutical ingredient when approaches to salt or polymorph formation do not meet expected targets. Co-crystallization can be effectively used to modulate solubility and dissolution, and it can also be used for modulation of physicochemical properties and enhancement of bioavailability, increases stability, and for in case of hygroscopic drugs, hygroscopicity can effectively decreased by co-crystallization, and morphology of an API can be modulated by using co-crystallization, and co-crystallization maintains intrinsic activity of the drug molecule. The solid forms, multi-component solid forms, crystalline solid forms are examples are hydrates, salts, co-crystals, solvates, etc. This co-crystals often has a definite crystal active, so they have increased stability. In case of solvates, hydrates, etc., they incorporate solvent or water molecules. And salt incorporate a counter ion or salt anion or cationic forms. These co-crystals are crystalline materials composed of 2 or more molecules in a crystal lattice, generally in a fixed stoichiometric ratio. Pharmaceutical co-crystals are multi-component crystalline compounds comprising of an active pharmaceutical ingredient and 1 or more unique pharmaceutically acceptable conformers in stability and dissolution rate without compromising the structural integrity of API. And so the bioactivity of the molecule does not affect by co-crystallization. Co-crystals can be considered advantageous in following situations, like if the drug molecule lack ionizable functional groups. So in that cases, these drug molecules can be, the properties of the drug molecules can be tuned by forming co-crystals. And in case of compounds which have particularly sensitive groups to treatment of acids or bases, they also can be effectively formed as a co-crystal. And moreover, there are large number of neutral gas molecules generally regarded as safe compounds as compared to counter ions to make pharmaceutical salt. Co-crystallization often overcomes problems in filtrability, hygroscopicity, stability, etc. Co-crystallization, for co-crystallization, a drug or API is co-crystallized treated with co-formers, and this co-crystallization modulates or alters the crystalline packing of the material. So the physical property, physiochemical property of the co-crystal is entirely different from that of the parent API. A design of co-crystals. Co-crystals can be designed by using crystal engineering and supramolecular synthesis. Interaction of API and co-formers is non-covalent, so that the structural integrity of the API is maintained. And the interaction is like hydrogen bond, van der Waals interaction, pi-phase stacking. According to Cambridge Structural Database, about 80% of non-co-crystals are hydrogen bonded. And co-formers are usually selected from DAS list, generally regarded as safe list. And some examples of co-formers are nicotinic acid, bipyridine, para-amino benzoic acid, succinic acid, adipic acid, saccharin for hydroxybenzoic acid, etc. Often hydrogen bond donor or acceptor functional groups in API forms synthones with the complementary functional groups in co-former, leading to formation of co-crystals. These are examples of homo or heterosynthones. In first case, the acid functional groups in the API and the same acid functional groups in the co-formers form homosynthones. And amide homosynthones. In case of acid pyridine, acid amide pyridine, amide acid, amide N oxide, etc., they form heterosynthones. Probability of co-crystal formation can be predicted by hydrogen bond propensity by a pKa rule, presence of synthones in API and co-formers, by Hansen solubility parameters and computational methods. For hydrogen bond propensity, it is the availability of best hydrogen bond donors and acceptors in an API and co-formers. In by syntonic engineering, the selection of co-formers which are capable of forming supramolecular synthones with API can be determined. And delta pKa less than 3, delta pKa between the API and the co-formers. If it is less than 3, it is a prerequisite for co-crystal formation. By Hansen solubility parameters, we can determine the miscibility of API co-formers. This would in turn predict co-crystal formation. There are several computational softwares like Cosmo, RS, etc. By this approach, we can find the appropriate co-crystal forming phase. And there are a lot of methods by which we can prepare co-crystals. First one is solution based. Slow evaporation of co-crystal.
Try to conclude, madam.
Co-crystal.
Any experimental result, focus and conclude. Yeah.
Pardon, sir?
If any experimental result analysis, you focus on that and try to conclude.
Okay, sir. Methods of preparations like solution-based grinding methods, with anti-solvent addition, gel methods, supercritical fluid technology methods, etc. Characterization of co-crystals can be done by IR spectroscopy, Raman spectroscopy, single crystal XRD, powder X-ray diffraction, differential scanning calorimetry. Differential thermal analysis and scanning electron microscopy can be done by using to know the morphological characters of API, and the elemental analysis can be determined to know the elemental composition of the co-former and API. This by evaluation of co-crystals can be done by studying its solubility, dissolution, bioavailability, photosensitivity, stability studies, and dose response relationship. And co-crystals can be effectively applied for the enhancement of physicochemical property of API due to the presence of co-formers present. The co-crystal generate a diverse array of solid state forms of API that lack ionizable functional group. Formation of co-crystals often shortens time period for drug development. Co-crystals have usually enhanced solubility, thus have enhanced bioavailability. And pharmaceutical co-crystals possess a high potential for API, physicochemical, and biopharmaceutical property enhancement while preserving the pharmacological properties of API. Since co-crystallization emerges as a novel technique for modulating physicochemical properties of drug, it finds wide acceptance among pharmaceutical researchers. It also gives an opportunity to patent new solid forms of API, increasing the intellectual property protection over a particular drug product, reducing the risk of costly litigation and market erosion. And these are the examples of some marketed formulations. Tegretol, Prozac, Sporanox, Viagra, etc. are the marketed formulations of pharmaceutical co-crystals. Thank you, sir.
So much, ma'am, for your valuable presentation.
Thank you, sir.
I request the next participant, the previous participant, Ms. Parkavi M, to present the screen. You can stop sharing the screen, ma'am. Ms. Parkavi, can you please respond?
Yes, sir, I'm here.
Kindly share the screen.
Okay, sir.
Ms. Parkavi, can you share?
Wait, sir.
First click on share screen, then click on your slide, then share it.
It's visible, sir?
Yes, ma'am, it's visible now. You can present the screen and you can start.
So myself, I'm Parkavi. I'm doing M.Pharm Pharmaceutical Chemistry in JKK Nataraja College of Pharmacy. Here I'm present a poster presentation on the basis on in silico studies. For in silico method, I selected a compound coumarin, which is synthesized by using resorcinol with ethyl acetoacetate in presence of sulfuric acid. It gives a coumarin. After that, I used a primary amine like aniline. To that, it produced the compound 7-hydroxy-4-methyl coumarin. A coumarin, it is an important structural moiety. It is present in variety of natural and synthetic products. It gives a variety of biological activities also, and a rapid synthesis of coumarin derivatives by pigment condensation reactions and microwave radiation. I prefer microwave radiation because of time consuming, and also it gives a good yield one. And after that, I go for characterization for IR prediction, then proton NMR, then 13 NMR, 13C NMR, and mass. Then I go for prediction of some softwares like Swiss ADME, mold inspiration, fast prediction. Likewise, I go. First, in mold inspiration, I predict biological activities and bioactivity scores also. Gives good yield and good bioactivity score also. Then in fast prediction, it gives almost an anti-neoplastic activity and also an CVS activity. Then in Swiss ADME, I predict a physiochemical and biological activity of a drug and also a boiled egg model. Then radar model also. In radar model, it gives an the drug's lipophilicity character, insaturation character, and polarizability, then flexibility. In boiled egg model, suppose if drug is produce an blue spot,
means it will produce an activity and it gets efflux out. If it is in red spot means it will produce an activity and it does not excrete out. It present inside and it produce an toxicity. Here in boiled egg model, the white color is represents a GIT, a gastrointestinal part. Then yellow color, it represents a CNS. That is, a drug will enter drug spots in white space means a drug will enter a GIT. If a drug spot in yellow color means a drug will penetrate a CNS. On the basis of coumarin, I have designed 40 models. Out of that, I represents two. One is E5 molecule and E15th molecule. The yield of E5 molecule is 62.93. Then E15 is 92.86. Then IR prediction, proton NMR prediction, C13 NMR, and mass. All I have represent in table. Then in silico properties, this is an pass prediction. In a score wise, I represent in a table for E5 fifth molecule. E5th molecule means it is chlorocoumarin. Then I predict using model inspiration and ADMET toolkits and pharmacokinetic profiles were investigated. The results shown good gastrointestinal absorption, very low blood-brain barrier permeability, low toxicity, and high bioavailability. Then a software package I have represented. First, I draw my structure in ChemSketch. Then I go for smile. I predict smiles and I represent in mold inspiration. That link I have represented, then pass prediction, then Swiss ADME. These four software I used and I predict properties. Then conclusion, by using in silico drug designing, we can produce an active lead molecule from the preclinical discovery stage to late stage clinical development. The lead molecules which are developed will help us to selection of only potent molecules leads to cure particular diseases. Further studies are warranted to check the efficiency of these molecules by various in vitro and in vivo methods and elucidation of mechanism of action. That I conclude, then references are there.
Thank you.
Thank you, sir.
Okay, you can stop sharing. Yeah, we can go for next.
Thank you so much,
Yeah, Dr.
Ms. Bhargavi, for your valuable presentation.
Anybody?
So with this, we have completed. I would like to call the rest of the participants who have not participated so far, like Mr. Simachal Panda. So Simachal Panda, are you available?
Mr. Simanchal Panda.
Can you hear us, Mr. Simachal Panda?
Not available, I think. You can check from the list
Mr.
of
Roshni Singh. Mr. Roshni Singh, are you available? Mr. Roshni Singh?
Dr. Siniket, they are not in the list, I think, in the connected list.
Absolutely, sir. They are not in the list, but I would like to.
Yeah.
Sometimes what they are doing is, no, sir, they are connecting with other names. That's the reason I'm just trying to call them again.
Yeah.
If anyone wants to share, they can share their thoughts. I would like to call Mr. Santosh Badkaria. Mr. Santosh Badkaria, are you present?
Whoever they are connected to us, if not presented earlier, they can respond and present their presentation.
Yes, absolutely. People who haven't, who are online but not shared or not approached now. So Shakuntala, Ms. Shakuntala, are you available? Mr. Subhash Sharma. Sir, Alifia Yusuf. Mr. Ishtiaq Jilani. Mr. Ishtiaq Jilani, can you present? Are you available?
Yes, I can hear you.
Mr. Ishtiaq, can you please start sharing the screen and proceed?
Yeah, shall I proceed with the presentation?
Yes, please. Thank you so much.
Yeah, just a moment, please. Well, is that visible? Can you
Yes, it's
see and
visible.
hear
It's
me?
visible. You can proceed.
Okay, thank you so much. So hello, everyone. Well, I'm very grateful to represent my research data through this poster at this quality research conference. And I'm Ishtiaq Jilani, and a PhD student from University of Toyama, Japan. Well, so this poster will describe new approaches to the synthesis of natural dibenzoalpha-pyrrone compounds. We used a palladium catalyzed biaryl coupling reaction of phenyl benzoate derivatives to make the 6H dibenzo-BD-pyrrone 6-on ring system to synthesize such compounds. We took the advantage of success of a palladium catalyzed coupling reaction for such compounds, which our group had previously identified. Separation and identification and isolation of such compounds through natural source is quite challenging and expensive task. So we proposed a synthetic scheme to overcome the obstacles. So let's go through the abstract of this research poster. A variety of natural compounds, natural products which possess the 6H dibenzo-BD-pyrrone 6-on core has attracted our interest because of their unique biological activities. A palladium-mediated intramolecular aryl-aryl coupling reaction was employed to the total synthesis of bioactive natural products, iodindyl C and urolithin C3 glucuronide, which possess 6H dibenzo-BD-pyrrone 6-on skeleton. These compounds are known to be naturally occurring bioactive compounds that possess a potential biological activities such as antifungal, antibacterial, antioxidant, larvicidal, and anti-inflammatory, as well as anti-cancer properties. Keeping in view the indispensable biological values of these compounds, we took the advantage of our established strategies to chemically synthesize hyalodendryl C and urolithin C3 glucuronide. We reported several natural product synthesis using the palladium catalyzed intramolecular biaryl coupling reaction with phenyl benzoate derivative for forming the 6H BD-pyrrone ring system. Utilizing this transformation, we planned the efficient synthesis of hyalodendryl C and urolithin C3 glucuronide. So here we show the primary sources and biological values of such compounds. So we can see pomegranates and walnuts are the primary sources of all these compounds. Also, these are extracted and isolated from endophytic funguses also. Well, however, there are some reports that show these compounds are also synthesized by intestinal bacteria and they show a variety of biological activity. So below this, there is a general scheme in which we have used palladium ligand and a base. And here we can see the coupling reaction. And below that, there is a simple mechanistical aspect of the palladium catalyzed biaryl coupling reaction. So on the left-hand side, there is a scheme of esterification between phenol derivative and corresponding benzoic acid derivative by using EDC, HCl, and dichloro-DMAP as a base was successful to yield the key precursor for the palladium catalyzed biaryl coupling reaction. Then we proceeded with the all-important intramolecular biaryl coupling reaction using palladium reagent was significant potential for application to the construction of compound 10, which was subsequently deproducted to yield the final product of the reaction. We examined the intramolecular biaryl coupling reaction using sodium acetate and DMF at 150 degrees Celsius, which produced 10 in a very moderate yield. However, replacing sodium acetate by palladium acetate in presence of silver carbonate at 190 degrees Celsius lead to an improvement in the yield of the final product. And finally, deproduction using 10% palladium carbon in presence of hydrogen gas produced hyaluronidriol C. So in the future, we will continue our efforts in the synthesis of other biologically vital natural products that are very difficult for isolation and separation from the natural sources. So in conclusion, we report the first successful total synthesis of hyaluronidriol C and urolithin C3 glucuronide by making use of palladium catalyzed coupling reaction. So finally, we can see the indispensable biological values of all these compounds, which contain 6H-dibenzobd-pyrrone 6-on-secalitan. So they show anti-allergic, antimicrobial, anti-allergic, antioxidant, and anti-cancer and anti-inflammatory effects. So this is probably all about this research poster. Thank you so much. So I am sticking to the time. So thank you so much for your time.
Thank you so much, Mr. Ishtiaq Jilani, for your valuable presentation. And with this, we have concluded with all the technical participants. And this is the end of the Day 1 conference for the 8th World Conference on Pharmaceutical Science and Drug Manufacturing. I thank all the keynote speakers and the professors, especially Subhash Chandra sir, for your valuable inputs and the support that you have given so far. Thanks so much, everyone. And we will meet tomorrow at 9 a.m. Indian Standard Timing for the Day 2 session of 8th World Conference on Pharmaceutical Science and Drug Manufacturing. Thank you so much, everyone. We will leave now. Thank you so much.
Thank you. Bye.